Deep in the frozen vacuum of space, chunks of ancient ice and rock are hurtling toward Earth at speeds that would vaporize steel on contact. Inside them, locked within crystalline structures older than our Sun, might be the answer to humanity's oldest question. Where did we come from? For decades, scientists dismissed comets as cosmic debris. Dirty snowballs with no real significance beyond their spectacular light shows. Then something changed. When we finally caught up to these objects, when our spacecraft touched them and tasted their chemistry, we found something nobody expected. The building blocks of life itself, fully formed, perfectly preserved, waiting. This isn't speculation anymore. It's data. Hard, repeatable, undeniable data that's forcing us to rewrite the story of life on Earth. Before we had telescopes, before we understood what comets were, they terrified us. Ancient civilizations saw them as omens, disasters written in fire across the night sky. The Romans called them hairy stars because of their streaming tails. Chinese astronomers meticulously recorded their appearances, convinced they predicted the fall of dynasties. Nobody imagined these objects might be delivering something rather than threatening it. The modern scientific view wasn't much kinder. Through most of the 20th century, comets were considered interesting but irrelevant to the story of life. They were leftovers from the solar system's formation, frozen relics that occasionally swooped through the inner planets before vanishing back into the dark. Beautiful, yes. Important to biology, not a chance. That assumption held until we started catching them. In 2004, NASA's Stardust mission did something remarkable.
Guidance is internal. Roll program initiated.
It flew through the coma of Comet Wild 2, that glowing cloud of gas and dust surrounding the nucleus, and collected samples. Not much, just microscopic particles captured in a substance called aerogel, but enough. When those samples returned to Earth 2 years later, the results stopped people in their tracks. Glycine. A simple amino acid, one of the fundamental building blocks that cells use to construct proteins. It was sitting right there in the comet dust, formed in the freezing darkness of space long before Earth even existed. That discovery opened a door. If glycine could form in comets, what else might be hiding in them? The answer came from an unlikely source. In 2014, the European Space Agency's Rosetta mission arrived at Comet 67P, a rubber duck-shaped chunk of ice about 4 kilometers across. Instead of just flying past, Rosetta stayed. It orbited for 2 years, watching the comet wake up as it approached the Sun, measuring everything it could. What Rosetta found wasn't just interesting, it was revolutionary. The comet was outgassing 16 different organic compounds, not simple molecules, but complex carbon-based structures. Formaldehyde, methanol, ethanol, amino acids, and something else. Something that made biologists sit up and pay attention. Phosphorus. An element absolutely essential for life as we know it. It forms the backbone of DNA. It's in every cell membrane. Without phosphorus, Earth-style biology simply cannot function. And here it was, streaming off a comet billions of miles from the nearest living thing. The implications were staggering. These weren't random chemicals. They were the exact ingredients you'd need to start building the machinery of life. But finding ingredients isn't the same as proving they created something. You can have flour, eggs, and sugar sitting on a counter, and they won't spontaneously become a cake. Something has to bring them together under the right conditions. Something has to do the mixing. That's where Earth's early history becomes critical. 4.5 billion years ago, our planet was a molten nightmare. Volcanic hellscape, no stable crust, certainly no oceans. Not exactly a promising environment for delicate organic chemistry. Yet somehow, within a few 100 million years, life appeared. Not gradually, not tentatively, but explosively. By 3.8 billion years ago, we have clear evidence of cellular life already thriving. That timeline has always bothered scientists. It's too fast. Earth barely had time to cool down before biology showed up. Where did all the necessary building blocks come from? How did they organize themselves so quickly? The traditional answer was that Earth made them itself. Lightning strikes in a primordial atmosphere, volcanic vents pumping out chemicals, ultraviolet radiation breaking apart simple molecules and recombining them into complex ones. The famous Miller-Urey experiment in 1953 seemed to prove this was possible. Put some basic gases in a flask, add electricity to simulate lightning, and amino acids appear. Except there's a problem. We got the atmosphere wrong. The Miller-Urey experiment used methane, ammonia, and hydrogen, a strongly reducing mixture that favors organic chemistry. But geological evidence now suggests early Earth's atmosphere was mostly carbon dioxide and nitrogen. Run the experiment with those gases and you get almost nothing. The chemistry doesn't work. So if Earth wasn't making its own organic molecules efficiently, where were they coming from? Look up. Every year, Earth sweeps through debris left behind by comets. We see it as meteor showers, beautiful streaks of light as tiny particles burn up in the atmosphere. But not everything burns. Some material makes it through, settling gently onto the surface. Modern estimates suggest Earth gains about 40,000 tons of cosmic dust annually. That's now, in a relatively clean solar system. 4 billion years ago, during the period we call the Late Heavy Bombardment, the rate was millions of times higher. Picture it. Young Earth, still cooling, oceans just beginning to condense from steam. And it's being absolutely pelted by comets and asteroids, hundreds of impacts every year, each one delivering its cargo of organic molecules. Not just simple stuff, but complex, ready-made building blocks for biochemistry. The entire planet becomes a chemistry lab being constantly restocked with new ingredients. This is where the math gets interesting. Calculate the total mass of cometary material that hit Earth during the first billion years. Factor in the concentration of organic compounds we've measured on real comets. Run the numbers and you get something shocking. Comets could have delivered more organic carbon to early Earth than the planet could possibly have made on its own. By a lot. Maybe by factors of thousands or millions. But quantity isn't everything. You also need the right delivery mechanism. Drop complex organic molecules onto a planet from space and they face a problem. They're moving at cosmic speeds, tens of kilometers per second. That kind of impact generates heat. Lots of heat. Enough to break apart most chemical bonds and destroy exactly the compounds you're trying to deliver. Except comets have a trick. They're not solid rock. They're mostly ice, mixed with dust and organics. When a comet hits an atmosphere, or even a primordial ocean, that ice acts like a cushion. It absorbs energy, vaporizes, creates a shock buffer. Calculations show that organic molecules buried inside cometary ice can survive impacts that would obliterate anything else. The ice protects its cargo all the way down. Even better, that ice itself is valuable. Early Earth needed water. Lots of water. Our planet formed close to the sun, in a region too hot for ice to condense. The water in our oceans had to come from somewhere colder. Comets fit perfectly. They're essentially giant ice cubes that formed in the outer solar system and then migrated inward. Each impact was a water delivery, bringing not just H2O but also everything dissolved in it. We can test this idea chemically. Water molecules come in different isotopes, variations with extra neutrons. The ratio of regular hydrogen to deuterium, heavy hydrogen, works like a fingerprint. Measure it in Earth's oceans and compare it to measurements from comets. If the ratios match, you've found your source. The results are messy. Some comets match Earth's ratio almost perfectly. Others are way off. What this tells us is that Earth's water probably came from multiple sources. Some comets, yes, but also asteroids, and possibly some water that was trapped in the rocks that originally formed our planet. It's a mixture, which actually makes sense. 4 billion years of bombardment meant Earth was sampling everything the solar system had to offer. But we're getting ahead of ourselves. Delivering organic molecules is one thing. Getting them to organize into something alive is something else entirely. The molecules are here. They're scattered across the early Earth's surface, dissolved in the oceans, maybe concentrated in tidal pools or around hydrothermal vents. But molecules don't spontaneously decide to become alive. They need to do something first. They need to replicate. This is the central mystery. Life, at its core, is information that copies itself. DNA holds instructions. Those instructions get read, executed, and then copied to make more DNA. But DNA can't copy itself alone. It needs proteins. Enzymes that unzip the double helix, read the code, assemble new strands. Proteins are molecular machines, and they're incredibly good at their jobs. Except proteins can't make themselves either. They need DNA to encode their blueprints. You see the problem. DNA needs proteins. Proteins need DNA. It's a chicken and egg paradox, except both the chicken and the egg are microscopically small and made of carbon chains. For decades, this looked unsolvable. Then, in the 1980s, scientists discovered something that changed everything. RNA, ribonucleic acid, DNA's simpler cousin. And RNA could do something nobody expected. It could act as both information storage and enzyme. Some RNA molecules, called ribozymes, could catalyze chemical reactions, including, critically, making copies of other RNA molecules. Suddenly there's a way out of the paradox. Forget DNA and proteins for now. Start with RNA. One molecule playing both roles. An RNA world, where these versatile chains are doing all the heavy lifting. Storing information, copying themselves, maybe even catalyzing the reactions needed to build their own components. It's elegant. Almost too elegant. But does it actually work? Test it. Take RNA molecules and put them under conditions that might have existed on early Earth. See if they can evolve, adapt, replicate without any help from modern biology. Researchers have done exactly this. They've created RNA molecules that can copy other RNA strands with reasonable accuracy. They've shown that random RNA sequences, given the right environment, can develop catalytic activity through pure trial and error. The chemistry works. RNA can bootstrap itself. But there's still a massive gap between one self-replicating RNA molecule and anything we'd recognize as alive. That molecule needs protection. It needs to separate itself from the environment, create an inside and an outside. Otherwise, every molecule it makes just diffuses away into the ocean. No concentration, no organization, no evolution. This is where membranes come in. Lipids, fatty molecules with one end that loves water and another that hates it, can spontaneously form bubbles. Vesicles. little spheres with a double-layered skin that keeps the inside separate from the outside. We see this in test tubes. Mix lipids and water and they self-assemble into something that looks startlingly like a cell membrane. No instructions needed. Just physics and chemistry doing their thing. Now imagine an RNA molecule that happens to be inside one of these lipid bubbles. It's replicating, making copies of itself. Those copies are trapped inside with it. The concentration builds. Maybe some of those RNA molecules catalyze reactions that help make more lipids, growing the membrane. The bubble gets bigger. Eventually it might split, dividing into two bubbles, each carrying RNA. You've just invented cellular division without involving a single protein or DNA molecule. This isn't life yet. Not quite. But it's something that can evolve. Bubbles with better RNA that replicates faster or makes useful products, will outcompete bubbles with worse RNA. Selection pressure exists even at this primitive level. And once you have selection, evolution is inevitable. The timeline here is staggering. We're talking about processes that might have taken tens of millions of years. Maybe 100 million. The early Earth had time. Lots of it. And it had variety. Thousands of different environments, each running its own chemical experiments. Shallow pools heated by the sun. Deep ocean vents with their chemical gradients. Underground water systems percolating through mineral-rich rock. Ice formations with liquid pockets. Every one of these is a potential laboratory. What's fascinating is that you don't need everything to happen in one place. Maybe RNA replication gets figured out in one environment, membrane formation in another. Then geological processes, volcanic eruptions, tidal movements, mix everything together. Combine the solutions. Evolution doesn't follow a neat, linear path. It's messy, parallel, full of dead ends and lucky accidents. But here's what really matters. Once you have something that can replicate and evolve, even if it's incredibly primitive, you've crossed a threshold. Because evolution is brutally good at solving problems. Give it enough time and enough variation, and it will find solutions to challenges that seem impossible. Like replacing fragile RNA with more stable DNA, or developing protein synthesis to build better enzymes than ribozymes could ever be. The transition from an RNA world to the DNA protein world we have now probably happened gradually. Maybe DNA started as just a more stable way to store backup copies of important RNA sequences. Then it got better at the job. Eventually it took over information storage completely, while proteins specialized in doing the actual work. RNA got relegated to the role it plays today, messenger, translator between DNA and proteins. We can see echoes of this ancient RNA world in modern cells. Ribosomes, the molecular machines that build proteins, are made mostly of RNA. The energy currency of the cell, ATP, is basically an RNA building block with some extra phosphate groups attached. Even DNA synthesis requires RNA primers to get started. These are fossils, chemical fossils, remnants of a time when RNA did everything. But proving this all happened is hard. RNA doesn't fossilize. It's too fragile. The oldest actual fossils we have are from about 3.5 billion years ago. Stromatolites, layered rock structures built by bacterial mats. By that point, life was already sophisticated. DNA, proteins, cell walls, photosynthesis. The really interesting stuff, the origin itself, happened earlier. Maybe 4 billion years ago, maybe even older, and it left almost no direct evidence. So we're forced to reconstruct it from clues, chemical signatures in ancient rocks, isotope ratios that suggest biological activity, computer simulations of prebiotic chemistry, lab experiments trying to recreate the conditions. Every piece of evidence is indirect, circumstantial. But they all point in the same direction. Life emerged from chemistry, not through some mysterious vital force, but through processes we can understand, replicate, and test. There's one more piece to this puzzle. One that makes the whole thing even stranger. The genetic code. The way DNA sequences get translated into amino acids, which then fold into proteins. This code is nearly universal. Bacteria use it. Plants use it. Animals use it. With tiny variations, every living thing on Earth reads DNA the same way. Which means the code was established early, probably before the last common ancestor of all life. And once it was set, it was locked in. Changing your genetic code would be like rewriting the operating system on a computer while it's running. Possible in theory, but catastrophically difficult in practice. Why this particular code? Is it optimal? Or just a frozen accident, the first solution that worked well enough? Some researchers argue it's actually pretty good at minimizing the damage from mutations. Similar codons often code for chemically similar amino acids. So if a mutation changes one letter in a codon, there's a decent chance the resulting protein still works. That's not random. That's error correction built into the system, but it's also consistent with evolution. An early, slightly error-prone genetic code that worked well enough to get life started. Then, over time, selection pressure refined it. Lineages with codes that handled errors better survived. Those with worse codes died out. What we're left with is the optimized version. Not perfect, but good enough to support the incredible diversity of life we see today. Step back and look at the full picture. 4 billion years ago, maybe earlier, Earth was a planet covered in organic chemistry experiments. Molecules delivered by comets and asteroids, synthesized in the atmosphere, cooked up around volcanic vents. RNA figuring out how to replicate. Lipids forming membranes. Everything mixing, reacting, competing. And somewhere in that chaos, a combination of molecules found a way to persist, to improve, to evolve. That combination was the first life. Not a single moment of creation, but a gradual transition from complex chemistry to simple biology. The boundary between non-living and living wasn't a line. It was a gradient. And we're still not entirely sure where exactly to draw it. Once life existed, even in its most primitive form, the rules changed. Natural selection kicked in. Not as a guiding force, but as an automatic filter. Anything that replicated more efficiently than its competitors spread. Anything less efficient vanished. No intention required. Just math playing out over deep time. The first cells were probably nothing like modern bacteria. Simpler, messier. Their membranes leaked. Their metabolism was inefficient. But they worked well enough to persist. And that's all evolution needs. a starting point. Once you have replication, variation, and selection, improvement becomes inevitable. Not planned, just statistically certain. Early Earth was divided into two broad domains: bacteria and archaea. They look superficially similar under a microscope. Single cells, no nucleus, circular DNA. But genetically, they're as different from each other as either is from us. Different membrane chemistry, different metabolic pathways, different cellular machinery. This split happened early, probably before 3.5 billion years ago. Why it happened is still debated. Maybe different environments selected for different solutions. Maybe it was just chance, two lineages drifting apart. Either way, both domains survived. Both thrived. Both are still here today. For the next 2 billion years, life stayed small. Microbial. Single cells doing their thing in the oceans, in sediments, around vents. It doesn't sound exciting, but this period was crucial. Because microbes invented almost everything: photosynthesis, respiration, nitrogen fixation, fermentation. Every major metabolic pathway we see in complex life today was pioneered by bacteria or archaea. They figured out how to extract energy from sunlight, from minerals, from organic molecules. They built the biochemical toolkit that all later life would inherit. Then, around 2.5 billion years ago, something dramatic happened. Cyanobacteria, a group of photosynthetic bacteria, started producing oxygen as a waste product. Not a little oxygen, industrial quantities, enough to fundamentally alter the planet's chemistry. This wasn't planned. Oxygen was toxic to most organisms at the time. But cyanobacteria had evolved a way to deal with it, and they thrived. Everyone else had to adapt or die. The buildup was slow at first. Oxygen reacted with dissolved iron in the oceans, forming rust that settled to the seafloor. Those deposits, called banded iron formations, are some of the richest iron ore reserves we have today. For hundreds of millions of years, the oceans soaked up the oxygen. But eventually, the iron ran out. Oxygen started accumulating in the atmosphere. This is called the Great Oxidation Event. It fundamentally restructured the biosphere. Most anaerobic organisms retreated to oxygen-free environments. Deep sediments, guts of animals, anywhere they could avoid the toxic gas. But other lineages adapted. They evolved ways to use oxygen for respiration, extracting far more energy from the same nutrients. Aerobic respiration is about 15 times more efficient than anaerobic metabolism. That's a game changer. It opened the door to larger, more complex organisms. But complexity didn't arrive immediately. For another billion years, life stayed stubbornly simple. Bacteria and archaea dominated. No multicellular organisms. No complex cells. Just microbes. Why the delay? We're not entirely sure. Maybe oxygen levels weren't high enough yet. Maybe nutrients were limiting. Maybe it just took that long for the right innovations to evolve. Evolution doesn't optimize for speed. It optimizes for survival. And single-celled life was surviving just fine. Then, around 1.5 to 2 billion years ago, something unprecedented happened. A bacterial cell got engulfed by an archaeal cell. Normally, that's dinner. But this time, the bacterial cell survived inside. It kept its own genome, its own membrane, and it started providing a service. It processed oxygen, generating energy for the host. In return, it got protection and nutrients. This wasn't cooperation. Not at first. More like a hostage situation that gradually became mutually beneficial. Over time, the bacterial cell lost most of its independence. Its genome shrank. Many of its genes migrated to the host's nucleus. But it kept its essential function: energy production. Today, we call these structures mitochondria. Every complex cell on Earth, every plant, every animal, every fungus, has them. They're the descendants of that ancient symbiotic event. Without mitochondria, complex life as we know it wouldn't exist. This wasn't a one-time fluke. It happened again with photosynthesis. An archaeal cell engulfed a cyanobacterium. Same deal. The cyanobacterium survived inside, kept photosynthesizing, became a permanent resident. Today, we call these chloroplasts. Every plant and algae has them. Two separate endosymbiotic events. Two different bacterial lineages incorporated into archaeal hosts. Both absolutely critical for the evolution of complex life. These hybrid cells are called eukaryotes. They're fundamentally different from bacteria and archaea. They have a nucleus, complex internal structures, linear chromosomes, sexual reproduction, the ability to grow large and differentiate into specialized cell types. Every organism you can see without a microscope is a eukaryote. Every tree, every insect, every fish, every human. We're all descendants of those ancient mergers. But even with eukaryotic cells, life stayed mostly single-celled for a long time. Maybe another billion years. Single eukaryotes floating around, eating bacteria, photosynthesizing. Then, gradually, some lineages started sticking together. Colonies formed. Simple at first. Just clusters of identical cells. But over time, cells within colonies began specializing. Some handled reproduction. Others handled feeding. Others provided structure. This is the origin of multicellularity. Not a sudden leap, but a gradual transition from independent cells to cooperative groups. Multicellularity evolved multiple times independently. Plants did it. Animals did it. Fungi did it. Even some bacteria and algae pulled it off. Each time, the pattern was similar. Cells stuck together, started dividing labor, became interdependent. Why? Because in certain environments, being big has advantages. You're harder to eat. You can capture more resources. You can differentiate into specialized structures. Roots, leaves, muscles, nerves. Suddenly, evolution had a whole new playground. By about 600 million years ago, the first complex multicellular organisms appeared in the fossil record. Weird, soft-bodied creatures called the Ediacaran biota. Some looked like quilted fronds. Others like segmented discs. They don't resemble anything alive today. Most of them went extinct without leaving descendants. But they proved that large, complex life was possible. They set the stage for what came next. Then, around 540 million years ago, the Cambrian explosion happened. Not an explosion in the literal sense. More like a sudden diversification. Within about 20 million years, most major animal body plans appeared. Arthropods, mollusks, chordates, echinoderms. Animals with hard shells, jointed legs, segmented bodies, eyes. The fossil record goes from sparse and strange to abundant and recognizable. What triggered the Cambrian explosion? Probably multiple factors. Oxygen levels rising to a critical threshold. Predator-prey dynamics creating evolutionary arms races. The evolution of developmental genes that allowed for complex body plans. Ecological feedback loops where one innovation triggered others. It wasn't a single cause. It was a cascade. Once it started, it accelerated. One of the most important innovations was eyes. The ability to see opened up entirely new survival strategies. Predators could hunt more effectively. Prey could escape more efficiently. Vision created selection pressure for faster movement, better armor, more sophisticated nervous systems. Suddenly, being smart mattered. Brains got bigger. Behavior got more complex. Another key innovation was hard body parts. Shells, exoskeletons, bones. Before the Cambrian, almost everything was soft-bodied. Hard structures provided protection, but also structural support. They allowed animals to grow larger, move faster, dig deeper. And crucially, they fossilized. Which is why the Cambrian fossil record is so much richer than anything before it. Not necessarily because life suddenly became more diverse, but because it became more preservable. From the Cambrian onward, evolution proceeded in recognizable patterns. Animals colonized the oceans, then the land. Plants followed, turning barren continents into forests. Insects evolved, then amphibians, then reptiles, then mammals and birds. Each group building on the innovations of the ones before. Each adapting to new environments, filling new niches. But here's the thing. Every step in this story, from the first self-replicating molecules to the Cambrian explosion, was contingent. Not inevitable. If any number of things had gone differently, life might not exist. Or it might exist in forms completely unrecognizable. The fact that we're here, examining our own origins, is the result of billions of years of accidents, near misses, and unlikely events. Earth could have stayed frozen during snowball Earth periods. Life might never have survived. The Great Oxidation Event could have poisoned everything without any lineages adapting. Endosymbiosis might never have happened. Multicellularity might never have evolved. Any of these failures would have left Earth as a microbial planet indefinitely. Interesting, sure, but not capable of producing anything like us. Yet it didn't fail. Every bottleneck, every crisis, every random event lined up just right. Not because of destiny, just because of probability playing out over unimaginable timescales. Given enough planets, enough time, improbable things become statistically likely. We're the outcome of one very long, very unlikely chain of events. And that chain is still unfolding. The story of life on Earth isn't just about what survived. It's about what almost didn't. About 5 times in the past 540 million years, mass extinction events wiped out most species on the planet. Not small die-offs. Catastrophic collapses where 70 to 95% of all species disappeared. These weren't gradual declines. They were biological apocalypses that reset evolution and forced life to rebuild from whatever happened to make it through. The first major extinction happened at the end of the Ordovician period, about 445 million years ago. At the time, most life still lived in the oceans. Trilobites, brachiopods, burly fish. The continents were barren rock. Then the climate destabilized. A rapid ice age locked up vast amounts of water as glaciers. Sea levels dropped dramatically. Shallow marine habitats, where most life existed, simply disappeared. Organisms adapted to warm, stable conditions suddenly faced freezing temperatures and habitat loss. About 85% of marine species went extinct. But here's what's interesting. The extinction happened in 2 pulses. First, when the Ice Age began. Second, when it ended and oceans warmed rapidly again. Life couldn't adapt to the cold. Then it couldn't adapt when things warmed back up. Whichever way the climate swung, something died. The survivors were generalists. Species that could tolerate wider ranges of temperature, salinity, oxygen levels. Specialists, finely tuned to specific conditions, were wiped out. This pattern repeats in every mass extinction. Adaptability beats optimization when the environment becomes unpredictable. The second major extinction came at the end of the Devonian period, around 375 million years ago. This one unfolded over several million years. Not a single catastrophic event, but a prolonged crisis. The leading hypothesis involves ocean anoxia. Large areas of the ocean losing oxygen. Fish, invertebrates, coral reefs, all suffocating. What caused the anoxia? Possibly massive algae blooms triggered by nutrient runoff from the newly forested continents. plants had just colonized land. Their roots broke down rock, releasing nutrients into rivers that flowed into the ocean. Too many nutrients caused explosive algae growth. When the algae died and decomposed, they consumed oxygen. The oceans became dead zones. About 75% of species disappeared. Reef ecosystems collapsed. Armored fish, which had dominated for millions of years, were devastated. Sharks, more adaptable, survived. This extinction reshaped marine life. It cleared space for new groups to evolve. But it took millions of years for biodiversity to recover. The third extinction is the big one. The Permian-Triassic event, 252 million years ago. The worst mass extinction in Earth's history. About 96% of marine species and 70% of terrestrial vertebrate species went extinct. Life came closer to complete annihilation than at any other time. The cause? Massive volcanic eruptions in what's now Siberia. Not a single volcano. An entire region erupting for hundreds of thousands of years. Lava flows covering an area larger than Europe. The eruptions released enormous amounts of carbon dioxide, sulfur dioxide, and other gases. Global temperatures spiked. Oceans acidified. Oxygen levels plummeted. On land, acid rain and greenhouse heating devastated ecosystems. In the oceans, warming and acidification killed off calcifying organisms. Coral reefs vanished. Food chains collapsed from the bottom up. Some researchers think the warming triggered the release of methane from ocean floor deposits. Methane is a far more potent greenhouse gas than CO2. If enough was released, it would have created a runaway warming effect. Temperatures potentially rose 10 degrees Celsius or more. The planet became a hothouse. Only the most resilient organisms survived. Small body size helped. Lower metabolic demands. Ability to hide underground or underwater. Generalist diets. Life that could eat almost anything had an advantage over specialists. The recovery took 10 million years. Longer than any other extinction. For millions of years after the Permian extinction, ecosystems remained impoverished. Low diversity. Simplified food webs. But eventually, life rebounded. And the groups that survived went on to dominate the Mesozoic. Including the archosaurs, the lineage that would produce dinosaurs. The fourth major extinction happened at the end of the Triassic period, about 201 million years ago. Another round of massive volcanism. This time in the central Atlantic, as the supercontinent Pangaea began to break apart. Rifting continents triggered volcanic eruptions that lasted for hundreds of thousands of years. Again, CO2 levels spiked. Oceans acidified. Temperatures soared. About 80% of species went extinct. But this extinction had an unusual outcome. It cleared the way for dinosaurs. Before the Triassic extinction, dinosaurs were relatively minor players. They existed, but so did many other reptile groups. Crocodile relatives, large amphibians, burly mammal ancestors. All competing for the same resources. The extinction removed most of those competitors. Dinosaurs, for whatever reason, were better at surviving the crisis. Maybe their upright stance. Maybe their respiratory systems. Maybe just luck. But in the aftermath, they diversified rapidly and became the dominant land animals for the next 135 million years. Which brings us to the fifth extinction. The one everyone knows. The End Cretaceous event, 66 million years ago. The asteroid impact that killed the dinosaurs. This one was different. Not a slow burn like the others. Not volcanism or climate shifts playing out over millennia. This was sudden. Catastrophic. A 10-kilometer-wide asteroid slammed into what's now the Yucatan Peninsula at 20 kilometers per second. The impact released energy equivalent to billions of nuclear bombs. It vaporized rock. Triggered earthquakes. Generated tsunamis. Ignited global wildfires. But the real killer was the aftermath. The impact threw trillions of tons of dust and aerosols into the atmosphere. Sunlight was blocked. Photosynthesis stopped. Temperatures plummeted. Plants died. Herbivores starved. Carnivores followed. Within months, ecosystems collapsed. About 75% of all species went extinct, including all non-avian dinosaurs. Not because they were weak. Not because they were poorly adapted. But because the rules changed instantly and completely. No amount of evolutionary success mattered when the sun disappeared. The survivors were small, burrowing animals, scavengers, anything that could hide underground or underwater, anything with low energy requirements. Birds made it through, though most species didn't. Mammals survived, but only the small ones. Crocodiles and turtles, with slow metabolisms, endured. In the aftermath, the planet was dark, cold, and nearly lifeless for years. Then, slowly, life returned. Plants regrow from seeds and roots. Survivors emerged and began to spread. Ecological niches, empty for the first time in millions of years, were filled by whatever happened to be around. Mammals, previously limited to small nocturnal roles, suddenly had opportunities. No more dinosaurs to compete with. No more giant predators. Within 10 million years, mammals had diversified into dozens of new forms. Browsers, grazers, predators, climbers, swimmers. The age of mammals had begun. Not because mammals were superior, but because dinosaurs were gone. Each of these 5 mass extinctions reset the clock. They didn't erase life, but they reorganized it. Groups that had dominated for millions of years vanished. New groups, previously marginal, took over. Evolution doesn't have a direction. It doesn't move toward complexity or intelligence. It just responds to whatever pressures exist at the time. And when those pressures are catastrophic, the response is equally extreme. Survival in a mass extinction isn't about being the best. It's about being lucky enough to have the right traits at the right moment. Small size when food is scarce. Generalist diet when ecosystems collapse. Burrowing behavior when the surface becomes uninhabitable. These aren't strengths in normal times. They're insurance policies for disasters. And here's the uncomfortable part. We're living in the 6th mass extinction right now. Species are disappearing at rates comparable to the Big Five. Not from asteroid impacts or volcanic eruptions. From us. Habitat destruction. Climate change. Overhunting. Pollution. We're not just witnesses to extinction. We're the cause. And unlike previous extinctions, this one is happening in real time. Fast enough to observe. Fast enough to measure. But possibly too fast to stop. The difference is, we know it's happening. We can see the patterns. We understand the mechanisms. Previous mass extinctions were blind cosmic accidents. This one is optional. Which makes it either more tragic or more hopeful, depending on what we do next. The question isn't whether Earth will survive. It will. Life has endured worse. The question is what form that life will take. Weather will be part of it. And what we're willing to do to change the trajectory. Earth's history isn't just a record of extinctions. It's a record of recoveries. And those recoveries follow patterns. Predictable, repeatable patterns that play out over millions of years. Understanding them reveals something crucial about how life actually works when everything falls apart. After each mass extinction, the planet enters what scientists call a dead zone. Not literally dead, but functionally empty. Food webs are shattered. Ecosystems lack structure. Energy flows are disrupted. The survivors exist, but they're not thriving. They're just hanging on in a world that doesn't work properly yet. This phase lasts anywhere from hundreds of thousands to millions of years. During this time, diversity remains low. A few generalist species spread widely, filling multiple roles because specialists haven't evolved yet. Think of it as ecological simplicity. Not because life can't be complex, but because complexity requires time to build. Then something shifts. Resources accumulate. Populations grow. Competition increases. And suddenly, evolution accelerates. New species branch off. Specialists emerge. Food webs gain layers. This is the recovery phase, where diversity explodes and ecosystems regain their structure. But here's what matters. The new ecosystems don't look like the old ones. They're built from different parts, organized around different principles, dominated by different groups. Recovery doesn't mean restoration. It means reinvention. After the end Permian extinction, the worst in Earth's history, recovery took roughly 10 million years. Longer than any other mass extinction. Why? Because the damage was deeper. Ocean chemistry was disrupted. Carbon cycles were broken. Climate remained unstable for millions of years. Life had to wait for the planet itself to stabilize before it could rebuild. The Triassic period that followed looked nothing like the Permian. Synapsids, which had dominated before, were reduced to minor roles. Reptiles, particularly archosaurs, took over. These were the ancestors of dinosaurs and crocodiles. They succeeded not because they were superior, but because they happened to have traits that worked in new conditions. After the end Cretaceous extinction that killed the dinosaurs, recovery was faster. About 2 to 3 million years before ecosystems regained complexity. Why quicker this time? The planet wasn't as severely damaged. Ocean chemistry recovered faster. Climate stabilized sooner. And crucially, more lineages survived, providing more raw material for evolution to work with. Mammals filled the empty niches left by dinosaurs. But they didn't just become small versions of what came before. They evolved entirely new body plans, new feeding strategies, new ways of moving and reproducing and interacting. Within 10 million years, mammals had diversified into forms ranging from tiny shrews to early whales. This reveals a fundamental truth about evolution. It's not progressive. It doesn't aim toward improvement. It's opportunistic. Whatever works in the current environment spreads. Whatever doesn't, disappears. The best organism is simply the one that survives long enough to reproduce. And survival depends heavily on luck. Being in the right place when disaster strikes. Having the right traits when conditions change. Being part of a lineage that happens to persist. Intelligence doesn't guarantee survival. Size doesn't. Strength doesn't. Adaptation to current conditions definitely doesn't, because those conditions can change overnight. This is why predicting recovery is so difficult. We can identify general patterns, but the specifics depend on which groups survive and what opportunities they find. After the dinosaurs, it could have been something other than mammals. If a few key lineages had gone extinct, Earth's history would look completely different. Now consider our current situation. We're in the early stages of a mass extinction. Species are vanishing rapidly. Ecosystems are degrading. But we're not yet in the dead zone. That comes later, if current trends continue. Which means we're at a decision point. We can still influence the outcome. What would recovery look like after a human-caused mass extinction? Hard to say. It depends on what survives. If large mammals disappear, something else will eventually fill those roles. Maybe birds. Maybe reptiles. Maybe something unexpected. Evolution doesn't care about our preferences. If humans disappeared tomorrow, Earth would recover. It might take millions of years, but diversity would return. New species would evolve. Ecosystems would rebuild. The planet has done this before, multiple times. It's resilient in ways that individual species aren't. But that's not a comforting thought. Because from our perspective, recovery after our extinction wouldn't include us. We'd be a thin layer in the fossil record. Evidence of a species that emerged, dominated briefly, then vanished. Just like the dinosaurs. Just like the trilobites. Just like thousands of other lineages that once seemed permanent. The real question isn't whether Earth will recover. It's whether we want to be part of that recovery. Whether we're willing to change our behavior to remain part of the story. Because unlike previous mass extinctions, this one comes with awareness. We understand what's happening. We can measure it. We can model future scenarios. We have knowledge that no species has ever possessed before. That knowledge creates responsibility. Not moral responsibility in some abstract sense, but practical responsibility. We're causing changes that will determine which species persist and which don't. Including possibly our own. And unlike an asteroid impact, these changes are gradual enough that we can respond. If we choose to. But here's the hard part. Responding effectively requires acting against immediate self-interest. Preserving habitat means not developing land. Reducing emissions means changing energy systems. Limiting consumption means accepting less material wealth. These aren't easy choices. They're not even obviously rational from an individual perspective. Yet they're necessary from a species perspective. From an ecosystem perspective. From a long-term survival perspective. The same perspective that matters when you're looking at geological timescales and extinction patterns. Because on those timescales, short-term gains vanish. Only persistence matters. Only what survives long enough to leave descendants. This is where human intelligence becomes either our greatest asset or our fatal flaw. We can understand long-term consequences in ways other species can't. We can plan beyond our own lifetimes. We can coordinate globally. But we can also rationalize inaction. Prioritize immediate comfort. Assume someone else will solve the problem. Other species don't face this dilemma. They can't choose to prevent extinction. They simply respond to immediate pressures, and either their traits work or they don't. Humans are the first species capable of consciously deciding whether to alter our trajectory. Whether to use our intelligence to ensure our persistence. The fossil record shows that dominance is temporary. Every group that once ruled eventually declined. Trilobites dominated for 270 million years, then vanished. Dinosaurs lasted 165 million years, then disappeared. humans have existed for maybe 300,000 years. We've been technologically advanced for less than 10,000. We're newcomers. And newcomers don't have track records of stability. What we do have is unprecedented power. Power to reshape ecosystems. Power to drive climate change. Power to cause extinctions at rates comparable to asteroid impacts. That power doesn't make us immune to extinction. It makes us capable of causing our own. The patterns from previous mass extinctions suggest what might happen next. If we continue current trends, diversity will decline. Ecosystems will simplify. Specialist species will disappear first. Then larger species. Then anything requiring stable conditions. Eventually, we reach a threshold where ecosystems can't provide the services we depend on. Clean water. Stable climate. Pollination. Soil formation. Food production. At that point, human civilization becomes unsustainable. Not because we lack technology, but because the biological foundation collapses. We can't engineer our way out of ecosystem failure. We depend on functional ecosystems the same way every other species does. Remove enough pieces, and the whole structure falls apart. This isn't speculation. It's pattern recognition. Every mass extinction followed similar trajectories. Stress accumulates. Systems become fragile. A final trigger causes collapse. The only difference now is we can see it coming. We have data. Models. Projections. We know which actions accelerate the problem and which might slow it. Whether we act on that knowledge determines what the next few million years look like. Whether the Anthropocene becomes a brief anomaly or a major extinction event. Whether humans persist or become another fossil layer. Whether intelligence, our defining trait, proves adaptive or maladaptive on evolutionary timescales. The answer isn't predetermined. Evolution doesn't script outcomes. It responds to conditions. And conditions, for the first time in Earth's history, are partially under the control of one species. That's never happened before. We don't have precedent for a species consciously shaping its own evolutionary future. Which makes this moment unique. Not just in human history, but in planetary history. We're the first species capable of understanding mass extinctions while potentially causing one. The first that can choose whether to continue down that path. The first with enough knowledge to know better and enough power to do something about it. What we do next matters. Not because the universe cares. Not because Earth cares. But because we care. Because we're capable of caring about things beyond our immediate survival. About other species. About future generations. About whether the story continues or ends with us. That capacity for long-term thinking is either our salvation or our epitaph. And we're in the process of finding out which. Looking back at the fossil record reveals something unsettling. Intelligence rarely appears as a survival trait. Most successful lineages, the ones lasting tens or hundreds of millions of years, rely on simplicity. Bacteria, jellyfish, crocodiles. They don't solve problems. They persist through durability. Intelligence is expensive. It requires large brains, extended development periods, high energy intake. For most of Earth's history, those costs outweighed the benefits. Only in specific circumstances, environments demanding flexibility and learning, did intelligence become advantageous. And even then, it remained rare. Out of billions of species across 4 billion years, only one developed technology. That's not a sign of inevitability. It's statistical noise. Which raises a question. If intelligence is so rare and so costly, what happens when it creates problems faster than it solves them? When the very trait that enabled survival becomes the mechanism of decline? We're testing that now. Human intelligence produced agriculture, medicine, technology. It extended lifespans, reduced infant mortality, enabled population growth beyond any precedent. But it also produced weapons capable of ending civilization in hours. Pollution that persists for millennia. Climate disruption that compounds across decades. We're intelligent enough to create these problems. Whether we're intelligent enough to solve them remains unproven. And the clock is running. Not in human time, where decades feel long. In geological time, where our entire civilization is a blink. If we disappeared tomorrow, future geologists would find a thin layer marking our existence. Plastics, concrete, radioisotopes, a brief spike in carbon isotopes, then silence. The planet would continue. Life would recover. It always does. Not the same life, not the same ecosystems, but something. Earth doesn't need humans. We need Earth. That asymmetry matters. We talk about saving the planet, but that's backwards. The planet will be fine. It survived 5 mass extinctions, supervolcano eruptions, asteroid impacts, ice ages, hothouse climates. It'll survive us. What we're actually trying to save is ourselves. Our way of life. The stable conditions we evolved to exploit. Those conditions aren't guaranteed. They're contingent on systems we're actively destabilizing. And once those systems cross certain thresholds, they don't snap back. They reorganize around new equilibria. Equilibria that might not include industrial civilization or large human populations or humans at all. That possibility should focus attention. Because unlike previous species facing extinction, we can see it coming. We have data showing ocean acidification rates, temperature projections, biodiversity decline curves, ecosystem collapse timelines. We know where current trajectories lead. And yet, Knowing doesn't guarantee action. Information alone never changes behavior. If it did, everyone would exercise, save money, avoid processed food. But knowing what's good for us and doing it are different things. The gap between knowledge and action defines the human condition. And right now, that gap is widening. We know fossil fuels drive climate change. We keep burning them. We know industrial agriculture depletes soil. We keep expanding it. We know overfishing collapses stocks. We keep fishing. Not because we're stupid. Because short-term incentives override long-term consequences. Because individual rationality produces collective irrationality. Because the benefits of harmful actions accrue to individuals while the costs distribute across everyone. That's not a moral failing. It's a structural problem. And structural problems require structural solutions. Individual action matters, but it's insufficient. Recycling, reducing consumption, changing diet, all positive steps. But they don't address the underlying systems driving ecosystem collapse. Those systems, energy infrastructure, agricultural practices, economic incentives, require coordinated policy changes. Which requires political will. Which requires public pressure. Which requires enough people caring enough to demand change. And that's where we are. In a narrow window where the trajectory is set but not locked. Where the momentum is strong but not irreversible. Where outcomes remain uncertain because they depend on choices not yet made. This moment won't last. Windows close. Systems reach tipping points. Momentum becomes irreversible. The question is whether we recognize the moment while we're in it. Whether we understand that what feels like normal is actually unprecedented. That the stability we take for granted is historically anomalous. That the future isn't guaranteed to resemble the past. Because it won't. The planet we're creating will be fundamentally different from the one we inherited. Hotter. Less biodiverse. Less stable. Whether it remains habitable for billions of humans depends entirely on how much hotter, how much less diverse, how much less stable. Those aren't abstract variables. They're outcomes of specific decisions. Energy policy. Land use. Consumption patterns. Trade agreements. Infrastructure investments. Decisions being made now, today, by governments, corporations, individuals. Each decision moves the needle. Slightly. Incrementally. But cumulatively, they determine trajectory. And trajectory determines destination. We like to think we're separate from nature. That technology insulates us from ecological limits. That intelligence exempts us from evolutionary pressures. But we're not separate. We're embedded. We breathe the same air. Drink the same water. Depend on the same ecosystem services as every other organism. Our cities, Our technology, our entire civilization rests on a foundation of functional ecosystems. Remove that foundation and everything collapses. Not might collapse. Will collapse. It's physics. You can't support a structure after removing its base. And we're systematically removing the base. Deforestation. Ocean acidification. Soil depletion. Aquifer exhaustion. Pollinator decline. Each one weakens the foundation. Each one moves us closer to structural failure. We're not facing one problem. We're facing interconnected system failures happening simultaneously. Climate change amplifies species extinction. Species extinction reduces ecosystem resilience. Reduced resilience accelerates climate impacts. Feedback loops compound. Tipping points cascade. And suddenly, gradual change becomes rapid collapse. That's how complex systems fail. Slowly, then all at once. The challenge is acting during the slow phase. When problems seem manageable. When denial remains plausible. When short-term costs feel more real than long-term consequences. Acting requires overriding our evolutionary programming. We evolved for immediate threats. Predators. Starvation. Injury. Our brains are wired for rapid response to visible danger. But climate change isn't a predator. It's a statistical shift. A gradual accumulation. A probability distribution changing shape. Our brains aren't built to process that. We don't feel threatened by parts per million. By decimal point temperature increases. By percentage changes in biodiversity. We feel threatened by things with teeth. By things that can hurt us now. Which is why this is hard. Why knowing isn't enough. Why information doesn't automatically translate to action. We're asking people to respond emotionally to spreadsheets. To feel urgency about graphs. To sacrifice immediate comfort for abstract future benefits. That's not how humans work. But it's what the situation requires. Which means we need to change how we work. Not our biology, that's too slow. But our systems. Our institutions. Our decision-making frameworks. We need structures that account for long-term consequences. That price in externalities. That prioritize sustainability over quarterly profits. That value ecosystem health over GDP growth. Those structures don't exist by default. They require intentional design. Political will. Social pressure. Cultural shifts. They require enough people demanding something different. Enough people recognizing that current systems are driving us toward collapse. Enough people willing to support changes that might be personally inconvenient but collectively necessary. And that's the real test. Not whether we can develop solutions, We have solutions. Renewable energy, regenerative agriculture, circular economies, ecosystem restoration. The technology exists. The knowledge exists. What's missing is implementation at scale. What's missing is political and economic systems that prioritize long-term survival over short-term extraction. What's missing is collective willingness to accept that infinite growth on a finite planet is mathematically impossible. That's the hard part. Not the science. Not the engineering. The acceptance that our current trajectory is unsustainable. That something has to change. That we can't keep doing what we're doing and expect different results. And we're running out of time to figure this out. Not in a dramatic, countdown clock way, but in the sense that every year of delay makes the problem harder. Every ton of carbon emitted stays in the atmosphere for decades. Every species lost is gone permanently. Every ecosystem threshold crossed is effectively irreversible on human timescales. We're making decisions now that will echo for thousands of years. Decisions about what kind of planet we leave behind. What kind of future we're creating. Whether intelligence, our defining characteristic, proves adaptive or catastrophic. The answer isn't written yet. We're writing it. Right now. Through choices large and small. Through policies and purchases. Through what we demand and what we accept. Through whether we treat this as an emergency or a distant concern. Evolution doesn't care which we choose. It'll respond to whatever conditions we create. It'll favor whatever traits prove adaptive in the world we're building. Maybe that world includes humans. Maybe it doesn't. Maybe intelligence remains advantageous. Maybe it becomes a liability. Maybe we're the ancestors of something extraordinary. Maybe we're just another failed experiment. Another branch that couldn't adapt fast enough. Another fossil layer marking a brief moment when one species thought it could outsmart 4 billion years of planetary processes. We won't know the answer for millions of years. But we're determining it now. Part 6 of 6. The story we've been told about comets, the one where they're just icy debris wandering through space, needs updating. Because the data keeps pointing in one direction. These objects aren't random. They're delivery systems. Packages sent from the outer solar system carrying exactly what early Earth needed but couldn't make efficiently on its own. Think about the timing. Earth forms hot. Too hot for organic chemistry. It needs to cool, needs water, needs the building blocks for biochemistry. And right on schedule, here come the comets. Billions of them during the late heavy bombardment. Each one carrying ice, organics, amino acids, phosphorus. Each impact a delivery. Not violent destruction, but violent creation. We've spent decades assuming life started here through local chemistry. Lightning strikes. Volcanic vents. Tidal pools. And maybe some of it did. But the numbers don't work. Early Earth's atmosphere wasn't right for making complex organics in quantity. The Miller-Urey experiment, elegant as it was, used the wrong gases. When you correct for that, the yield drops dramatically. Local production wasn't enough. But comets? They're making organics constantly. In the cold dark between stars. No atmosphere to worry about. No oxygen to interfere. Just carbon, hydrogen, nitrogen, oxygen, and time. Lots of time. These molecules form, accumulate, get locked into ice matrices that protect them. Then they wait. Sometimes for billions of years. Until gravity pulls them inward. Until they slam into young planets. Until they deliver their cargo. What we found on Wild 2 and 67P isn't an accident. It's a pattern. Comets everywhere are doing this. Making the same molecules. Carrying the same building blocks. It's chemistry operating at solar system scale. And Earth was in the path of that delivery route for hundreds of millions of years. The ice itself mattered just as much as what was in it. Early Earth needed water. Vast amounts. Our oceans didn't condense from the primordial atmosphere. They came from outside. From objects that formed in colder regions and then migrated inward. Some from asteroids, yes. But comets contributed. Isotope ratios prove it. Not all our water is cometary, but some is. Enough to matter. So now picture the actual scenario. 4 billion years ago. Earth with thin oceans, no continents yet, active volcanism everywhere. And it's being bombarded constantly. Not planet-killing impacts, but steady, relentless delivery. Each comet brings water, organics, minerals. The ice cushions the impact, protects the cargo. The organics survive, dissolve into the ocean, accumulate. The minerals settle. The carbon compounds concentrate in tidal zones, around vents, in shallow pools. You're not starting with nothing. You're starting with a planet being continuously restocked with prebiotic chemistry. With molecules that already have some complexity. With building blocks ready to assemble into something more sophisticated. The local chemistry, the lightning and vents and UV radiation, they're still happening. But now they're working with better raw materials. They're not creating from scratch. They're refining what's already there. And here's what makes this profound. It means life's origin isn't purely local. It's not Earth solving the problem alone. It's Earth benefiting from processes that happened across the entire solar system. From chemistry in molecular clouds. From reactions in the outer solar system. From delivery mechanisms that operated for hundreds of millions of years. Life's origin is a collaboration between a planet and its cosmic environment. Which raises an obvious question. If this happened here, did it happen elsewhere? Are there other planets, right now, being seeded by comets? Other oceans accumulating organic molecules? Other worlds where chemistry is transitioning toward biology? We don't know. But the statistics suggest yes. Our solar system isn't unique. Comets form around other stars. Planets get bombarded everywhere. The same physical processes, the same chemistry, the same delivery mechanisms. If it worked once, it can work again. Maybe it's working right now on planets we haven't discovered yet. Maybe it's worked countless times on worlds that existed billions of years ago. Maybe we're not the first. Maybe we won't be the last. That doesn't make life inevitable. Chemistry alone doesn't guarantee biology. You need the right conditions, the right timing, the right sequence of events. But it makes life less miraculous, less impossibly unlikely. It shifts the question from, how did life beat astronomical odds? To, given favorable conditions and enough time, how often does chemistry become biology? And that's a different question. One with potentially different answers. Because chemistry is patient. It doesn't care about timescales. It just keeps trying combinations. Keep mixing molecules for 1 billion years and eventually something interesting happens. Not guaranteed, but probable. And probability, given enough opportunities, approaches certainty. We're still missing pieces. We don't know exactly how RNA formed, how membranes and replication combined, how the genetic code got established. Those are hard problems, but they're becoming tractable. Every year, lab experiments get closer to recreating conditions that produce self-replicating systems. Every year, we understand more about what early Earth was actually like. Every year, the gap between non-life and life gets smaller. Comets didn't solve the origin of life. They solved the supply problem. They provided raw materials that early Earth couldn't efficiently produce. They turned an improbable event into something merely unlikely. And on planetary timescales, unlikely events happen constantly. You just need to wait. The evidence keeps accumulating. Not just from comets we visited, but from meteorites, from interstellar chemistry, from laboratory experiments. All of it pointing toward the same conclusion. The building blocks of life aren't rare. They're common. They form naturally in cold environments. They survive delivery to planetary surfaces. They accumulate. They react. They experiment. What's rare isn't the chemistry. It's the conditions that let chemistry become biology. Stable liquid water. Energy sources. Mineral surfaces for catalysis. Protection from radiation. Enough time without catastrophic disruption. Those conditions are specific. Not impossible, but requiring a particular combination of planetary characteristics. Earth had those characteristics. Maybe other planets do too. Maybe they're common. Maybe they're rare. We're only beginning to survey the possibilities. Only beginning to understand how many Earth-like worlds exist. Only beginning to look for biosignatures, chemical signs that biology is happening somewhere else. The search is underway. Telescopes scanning exoplanet atmospheres for oxygen, methane, combinations that suggest life. Probes examining Europa and Enceladus, moons with subsurface oceans that might harbor biology. Future missions to Venus, Mars, Titan, looking for current life or fossil evidence of past life. Each mission refines our understanding. Each discovery adjusts the probabilities. And those probabilities matter, because they determine whether we're alone or part of something larger. Whether life's origin was a singular miracle or a common occurrence. Whether intelligence is unique to Earth or inevitable given enough time. Whether we're the only species capable of asking these questions or one of many scattered across the galaxy. We don't have those answers yet. But we're closer than we've ever been. Closer to understanding our origins. Closer to knowing whether life elsewhere exists. Closer to determining our place in the universe. Not through philosophy or speculation, but through data. Through chemistry. Through missions that touch these ancient objects and read their composition. Comets are messengers. Not from gods or fate, but from the early solar system. They carry information about conditions 4.5 billion years ago. About chemistry in the outer regions. About what was available when Earth formed. That information is still readable, still preserved in ice that hasn't melted since the solar system's beginning. Every comet we study refines the picture. Every measurement adds detail. Every discovery adjusts our understanding. We're not guessing anymore about what these objects contain. We're measuring it directly. And what we're measuring consistently shows organic complexity. Amino acids. Nucleobases. Phosphorus. Water. Everything life needs. That doesn't prove comets created life. Proof would require showing the mechanism, demonstrating that delivered organics actually assembled into something living. We can't do that. We weren't there. We don't have recordings. We have only evidence, circumstantial but compelling, pointing toward cometary delivery as a crucial part of life's origin. The alternative is that all of this is coincidence. That comets happen to carry exactly the molecules life uses, but contributed nothing. That early Earth independently synthesized everything despite having the wrong atmospheric chemistry. That the timing of heavy bombardment and life's appearance is unrelated. Possible, but increasingly implausible. Science works by eliminating alternatives until only one explanation fits the data. We're not there yet. But we're moving in that direction. Each study, each mission, each experiment narrows the possibilities. And what's emerging is a story where life's origin wasn't purely terrestrial. Where Earth's chemistry was supplemented by cosmic delivery. where our existence depended on objects formed in a cold dark carrying cargo across billions of kilometers. It's a strange story, not the one we expected. But data doesn't care about expectations. It cares about what actually happened. And what appears to have happened is that life's ingredients came from space. Not all of them, but enough to matter. Enough to tip the balance from impossible to merely improbable. Enough to give chemistry the raw materials it needed to begin experimenting with biology. We're the result of that experiment. Not the only possible result, but one that worked. One that survived. One that eventually became capable of looking back and asking where it came from. And now we have an answer, partial but growing more complete. We came from Earth, yes. But Earth came from the solar system. And the solar system provided, through comets, the chemical foundation that made us possible. That foundation is still out there. Still orbiting. Still carrying the same molecules. Still waiting to be studied. Each comet is a time capsule from the solar system's formation. Each one holds clues about conditions back then. About chemistry. About the processes that led to us. We've only examined a handful. Thousands more are accessible. Each one potentially holding new information, new insights, new pieces of the puzzle. The work continues. Missions are being planned to return samples from comets. To land on their surfaces. To drill into their cores and retrieve pristine material that's been frozen since the beginning. Each sample will be analyzed in laboratories with instruments far more sensitive than what existed even a decade ago. Each analysis will reveal more about what comets contain and how those contents relate to biology. We're at the beginning of understanding this connection, not the end. The evidence so far is suggestive, compelling, but incomplete. We need more data, more samples, more experiments, more missions. The story of how comets contributed to life's origin is still being written. But the outline is visible. The general shape is clear. And it's changing how we think about our place in the universe. Because if comets seeded Earth, they're seeding other planets too. If organic delivery is common, then the building blocks of life are widespread. If chemistry can transition to biology given the right conditions, then those conditions might exist elsewhere. The universe might be full of experiments similar to the one that produced us. Some failing, some succeeding, some just beginning, some already billions of years old. We don't know yet, but we're looking. And every year, our tools get better. Our understanding deepens. Our search expands. The question of whether we're alone is becoming answerable. Not through speculation, but through observation. Through detecting biosignatures on distant worlds. Through finding life in our own solar system beyond Earth. Through understanding how common the processes that created us actually are. That understanding starts with comets. With these frozen archives carrying evidence from our beginning. With missions that brave enough to catch up to them, touch them, taste their chemistry. With scientists patient enough to analyze the data, piece together the story, follow wherever the evidence leads. And where it's leading is toward a cosmos more interconnected than we imagined. Where life's origin isn't isolated to individual planets, but involves solar system-wide processes. Where chemistry in one region affects biology in another. Where what happens in the cold outer darkness eventually influences what emerges in the warm inner light. We're part of that story. Products of processes that began before Earth existed. Beneficiaries of delivery systems that operated for hundreds of millions of years. Descendants of chemistry that started in molecular clouds and ended in living cells. The path from there to here was long, improbable, contingent on countless specific events. But it happened. And understanding how it happened changes everything about how we see ourselves and our origins.
A world exists that orbits no star. Its surface, colder than anything you can imagine, receives no warmth from a distant sun. Deep beneath frozen layers measured in kilometers, residual heat from a violent birth billions of years ago might sustain something we'd hesitate to call life. This planet has been traveling through the void for longer than Earth has supported complex organisms, and recently, astronomers detected one of these wanderers closer than we've ever confirmed before. Rogue planets represent one of astronomy's most unsettling discoveries. These aren't comets or asteroids. They're full-scale worlds, some larger than Jupiter, drifting through interstellar darkness without gravitational allegiance to any star. Until recently, we assumed planets required stars. The very definition seemed to demand it. Then observations started revealing something unexpected. The galaxy is absolutely crowded with these orphaned worlds. The numbers are staggering in ways that fundamentally challenge our understanding of planetary demographics. The Milky Way might contain 8 trillion rogue planets. Let that settle for a moment. 8 trillion. Our galaxy has roughly 400 billion stars, which already seems incomprehensible. But for every star burning in the night sky, there could be 20 planets wandering alone through the darkness between stellar systems. On January 2nd, 2026, an international team announced they discovered a new rogue planet about 9,800 light-years from Earth. What makes this detection revolutionary isn't just that they found another wanderer. For the first time, astronomers obtained a direct mass measurement, not a statistical estimate based on probability distributions. They know for certain this object has a mass similar to Saturn. The discovery came through a microlensing event with the rather unglamorous designation KMT 2024 BLG 0792/OGLE 2024 BLG 0516. Multiple telescopes on the ground and in space watched simultaneously as this dark world passed between Earth and a distant background star, its gravity bending light in ways that revealed its presence and properties with unprecedented precision. But here's what keeps astronomers awake. We're only detecting the largest ones, and even then, only when they happen to pass directly between us and another light source. The smaller rocky worlds, the planets that might resemble Earth or Mars, remain completely invisible with current technology. They emit almost no infrared radiation, reflect no starlight, and only reveal themselves through gravitational effects that require extraordinary luck to observe. Think about the implications. Every planetary system that forms goes through a violent settling period. Computer simulations tracking the formation of 100 different solar systems, each containing 3 to 10 planets orbiting a sun-like star, revealed something disturbing when researchers let the equations run for 1 billion simulated years. For every stable system like ours that emerges, at least one gas giant gets ejected into interstellar space. And 5 to 10 smaller, rocky worlds get flung out alongside it. Our own solar system might have participated in this cosmic exile program. Some researchers theorize we once had a fifth giant planet alongside Jupiter, Saturn, Uranus, and Neptune. During a chaotic period called late instability, gravitational interactions between these massive worlds became unstable. The unfortunate fifth planet got accelerated to escape velocity and vanished into the dark. With the survivors, the planets that happened to find stable orbits after the violence settled. The ejection mechanisms are diverse and brutal. In systems with two stars orbiting each other, planets can get caught in gravitational tug-of-war scenarios that eventually tear them free. When stars pass unusually close to each other in wide binary systems, the orbital dynamics can shift so dramatically that planets get stolen or ejected entirely. But the most common mechanism is simpler and more direct: planet-planet scattering. Picture two planets whose orbits bring them too close. The gravitational interaction becomes chaotic. One planet, usually the less massive of the two, gains enormous orbital energy in a single encounter. Within a few orbits, it's moving fast enough to break free from the star's gravity entirely. The other planet settles into a more stable orbit, its trajectory only slightly altered. One world continues its predictable life. The other begins a journey through darkness that might last billions of years. These ejected planets retain velocity signatures that tell their origin stories. Astronomers measure something called velocity dispersion, essentially how fast these objects move relative to surrounding stars. Rogue planets kicked out through planet-planet scattering travel at 8 to 16 kilometers per second. Those ejected through binary star interactions move 3 times faster. These velocities serve as fingerprints, revealing the violence of their exile long after the event occurred. Not all rogue planets started their existence orbiting stars. Some formed alone in interstellar space, condensing from clouds of gas and dust through processes more similar to star formation than traditional planet formation. These objects collapse under their own gravity, accreting material directly from surrounding nebulae, but never gathering enough mass to ignite fusion and become true stars. The International Astronomical Union proposed calling them sub-brown dwarfs to distinguish them from planets that formed in protoplanetary disks and were subsequently ejected. The distinction matters because it reveals fundamentally different formation pathways. An ejected planet carries with it the chemical signature of the stellar system where it formed. Its composition reflects the specific mix of elements present in that particular protoplanetary disk. A planet that formed alone in interstellar space would have a completely different chemical profile, one that reflects the average composition of the interstellar medium rather than a specific stellar environment. In 2025, astronomers observed something that challenged previous assumptions about these isolated worlds. They found a rogue planet designated CHA 1107 to 7626, located about 620 light-years away in the constellation Chameleon. This object has a mass 5 to 10 times that of Jupiter. But what stunned researchers was watching it actively accrete material from a surrounding disk of gas and dust at a rate of 6 billion tons per second. This represents the strongest accretion episode ever recorded for a planetary mass object. The discovery revealed that some rogue planets can exhibit star-like behavior during formation, with magnetic activity driving material infall in ways previously thought exclusive to stellar evolution. The planet is still forming, still growing, still pulling in matter from its environment despite having no parent star to organize that material into a traditional protoplanetary disk. Detecting these objects requires monitoring millions of stars continuously, watching for the brief moments when a rogue planet's gravity bends light from a background source. The technique, called gravitational microlensing, works because even planets have enough mass to warp spacetime according to Einstein's general relativity. When alignment occurs, the background star brightens temporarily, sometimes for just a few hours. The challenge is enormous. Only one star in 100,000 gets microlensed at any given time. To find planets, you need to observe millions of stars on timescales of several minutes, checking at least several times per hour. Survey teams aim their telescopes towards Sagittarius, which lies in the direction of our galaxy's dense center where about 400 million detectable stars cluster together. Even then, hundreds of events per year toward the galactic bulge still represent a tiny fraction of the actual population. The upcoming Nancy Grace Roman Space Telescope, scheduled to launch by May 2027, should revolutionize this field entirely. Scientists expect it will discover at least 400 more rogue planets, potentially several thousand. The telescope can image the entire sky 1,000 times faster than Hubble, using both microlensing and direct infrared imaging to find worlds potentially as small as Mars. But here's the question that shifts this from purely academic interest to something more immediate. What happens if one of these wanderers enters our solar system? The odds are extraordinarily low. Astronomers calculate roughly 1 in 1 trillion chance within the next 1,000 years. Space is vast enough that even with trillions of rogue planets wandering the Milky Way, the distances between objects make close encounters vanishingly rare. Yet the theoretical consequences are worth understanding because they reveal just how delicately balanced our solar system actually is. If a rogue planet approaching 13 times Jupiter's mass crossed into our solar system, the gravitational disturbance would begin long before any visible approach. Our planets orbit the Sun in relatively narrow bands we call the habitable zone, regions where conditions remain stable enough for liquid water and predictable seasons. Earth sits comfortably within this zone, neither too close to freeze nor too near to burn. But that comfortable position depends entirely on gravitational stability maintained over billions of years. The intruder would warp that stability immediately. Its gravitational field would reach outward like invisible fingers, tugging on objects billions of kilometers away. Neptune would feel the pull first, its orbit shifting microscopically at first, then more dramatically as the rogue planet advanced inward. Uranus would follow, then Saturn, then Jupiter. Each giant planet's orbit would stretch, becoming more elliptical, less circular. The perfectly synchronized dance that has kept our solar system stable since its formation would begin breaking down. Earth's orbit would change too. Not necessarily in ways that eject us from the habitable zone entirely, but in ways that fundamentally alter how we experience seasons and climate. Picture our planet's orbit stretching into an elongated ellipse. For part of the year, we'd swing closer to the Sun than we've been in 4 billion years. Temperatures would spike dramatically during these brief summers, potentially reaching levels that make current heat waves seem mild. Ocean surfaces would heat rapidly. Ice caps would melt at accelerated rates. Weather patterns would destabilize completely. Then the orbit would carry us outward again. These winters would last far longer than the summers, plunging Earth into extended cold periods that could span years or decades depending on the severity of orbital disruption. Agriculture would collapse almost immediately. Crops require specific growing seasons, predictable temperatures, stable water cycles. When summer lasts only a fraction of its normal duration, and winter extends indefinitely, food production becomes impossible at scale. Within a single orbital period, humanity would face resource shortages unlike anything in recorded history. But orbital disruption represents just one category of threat. The rogue planet's passage would also destabilize the asteroid belt between Mars and Jupiter. This region contains millions of rocky bodies, most following stable orbits that keep them safely distant from inner planets. Gravitational perturbation from a massive intruder would send countless asteroids careening in unpredictable directions. Some would fall toward the Sun. Others would be flung outward into deep space. And some would find trajectories that intersect with Earth's orbit. The bombardment wouldn't happen all at once. Instead, we'd face an extended period of elevated impact risk lasting potentially thousands of years as the disturbed asteroid population gradually restabilized. Each impact would carry consequences proportional to the impactor's size. Small asteroids would burn up in the atmosphere or create localized damage. Larger ones would generate regional devastation. And the largest could trigger mass extinction events comparable to the impact that ended the dinosaurs 66 million years ago. The outer solar system would experience even more dramatic effects. The Kuiper Belt and Oort cloud contain trillions of icy bodies, most in stable distant orbits that keep them far from the inner planets. These regions function as cosmic deep freezers, preserving pristine samples of material from the solar system's formation. When a rogue planet passes through, it doesn't just disturb a few objects. It reorganizes entire populations, sending waves of comets toward the inner solar system. Historical records show that even minor perturbations in the Oort cloud can increase comet activity measurably. A full-scale disruption would unleash a comet storm lasting millions of years. The night sky would fill with visible comets, dozens at a time, their tails stretching across the heavens. Beautiful from a distance. Terrifying when you realize each represents a potential impact threat moving at tens of kilometers per second. Yet direct collision with the rogue planet itself would dwarf all these other scenarios. The kinetic energy involved defies comprehension. A Jupiter-mass object moving at typical interstellar velocities, somewhere between 50 and 200 kilometers per second, carries momentum that makes terrestrial disasters seem insignificant by comparison. If Earth happened to lie directly in its path, the collision wouldn't just destroy our planet. It would vaporize it completely. The impact would release more energy than all nuclear weapons ever built, multiplied by billions. Both worlds would transform instantly into expanding clouds of superheated plasma, temperatures reaching millions of degrees, hot enough to strip electrons from atoms. The debris cloud would glow brighter than the sun briefly before beginning its long cooling process. Over millions of years, this material might eventually coalesce into new bodies. But nothing resembling Earth would remain. No fossils. No geological record. No evidence that our planet ever existed beyond chemical signatures in the debris field. The probability of such a direct collision remains vanishingly small. Space is vast. Even within our solar system, the distances between objects dwarf the sizes of the objects themselves. A rogue planet could pass through the solar system and miss every planet purely by chance, simply because empty space dominates the architecture. The odds of threading the needle to hit Earth specifically are so low that astronomers calculate perhaps one such event per trillion years. But near misses carry their own dangers. The rogue planet doesn't need to hit Earth to devastate it. A close approach, even one that misses by millions of kilometers, would generate tidal forces powerful enough to stress our planet's interior. Imagine Earth's entire mass being pulled unevenly, the near side attracted more strongly than the far side. This differential force, called a tidal gradient, can trigger geological activity on unprecedented scales. Volcanic eruptions would increase in frequency and intensity as the planet's mantle convects more vigorously under tidal heating. Earthquake activity would spike globally. Tectonic plates would shift more rapidly than normal, potentially triggering mega-earthquakes that make the most powerful historical quakes seem minor. Coastlines would experience massive tsunamis as ocean water responds to the changing gravitational field. The planet wouldn't tear apart, but it would experience geological violence concentrated into a brief period rather than spread across millions of years. Then there's the Moon. Our natural satellite orbits at a distance where Earth's gravity dominates, but that dominance isn't absolute. A massive object passing nearby could perturb the Moon's orbit significantly, potentially even capturing it entirely if the geometry aligned precisely. Losing the moon would alter Earth's axial tilt over time, destabilizing climate patterns that depend on that tilt remaining relatively constant. Tides would weaken dramatically, affecting ocean circulation patterns that regulate global temperature distribution. Research into these scenarios isn't purely academic. Scientists study them to understand the limits of planetary system stability, the frequency of catastrophic events over cosmic timescales, and the resilience of life under extreme conditions. The models they build require simulating gravitational interactions between multiple massive bodies over millions of years, tracking how small perturbations compound into large-scale reorganization. These simulations reveal something crucial about our solar system's architecture. The current configuration isn't random. It represents the stable endpoint of billions of years of gravitational evolution. Early in the solar system's history, planetary orbits were far less regular. Planets migrated inward and outward. Some collided. Others were ejected entirely. The NICE model, named after the French city where researchers developed it, suggests that our gas giants once occupied completely different orbital positions before gravitational interactions shuffled them into their current locations. That early chaos may have ejected a fifth giant planet entirely. Computer models show that systems starting with 5 gas giants tend to eject one, leaving 4 in stable orbits that match what we observe today. If true, our solar system has already experienced the kind of gravitational violence that creates rogue planets. We may have contributed to the population of wanderers drifting through the galaxy. Somewhere out there, a Jupiter-sized world might be traveling alone through the darkness, carrying with it chemical signatures that would identify it as having formed in our solar system billions of years ago. Understanding rogue planet encounters also helps astronomers estimate how common such events are throughout the galaxy. If systems typically eject multiple planets during formation, and if billions of systems have formed over the galaxy's lifetime, then trillions of rogue planets represent a natural consequence of planet formation itself. The process is violent and chaotic, with only a fraction of formed planets remaining gravitationally bound to their parent stars. That chaos produced something else beyond ejected planets. It created a testing ground for understanding how gravitational interactions scale across different masses and distances. When astronomers model rogue planet encounters, they're not just calculating abstract trajectories. They're mapping the boundaries between stability and catastrophe, between systems that endure and systems that disintegrate. The math reveals patterns that hold true regardless of scale, from moons orbiting planets to planets orbiting stars to stars orbiting galactic centers. These patterns explain why certain orbital configurations persist while others collapse rapidly. Resonances matter enormously. When 2 orbits have periods that form simple ratios, 3:2 or 2:1, the bodies repeatedly align at the same points in their orbits. These alignments accumulate gravitational nudges that either stabilize the system or destabilize it, depending on the specific geometry. Jupiter and Saturn demonstrate this principle. Their orbital periods nearly form a 5:2 ratio, close enough to create significant long-term effects but far enough from exact resonance to avoid catastrophic instability. A rogue planet passing through disrupts these delicate balances. Even a relatively distant encounter can perturb orbital periods enough to shift a stable resonance into an unstable one. The effect cascades. Jupiter's orbit changes slightly. That alters its gravitational influence on Saturn. Saturn's shifted orbit affects Uranus. The inner planets feel the effects later but inevitably. Over thousands or millions of years, orbits that appeared stable reveal themselves to be metastable, waiting for a trigger to push them into new configurations. This vulnerability explains why finding rogue planets matters beyond academic curiosity. Each discovery provides data points for estimating encounter frequencies. Astronomers can calculate how many rogue planets exist within certain distance ranges, how fast they typically travel, and therefore how often close approaches occur statistically. The numbers suggest that over the lifetime of a typical planetary system, at least one moderately close encounter with a rogue planet is more likely than not. But what constitutes close in cosmic terms? A rogue planet passing within 1 light-year would have minimal effect. At that distance, even a massive interloper exerts negligible gravitational influence over the brief time it takes to pass. Move that encounter to within 100 astronomical units, roughly 3 times Neptune's orbital distance, and the story changes. Now the rogue planet spends years or decades within the outer solar system's gravitational sphere of influence, plenty of time for orbital perturbations to accumulate. The difference between a harmless distant passage and a system-disrupting encounter comes down to velocity and trajectory. A fast-moving rogue planet on a perpendicular trajectory might slice through the outer solar system in just a few years, limiting the time available for gravitational interactions. A slower object on an aligned trajectory could remain gravitationally engaged for decades or centuries, maximizing the cumulative perturbations. The geometry matters as much as the mass. Detecting such an encounter in advance presents technological challenges that current capabilities barely address. Rogue planets emit almost no light. They don't reflect starlight because they're not near stars. Their thermal emission peaks in infrared wavelengths, but even then, a Jupiter-mass object several hundred astronomical units away would appear incredibly faint. Current surveys might detect it a few years before closest approach, not the decades or centuries that would allow meaningful preparation. That detection gap creates an uncomfortable situation. We know rogue planets exist in large numbers. We understand the potential consequences of close encounters. Yet we have no systematic early warning system capable of identifying incoming objects with sufficient lead time. The upcoming Nancy Grace Roman Space Telescope will improve this situation dramatically. Scheduled to launch by May 2027, it will conduct a galactic bulge time domain survey, potentially discovering hundreds to thousands of rogue planets and refining mass estimates across the population. Roman's capabilities extend beyond simple detection. By observing microlensing events where rogue planets pass in front of background stars, astronomers can measure not just presence but mass with unprecedented accuracy. The January 2026 discovery of a Saturn-mass rogue planet roughly 9,800 light-years away demonstrated this technique's power. For the first time, researchers obtained a direct mass measurement rather than statistical estimates. That single data point improved population models significantly. More discoveries like that one will paint a clearer picture of the rogue planet population structure. Are most ejected worlds small rocky planets, or do gas giants dominate? Do formation mechanisms leave distinctive signatures in the mass distribution? How does ejection velocity correlate with planet type? These questions matter for risk assessment. A galaxy filled with Earth mass rogues presents different statistical risks than one dominated by Jupiter mass wanderers. The former are more numerous but less gravitationally disruptive. The latter are rarer but capable of greater orbital perturbations. Understanding the population structure also illuminates planet formation physics. Computer simulations suggest that for every solar system like ours, at least one gas giant and 5 to 10 smaller rocky worlds get ejected during formation. Those numbers come from modeling hundreds of systems with varying initial conditions, then running the physics forward for billions of simulated years. The simulations consistently show that ejection isn't occasional. It's standard. Most planetary systems shed members during their chaotic early evolution. This raises a peculiar question about our own history. If ejection is standard, did our solar system expel worlds during its formation? The nice model suggests yes. Named after the French city where researchers developed it, this model proposes that our gas giants once occupied different orbital positions. Gravitational interactions shuffled them into their current locations, potentially ejecting a fifth giant planet entirely in the process. Evidence supporting this scenario includes the orbital distributions of asteroids and comets, the late heavy bombardment that scarred the Moon roughly 4 billion years ago, and the unusual orbits of certain Kuiper belt objects. If we did eject a planet, it's out there somewhere. It would carry chemical signatures linking it to our solar system, isotopic ratios that formed in our protoplanetary disk. Finding such an object would confirm the nice model directly and demonstrate that even stable-appearing systems like ours experienced violent gravitational upheavals. The search for our hypothetical lost sibling represents one of astronomy's more speculative pursuits, but improving detection methods make it incrementally less impossible. Meanwhile, the search for Planet 9 continues. This hypothetical massive planet in the outer solar system could explain the peculiar clustering of certain trans-Neptunian object orbits. These extreme objects, orbiting beyond Neptune at distances averaging more than 250 astronomical units, tend to make their closest solar approaches in one sector. Their orbits tilt similarly. Random distribution would spread them more evenly. Something massive appears to be shepherding them gravitationally. Planet 9, if it exists, would have a mass roughly 10 times Earth's and orbit about 20 times farther from the Sun than Neptune. Several formation scenarios could produce such an object. It might be the core of a gas giant that Jupiter ejected during the solar system's youth. It could be a rogue planet that our Sun captured from interstellar space. Or it might have formed on a distant orbit and been pulled into its current eccentric path by a passing star. The capture hypothesis particularly intrigues researchers. Studies show that the Sun's gravitational field, combined with galactic tidal forces, creates regions where passing objects can become permanently trapped through a mechanism called permanent weak capture. An interstellar object entering these zones might remain on a chaotic orbit for millions of years before settling into a stable configuration. The Sun could potentially collect passing rogue planets and hold them at the solar system's outskirts, occasionally trading them with neighboring star systems like Alpha Centauri. This capture mechanism depends on precise conditions. The incoming object needs the right velocity and trajectory to dissipate enough orbital energy without escaping entirely. Galactic tides help by subtly altering the object's motion as it travels through regions where the Milky Way's gravitational field varies. These variations are tiny, but over millions of years, they can shift an initially unbound orbit into a bound one. The process works in reverse too, potentially releasing captured objects after long periods of gravitational confinement. Whether Planet 9 is a captured rogue or a native object, finding it would answer fundamental questions about solar system architecture. The Vera C. Rubin Observatory, currently under construction in Chile, should either detect it or rule out its existence within a few years through comprehensive sky surveys. The observatory's wide field of view and sensitive detectors will image the entire visible sky repeatedly, tracking anything that moves against the background stars. If Planet 9 lurks out there, Rubin will spot it. That pending discovery or non-discovery represents a broader shift in astronomy. We're moving from an era where we knew our immediate neighborhood reasonably well but understood little about the wider galaxy to one where we're cataloging thousands of exoplanets, detecting rogue planets light years away, and mapping the galaxy's structure in unprecedented detail. Each discovery refines our understanding of how planetary systems form, devolve, and sometimes disintegrate. The technology driving these discoveries has evolved dramatically. Finding rogue planets requires detecting objects that emit virtually no light and reflect nothing from nearby stars. They're invisible in traditional surveys. Microlensing changed everything. The technique exploits Einstein's general relativity. Any massive object warps spacetime around it, bending light that passes nearby. When a rogue planet drifts between Earth and a distant star, it acts as a gravitational lens, briefly magnifying the background star's light. The effect lasts hours to days depending on the planet's mass and velocity. Astronomers monitoring millions of stars can catch these fleeting signals. The practical challenges are staggering. Only 1 in 100,000 stars undergoes microlensing at any moment. Survey teams point their telescopes toward the galactic bulge in Sagittarius, where 400 million detectable stars cluster densely. Even there, finding planets requires observing millions of targets multiple times per hour. The OGLE and MOA projects identified hundreds of candidate events by 2007. Most were stellar lenses, but occasionally a light curve showed the characteristic spike indicating a planetary companion. Coordinated telescope networks spanning the globe enable continuous coverage, preventing observers from missing brief events that occurred during daylight hours at any single site. January 2026 brought a breakthrough that refined these techniques dramatically. An international team announced detecting a Saturn-mass rogue planet 9,800 light-years away through microlensing event KMT 2024 BLG 0792. What made this discovery extraordinary was simultaneous observation from ground-based telescopes in space. The parallax measurements from different viewing angles allowed direct mass calculation rather than statistical estimates. For the first time, astronomers knew a rogue planet's mass with certainty. The object weighed approximately 95 Earth masses, placing it firmly in the gas giant category. Previous detections relied on models comparing light curve characteristics to simulations. This measurement established a new standard for confirmation. But mass alone doesn't reveal origin stories. Velocity provides crucial forensic evidence about how these worlds became orphans. Ejected planets retain significant excess velocities, typically 8 to 16 kilometers per second above the background motion of stars in their birth regions. This speed difference acts as a fingerprint. Planets ejected through interactions with binary companions move even faster, roughly 3 times the velocity of worlds expelled through planet-planet scattering. These measurements let astronomers reconstruct the gravitational violence that launched specific objects into interstellar space. Each rogue planet carries evidence of its past encoded in its trajectory. Some rogue planets might never have had parent stars. Isolated formation theory proposes these objects form like stars do, from collapsing gas clouds, but halt at planetary scales. Insufficient initial cloud mass or external disruptions truncate accretion before the object can grow large enough for fusion. Turbulence within molecular clouds generates density perturbations that fragment into multiple low-mass clumps. These cores collect gas directly from surrounding nebulae, building planetary mass bodies without protoplanetary disks. The International Astronomical Union proposed calling such objects sub-brown dwarfs to distinguish them from ejected planets. Whether this distinction matters philosophically or just taxonomically remains debated. Another 2025 discovery challenged assumptions about rogue planet behavior. Astronomers identified CHA 1107 to 7626, located 620 light-years away in Chameleon constellation. This free-floating planet exhibits a mass 5 to 10 times Jupiter's and displays something unprecedented, an enormous growth spurt. The object accretes gas and dust from surrounding material at 6 billion tons per second, the strongest episode ever recorded for planetary mass bodies. This rogue planet possesses its own accretion disk, behaving more like a young star than a typical planet. Magnetic activity appears to drive material infall in ways previously thought exclusive to stellar evolution. The discovery suggests rogue planets can remain dynamically active long after ejection or formation. These findings reveal how violent planetary system formation actually is. Computer simulations model this chaos systematically. Researchers created 100 virtual planetary systems, each containing 3 to 10 planets on near-circular coplanar orbits around Sun-like stars. Running the equations forward for 1 billion simulated years produced consistent results. For every stable system like ours that forms, at least one gas giant and approximately 5 to 10 smaller rocky worlds get ejected into interstellar space. The process is standard, not exceptional. Ejection represents a normal byproduct of gravitational interactions during the first several million years when systems haven't settled into stable configurations yet. Binary star systems amplify these dynamics. Planets in binary systems experience more complex gravitational perturbations than those orbiting single stars. Even wide binaries contribute. Passing stars can perturb the eccentricity of stellar orbits, occasionally sending binary companions close together. These encounters trigger planetary ejections or even steal planets from one star's gravitational sphere. Research focusing on circumbinary systems, planets orbiting both stars in a binary, shows that misaligned orbital planes create instabilities ejecting planets with much greater frequency. Given that binary stars comprise more than half of all stellar systems, they likely contribute disproportionately to the rogue planet population. Planet-planet scattering in single-star systems follows predictable patterns. When two planets approach too closely, gravitational interactions transfer orbital energy between them. Usually the less massive planet gains energy, accelerating to escape velocity while its massive companion's orbit shrinks slightly. The exchange conserves momentum but redistributes it dramatically. Close encounters during migration through gas-rich protoplanetary disks can trigger these events. Planets forming in compact configurations face higher collision and ejection probabilities. Our solar system possibly experienced such violence. Some researchers theorize we once had a fifth giant planet alongside Jupiter, Saturn, Uranus, and Neptune during a chaotic period called late instability. Gravitational interactions with other giants would have exiled this unfortunate world. The Nancy Grace Roman Space Telescope, launching by May 2027, will revolutionize rogue planet studies. Its wide field of view images the entire sky 1,000 times faster than Hubble. The Galactic Bulge Time Domain Survey will detect several hundred to several thousand free-floating planets, providing a robust statistical sample. Roman will employ both microlensing and direct infrared imaging, potentially finding planets as small as Mars. Mass measurements from parallax observations will become routine rather than exceptional. Within a few years, astronomers expect to understand rogue planet mass distributions and spatial distributions throughout the galaxy with unprecedented precision. Roman's survey scope extends beyond planets. The low mass range includes ejected planets. Brown dwarfs occupy the middle ground, too massive for planets, insufficiently massive for fusion-like stars. The high end encompasses stellar corpses, neutron stars, and black holes. Astronomers estimate our galaxy contains approximately 100 million stellar mass black holes, but isolated ones remain invisible. Roman's mass-dependent detection method will find them even without nearby matter to illuminate their presence. This comprehensive census of dark objects will reveal the hidden population of massive bodies lurking between stars. How likely is a rogue planet encounter threatening Earth? The odds within the next 1,000 years sit at approximately 1 in 1 trillion. Space's vastness provides protective buffering. Even with trillions of rogue planets wandering the Milky Way, typical distances between objects make close encounters extraordinarily rare. Our solar system occupies a relatively sparse region. Stellar density near the galactic core increases encounter probabilities, but we orbit 26,000 light-years from that dangerous neighborhood. The emptiness between stars shields us far more effectively than any planetary defense system could. but theoretical scenarios illuminate what could happen if one arrived. A rogue planet 13 times Jupiter's mass would gravitationally disrupt every planetary orbit in our system. Earth's path would become more elliptical, carrying us closer to the Sun during perihelion and farther during aphelion. We orbit within the narrow habitable zone where liquid water exists. Increased orbital eccentricity might not push us outside this zone permanently, but it would create extremely short, hot summers followed by prolonged, frigid winters. Compressed growing seasons would devastate agriculture. By the first never-ending winter's end, food shortages could threaten civilization itself. The gravitational architecture would fundamentally transform. Direct collision with Earth would vaporize both worlds, creating debris clouds of molten rock and gas eventually coalescing into new bodies. The kinetic energy from an object moving hundreds of kilometers per second would release energy equivalent to billions of nuclear weapons. Impact temperatures would reach solar surface levels. Any life would be instantly annihilated. More likely, though, a massive intruder would pass through without colliding, scattering inner planets, destabilizing asteroid and comet orbits, and potentially ejecting smaller worlds entirely. The solar system's current configuration represents billions of years of gravitational settling. One massive visitor would reset everything. Our solar system already possesses capture capability under specific circumstances. Research describes permanent weak capture where interstellar objects become trapped in the Sun's orbit through combined gravitational effects from both the Sun and the entire Milky Way galaxy. The mechanism works when an object enters specific regions where the Sun's gravitational influence dominates, particularly around Lagrange points where gravitational forces balance in complex ways. The object initially follows a chaotic orbit, remaining unstable for extended periods before eventually settling into a stable, long-term path. Think of it like a ball rolling on a complex landscape with valleys and hills, eventually finding a stable valley to rest in. The Sun could theoretically collect passing rogue planets and hold them at the solar system's outskirts for millions of years, occasionally trading them with Alpha Centauri, our nearest stellar neighbor. It's entirely possible captured rogue planets already exist in our system's distant reaches, invisible and undetected. This brings us to one of astronomy's most intriguing mysteries. Planet 9. The hypothetical ninth planet lurking in our solar system's outer region has generated fierce scientific debate. Its existence remains unconfirmed, but the evidence supporting it keeps mounting. Astronomers noticed something peculiar about extreme trans-Neptunian objects, bodies beyond Neptune orbiting the Sun at distances averaging more than 250 astronomical units. These distant objects cluster in unusual ways. They tend to make their closest approaches to the Sun in one sector, and their orbits tilt similarly. Random distribution would create scattered orientations. Instead, they align. Something massive appears to be shepherding them gravitationally. Planet 9 would have a mass about 10 times Earth's and orbit roughly 20 times farther from the Sun than Neptune does. At that distance, it would take between 10,000 and 20,000 years to complete a single orbit. Astronomers Konstantin Batygin and Michael Brown proposed several origin scenarios. Perhaps Planet 9 represents a giant planet's core ejected from its original orbit by Jupiter during the solar system's violent youth. Or maybe we captured it from another star during a close stellar encounter billions of years ago. Here's where it gets interesting. Planet 9 might be a rogue planet we captured. The captured rogue planet hypothesis elegantly explains several puzzling features. First, the extreme orbital distance makes sense if the object originated elsewhere and was caught in a weak gravitational trap. Second, the tilted orbit relative to the solar system's plane fits better with capture than with formation in our protoplanetary disk. Third, the mass range matches typical rogue planets detected elsewhere in the galaxy. But finding Planet 9 has proven extraordinarily difficult. The Kuiper belt stretches vast and dark. Any planet at those distances would reflect minimal sunlight, appearing as an incredibly faint dot moving imperceptibly slowly against background stars. Astronomers have conducted large-scale sky surveys and targeted searches based on predicted positions. Both approaches have come up empty so far. Some researchers question whether Planet 9 exists at all. They argue the clustering of trans-Neptunian object orbits might result from observational biases. We discover and track these objects during limited portions of each year when they're visible from Earth. Perhaps the apparent clustering reflects when and where we're looking rather than real gravitational influence from an undiscovered planet. The Vera C. Rubin Observatory, currently under construction in Chile, will settle this debate. Starting operations soon, it will conduct comprehensive sky surveys with unprecedented sensitivity and coverage. Within a few years, Rubin will either find Planet 9 or definitively rule out its existence in the predicted mass and distance ranges. The waiting continues. Whether Planet 9 exists or not, the broader implications remain fascinating. Our solar system's architecture might not be as stable and unchanging as we once assumed. Planets can be captured from interstellar space. Giant worlds might lurk in the darkness beyond Neptune's orbit. The solar system we see today represents just the survivors of a far more chaotic past. What about conditions on rogue planets themselves? Would anything survive there? Without starlight, rogue planets exist in extraordinary cold and darkness. The outer layers, exposed directly to space, reach temperatures just a handful of degrees above absolute zero. At those temperatures, molecules barely move. Chemistry essentially stops. Any water would be frozen harder than granite. Atmospheres, if they existed initially, would condense and freeze solid on the surface, creating bizarre landscapes of nitrogen ice and frozen carbon dioxide. But deep beneath the surface, conditions differ dramatically. Planetary formation releases enormous heat. Large planets contain radioactive elements whose decay generates warmth for billions of years. Tidal heating from internal friction can add energy if the planet has large moons. These internal heat sources persist regardless of whether a star shines overhead. Scientists theorize that sufficiently large rogue planets might maintain subsurface liquid water oceans insulated by thick ice shells. Similar oceans exist beneath the ice covering Jupiter's moon Europa and Saturn's moon Enceladus, kept liquid by tidal heating despite surface temperatures around -200 degrees Celsius. A rogue planet's internal heat could sustain comparable environments. Could life exist in those dark oceans? On Earth, entire ecosystems thrive around deep-sea hydrothermal vents, completely independent of sunlight. Chemosynthetic bacteria derive energy from chemical reactions between vent minerals and seawater. Complex organisms have evolved to exploit these bacteria, creating food webs in absolute darkness. A rogue planet's subsurface ocean might host similar chemistry. Hydrothermal vents could form where the molten core's heat reaches the ocean floor. Dissolved minerals would provide chemical energy. Life wouldn't need photosynthesis, just chemistry. The same fundamental processes that began life on Earth billions of years ago could operate beneath a rogue planet's frozen surface. This possibility transforms our understanding of habitability. We've focused intensely on planets orbiting within their star's habitable zone where surface temperatures permit liquid water. But if life can exist in subsurface oceans heated by internal planetary processes, habitability extends throughout the galaxy. Every sufficiently large rogue planet becomes potentially habitable regardless of distance from any star. Billions to trillions of rogue planets wander our galaxy. If even a tiny fraction harbor subsurface biospheres, life might be far more common than we've imagined. The galaxy might teem with living worlds we can't even detect from Earth because they emit no light and reflect none from nearby stars. Detection remains the critical challenge. How do you find life on a dark, frozen world drifting through interstellar space? Traditional biosignatures like oxygen or methane in atmospheres don't work because rogue planet atmospheres freeze solid. Remote sensing can't detect subsurface oceans through kilometers of ice. Even if we identified a rogue planet passing relatively nearby, sending a probe would require decades or centuries of travel time. The best near-term hope might be detecting heat signatures. Large rogue planets with active internal geology should emit detectable infrared radiation. Future space telescopes optimized for infrared detection might catalog thousands of rogue planets based on their thermal emissions. Analyzing those heat signatures could reveal which planets likely maintain internal warmth sufficient for subsurface liquid water. Beyond detection, the philosophical implications stagger the imagination. If life commonly exists on rogue planets, it evolved in eternal darkness, powered by planetary heat rather than starlight. Such life would have no concept of day or night, seasons, or the sky. Their entire universe would be the ocean they inhabit, bounded by ice above and rock below. They would never see stars. Would such life develop intelligence? Could technological civilization emerge in a subsurface ocean with no access to fire, metals, or the night sky that inspired human astronomy? These questions remain purely speculative, but they challenge our assumptions about life's requirements and possibilities. The rogue planet phenomenon forces us to reconsider everything we thought we knew about planets, habitability, and our place in the cosmos. These worlds represent a hidden population potentially outnumbering conventional planets around stars. They demonstrate that planet formation is far more violent and chaotic than we imagined, regularly casting worlds into the void. They might harbor life in forms we've never considered, thriving in darkness and cold on chemistry alone. As detection capabilities improve and our catalog of rogue planets expands, we'll answer fundamental questions about planetary demographics, formation mechanisms, and habitability limits. The Nancy Grace Roman Space Telescope will provide unprecedented statistical data. Future infrared observatories will probe their physical characteristics. Eventually, we might even send probes to study nearby examples directly. The rogue planet story represents more than just astronomical curiosity. It's a window into the violent chaos underlying cosmic order. Consider what happens during planet formation. You picture a serene disk of gas and dust gradually coalescing into worlds. The reality involves gravitational warfare. Planets collide, merge, scatter each other like cosmic billiard balls. Some grow massive enough to dominate. Others get flung into the void at thousands of kilometers per second. Computer simulations reveal the brutality. Researchers modeled 100 planetary systems, each containing 3 to 10 planets in stable circular orbits around Sun-like stars. They ran the physics forward for 1 billion simulated years. Nearly every system eventually destabilized. Close encounters between planets triggered cascading chaos. The typical outcome: at least one gas giant ejected. 5 to 10 smaller rocky worlds cast into darkness. Our solar system probably followed this pattern. Evidence suggests we once had a 5th giant planet. During a chaotic period astronomers call late instability, gravitational interactions with Jupiter, Saturn, Uranus, and Neptune shifted this world's orbit. Eventually, those same giants exiled it completely. That lost planet now wanders interstellar space, a frozen monument to our system's violent youth. Binary star systems produce even more chaos. When two stars orbit each other, any planets face competing gravitational influences. Passing stars can perturb those wide binary orbits, occasionally sending the pair much closer together. The resulting gravitational turbulence ejects planets or even steals worlds from one star and gives them to its companion. More than half of all star systems are binary. The rogue planet production rate might dwarf single-star systems like ours. But here's where it gets stranger. Some rogue planets never orbited any star. They formed alone in interstellar space through processes resembling star formation. Collapsing clouds of gas and dust contract under their own gravity, accreting material directly from surrounding nebulae. They just don't gather enough mass to ignite fusion. Instead, they halt at planetary scales, becoming objects the International Astronomical Union calls sub-brown dwarfs. How do you tell these formation pathways apart? Velocity provides the crucial fingerprint. Ejected planets retain significant excess speeds, typically 8 to 16 kilometers per second. That's much higher than normal stellar velocities in star-forming regions. Planets ejected by binary star interactions move even faster, roughly 3 times the velocity of those scattered by planet-planet collisions. These measurements let astronomers reconstruct each world's origin story from its current motion alone. The numbers grow more staggering the deeper you examine them. Current estimates suggest 7 rogue planets per star in our galaxy. With 400 billion stars, that implies nearly 3 trillion wandering worlds. Some researchers propose even higher ratios, potentially 20 rogue planets per star. 8 trillion dark spheres drifting through the Milky Way. More planets floating free than orbiting stars. This fundamentally rewrites planetary demographics. We've spent decades cataloging exoplanets around other stars. Thousands discovered. Potentially billions waiting. But the true planetary population might lie hidden in darkness, vastly outnumbering conventional worlds. Most planets in the galaxy could be rogues. Recent discoveries confirm their abundance. In January 2026, astronomers announced finding a Saturn-mass rogue planet about 9,800 light-years away. What makes this discovery revolutionary? For the first time, researchers directly measured a rogue planet's mass rather than making statistical estimates. The observation combined ground telescopes and space instruments to triangulate distance and calculate mass with unprecedented precision during a microlensing event. Another 2025 discovery revealed truly bizarre behavior. A rogue planet designated Cha 1107 to 7626 sits about 620 light-years away in Chameleon. Its mass is 5 to 10 times Jupiter. And it's eating. This world accretes gas and dust from surroundings at 6 billion tons per second, the strongest accretion episode ever recorded for any planetary mass object. It behaves more like a forming star than a planet, with magnetic activity driving material infall in ways previously thought exclusive to stellar evolution. These discoveries raise profound questions about what defines a planet. If objects form through star-like processes but halt at planetary masses, are they really planets? If worlds wander freely never having orbited stars, do they count? Traditional definitions centered on objects orbiting stars. Rogue planets demolish those boundaries. The categories blur. We need new frameworks. Detection remains extraordinarily difficult. Rogue planets emit virtually no light. They reflect none from nearby stars because there are no nearby stars. Microlensing offers the primary technique. When a rogue planet passes directly between Earth and a distant star, its gravity bends the star's light, briefly magnifying its brightness. These events last hours to days maximum. You must monitor millions of stars continuously to catch them. Survey teams point telescopes toward Sagittarius, looking toward our galaxy's dense central bulge where 400 million detectable stars crowd together. Just 1 in 100,000 stars undergoes microlensing at any moment. You need massive statistical samples. In 2007, the OGLE project identified 611 candidate events. MOA found 488. Those numbers have grown as detection networks expanded, but identifying the brief signature of a planet within stellar microlensing events requires extraordinary precision. The microlensing light curve reveals mass, but distance remains approximate. When dealing with objects tens of thousands of light-years away, uncertainties span thousands of light-years themselves. You know a planet exists. You know its approximate mass. But pinpointing its location stays frustratingly vague. Everything changes with the Nancy Grace Roman Space Telescope launching by May 2027. This instrument images the entire sky 1,000 times faster than Hubble. Roman's Galactic Bulge Time Domain Survey should detect several hundred to several thousand rogue planets, providing the first comprehensive statistical census. The mission will employ both microlensing and direct infrared imaging, potentially finding worlds as small as Mars. Roman will also catalog the broader population of dark objects lurking between stars. Beyond rogue planets lie brown dwarfs, objects too massive for planets but insufficient for stellar fusion. Beyond those sit stellar corpses, neutron stars and black holes. Astronomers estimate 100 million stellar mass black holes drift through our galaxy, but we've never definitively found one floating alone because they're invisible. Roman's mass-based detection will find them regardless of nearby matter revealing their presence. This comprehensive dark object census will answer fundamental questions about galactic structure. How much unseen mass fills interstellar space? How violent was early planet formation across the galaxy? What fraction of planets get ejected versus remaining in stable systems? These answers reshape our understanding of cosmic architecture. The threat assessment remains clear. Odds of a rogue planet entering our solar system within the next 1,000 years. Approximately 1 in 1 trillion. Space is incomprehensibly vast. Even with trillions of rogue planets wandering the Milky Way, distances between objects make close encounters vanishingly rare. The emptiness serves as natural protection. Our solar system remains effectively isolated despite the hidden population surrounding us. But what if? If a rogue planet did enter our system, consequences would depend entirely on mass, trajectory, and approach distance. A Jupiter-mass world passing through the outer solar system might only slightly perturb orbits. A super-Jupiter 13 times more massive could fundamentally reshape everything. Earth's orbit might become more elliptical, shifting us closer to the Sun during perihelion and farther during aphelion. Even without direct collision, orbital changes would prove catastrophic. Shorter, hotter summers. Longer, frozen winters. Growing seasons collapse. Food production fails globally. Civilization faces extinction not from impact but from climate chaos induced by gravitational perturbation alone. If we were unlucky enough to face direct collision. Complete annihilation. Both worlds vaporize. The energy release would match billions of nuclear weapons simultaneously detonating. Yet our solar system might already host captured rogues. Research suggests the Sun can permanently trap passing interstellar objects through a mechanism called permanent weak capture. Combined gravitational effects of the Sun and the entire Milky Way galaxy can draw objects into chaotic orbits that eventually stabilize. These captured worlds might orbit at extreme distances, occasionally trading with nearby Alpha Centauri through complex gravitational interactions. Planet 9 could represent exactly this scenario. A hypothetical world 10 times Earth's mass orbiting 20 times farther than Neptune might explain the peculiar clustering of extreme trans-Neptunian objects. These bodies beyond Neptune orbit at distances averaging more than 250 astronomical units, and their orbits cluster suspiciously. Some astronomers propose Planet 9 was captured from another star. Others suggest it formed in our system but got kicked to distant orbits. The Vera Rubin Observatory under construction in Chile should either find it or definitively rule out its existence within years. Rogue planets force us to confront how little we actually know about our cosmic neighborhood. The galaxy contains hidden populations vastly outnumbering visible objects. Most planets might wander in eternal darkness. Some potentially harbor life in subsurface oceans heated by internal geology rather than starlight. We've built our understanding of habitability around stars, but the true story might unfold in places we can't even see. The darkness between stars isn't empty. It's filled with exiled worlds carrying the scars of ancient violence, drifting through the void as silent testimony to the chaos underlying cosmic order. Every planet we've ever studied orbits a star. Every definition of habitability we've constructed assumes stellar energy. Our entire framework for understanding planetary systems depends on gravitational hierarchies centered on fusion-powered spheres of plasma. Rogue planets demolish these assumptions completely. They represent a population potentially outnumbering conventional planets by factors we're only beginning to grasp. 8 trillion wandering worlds might drift through our galaxy alone. More planets floating free than orbiting stars. Most of the galaxy's planetary population exists in permanent darkness, following trajectories determined by violent ejections billions of years ago. Think about what that means for the search for life. We've focused exclusively on planets within habitable zones around stars, narrow bands where surface temperatures permit liquid water. But if large rogue planets maintain subsurface oceans heated by internal geology, habitability extends throughout interstellar space. Every sufficiently massive rogue becomes potentially habitable regardless of stellar proximity. Life on Earth began in darkness. Hydrothermal vents on the ocean floor supported entire ecosystems before photosynthesis even evolved. Those same chemical processes could operate beneath kilometers of ice on rogue planets, powered by planetary heat rather than sunlight. If even a small fraction of wandering worlds harbor such biospheres, life might be far more common than every optimistic estimate we've made based on stellar habitable zones. We'd never detect it with current methods. These hypothetical organisms would exist in sealed environments completely isolated from space. No atmospheric biosignatures to detect. No reflected starlight to analyze. Just dark frozen spheres drifting through the void, potentially teeming with alien chemistry in oceans we can't observe from trillions of kilometers away. The Nancy Grace Roman Space Telescope will transform this field when it launches. Hundreds or thousands of new detections. Direct mass measurements becoming routine rather than exceptional. Statistical distributions revealing formation mechanisms and ejection dynamics. Within a decade, we'll understand rogue planet demographics with precision currently impossible. But understanding population statistics differs dramatically from studying individual worlds up close. Sending probes to nearby rogue planets requires technology we don't yet possess. Interstellar travel at meaningful speeds remains firmly in the realm of theoretical physics. Even identifying which nearby rogue planets might harbor subsurface oceans presents challenges requiring infrared observatories we haven't built yet. The philosophical implications cut deeper than detection challenges. If technological civilizations can emerge in subsurface oceans on rogue planets, they develop completely different relationships with the cosmos than we have. No sky to observe. No stars to navigate by. No concept of day or night, seasons, or the broader universe beyond their frozen shell. Their entire cosmology would center on the ocean they inhabit, bounded by ice above and rock below. Would they ever develop astronomy? Could they even discover they existed on a planet without external reference points? The night sky inspired humanity's earliest scientific questions. What happens to curiosity about the universe when you live in eternal darkness with no universe visible? Our own solar system carries scars from the same violence that creates rogue planets. We probably ejected a fifth giant planet during late instability. The asteroid belt structure reflects ancient gravitational chaos. The late heavy bombardment that scarred the moon resulted from orbital reshuffling among our gas giants. We're survivors of the same chaotic processes that filled the galaxy with wanderers. Planet 9, if it exists, might represent a captured rogue. Or perhaps a native world kicked to extreme distances. Either way, its potential presence reveals how dynamic and unstable even mature planetary systems remain. The solar system we observe today isn't frozen in place. It's a temporary configuration that will eventually change again given enough time. The statistical risk remains infinitesimal. 1 in 1 trillion chance of a rogue planet entering our solar system within the next 1,000 years. Space's vastness protects us far more effectively than any defense system. But the theoretical consequences illuminate just how delicately balanced our existence actually is. Earth's stable orbit within the habitable zone depends on gravitational architecture that took billions of years to establish. One massive intruder could reset everything. We don't need to worry about rogue planets destroying Earth. We need to understand them because they represent the majority of our galaxy's planetary population. They reveal how violent and chaotic planet formation actually is. They potentially harbor life in forms we never considered. They challenge every assumption we've made about what planets are and where they can exist. The darkness between stars isn't empty void. It's filled with worlds. Some larger than Jupiter. Others smaller than Earth. All carrying chemical signatures from their birth systems. All following trajectories determined by gravitational violence that occurred when their parent systems were young and unstable. They're monuments to chaos, frozen in time, drifting through the galaxy as permanent reminders that the ordered solar system we inhabit represents a rare exception rather than the cosmic norm. Detection methods will improve. Catalogs will expand. Eventually, we'll understand rogue planet populations with the same precision we currently understand exoplanets around stars. The Nancy Grace Roman Space Telescope starts this process. Future infrared observatories will continue it. Decades from now, we might even send probes to study nearby examples directly, analyzing their composition and searching for signs of subsurface oceans. But the fundamental revelation has already occurred. Most planets in the galaxy don't orbit stars. They wander alone through interstellar space, ejected during the violent formation of planetary systems or formed independently in collapsing gas clouds. The universe contains far more dark worlds than illuminated ones. We've spent centuries studying the exceptions while remaining completely ignorant of the rule. Rogue planets aren't anomalies. They're the norm. The galaxy is filled with orphaned worlds drifting through darkness, and we're only beginning to understand what that means for planet formation, the prevalence of life, and our place in a cosmos far stranger than we imagined.
A moon the size of Arizona is shooting water into space right now. Not metaphorically, literally spraying massive plumes of liquid water hundreds of kilometers above its frozen surface. And we've been watching it happen for nearly 2 decades. Yet somehow, most people still don't know that Enceladus exists, let alone that it might be harboring life beneath its icy shell. That's about to change, because recent discoveries have transformed this tiny world from a curiosity into what might be the single most important place in our solar system. Enceladus orbits Saturn, tucked away in the outer reaches where sunlight is 100 times weaker than on Earth. At first glance, it's nothing special. Just another ice-covered rock barely 500 kilometers across, small enough to fit inside the borders of Texas. But in 2005, the Cassini spacecraft flew past and spotted something that made absolutely no sense. Jets of water vapor and ice particles were erupting from cracks near the South Pole, shooting material so high that it was feeding Saturn's earring. This wasn't supposed to be possible. Small moons are supposed to be geologically dead, their interiors long frozen solid. Enceladus was breaking every rule we thought we understood about planetary science. The discovery sparked an immediate question. Where was all that water coming from? You can't just spray hundreds of kilograms of material into space every second without a source. The only explanation that fit the data was shocking. Beneath that frozen crust, Enceladus was hiding a global ocean of liquid water. Not puddles, not ice, an actual ocean, potentially tens of kilometers deep, sitting in direct contact with a rocky core. That alone would make Enceladus remarkable, but what happened next pushed it into an entirely different category. Scientists didn't just detect water in those plumes. They found organic molecules, the carbon-based building blocks that life as we know it requires. Then they found salts, suggesting the ocean was interacting chemically with rock below. Then hydrogen gas, a potential energy source for microbial life. Then silica particles that could only form under very specific hydrothermal conditions. Each new measurement added another piece to a puzzle that was starting to look disturbingly biological. Think about what this means for a moment. Every single ingredient we consider essential for life, water, organic chemistry, energy sources, and mineral-rich environments, exists right now on a moon that's barely larger than some asteroids. We're not speculating about ancient conditions that might have existed billions of years ago. We're talking about an active, chemically rich ocean that's being sampled in real time as Enceladus conveniently sprays its contents into space where our instruments can analyze them. The implications hit differently when you understand how rare this combination actually is. Mars probably had water billions of years ago, but it's mostly gone now. Europa definitely has an ocean, but it's trapped beneath kilometers of solid ice with no easy way to sample it. Titan has lakes and rivers, but they're made of liquid methane at temperatures where water is harder than granite. Enceladus is the only place in the solar system where we've found all the right conditions actively operating and accessible for study, all at the same time. But here's where the story gets genuinely strange. The more we learned about Enceladus, the less sense it made. This moon should be frozen solid, a dead chunk of ice that stopped being interesting billions of years ago. Its small size means it should have lost its internal heat almost immediately after formation. There's no large planet nearby generating tidal forces strong enough to keep it warm. By every conventional model, Enceladus should look like our own moon, cold, cratered, and utterly lifeless. Instead, it's one of the most geologically active bodies we've ever discovered. Something is pumping enormous amounts of energy into this tiny world, enough to maintain a liquid ocean and power those dramatic plumes. For years, scientists assumed tidal heating from Saturn must be responsible. As Enceladus orbits, Saturn's gravity flexes and squeezes it, converting that mechanical stress into heat through friction. It's the same process that keeps Jupiter's moon Io volcanically active. The math seemed to work, barely, though it required some generous assumptions about the moon's interior structure. Then in 2014, measurements from Cassini's final close flybys revealed something that made those calculations fall apart. The amount of heat coming from Enceladus was far higher than tidal forces alone could explain. We're talking about 10 times more energy than the models predicted. That's not a small discrepancy you can explain away with measurement error. That's a fundamental problem requiring a completely different explanation, one that scientists are still actively debating. Several competing theories have emerged, each more interesting than the last. Some researchers think the moon's core might contain radioactive elements generating heat through decay, though that doesn't fully explain the localized heating near the South Pole. Others propose that the ocean itself might be chemically active, with reactions between water and rock producing heat along with those hydrogen molecules we detected. A few scientists have even suggested that tidal forces might be working in unexpected ways, perhaps amplified by the specific structure of Enceladus's interior in ways we don't yet understand. What makes this mystery particularly compelling is what it means for life. If conventional tidal heating can't explain Enceladus's energy budget, and if chemical reactions in the ocean are contributing significantly to that heat, then we're looking at an environment that's not just potentially habitable but actively energetic. Life needs energy. The more energy available, the more complex the chemistry that becomes possible. Earth's deep-sea hydrothermal vents, powered by similar chemical reactions, support entire ecosystems in complete darkness. Could something similar exist on Enceladus? The chemistry we've detected in those plumes keeps pointing in that direction. In 2018, complex organic molecules were identified, including compounds with chains of carbon atoms that on Earth are typically associated with biological processes. These weren't simple molecules like methane. We found materials like benzene and other aromatic hydrocarbons, structures that require specific conditions to form. While these can be produced through non-biological chemistry, their presence alongside water, salts, and hydrogen creates what astrobiologists call a biosignature ensemble, a collection of indicators that together become harder to explain without invoking life. Then came the phosphorus discovery in 2023, announced just months ago. Phosphorus is the final piece of the puzzle, the last major element required for life as we know it that we hadn't yet confirmed on Enceladus. It's essential for DNA, for cell membranes, for the energy-carrying molecules that power every living thing on Earth. Finding it in Enceladus's ocean, in a form that's accessible and reactive, meant that every single chemical requirement for life was now confirmed. Not theoretically possible. Actually present, measured, verified. Scientists don't use the word proven lightly when it comes to extraterrestrial life, and for good reason. Detecting the chemical ingredients for life isn't the same as detecting life itself. You can have all the right components without ever assembling them into something living. Earth's early oceans probably contained everything necessary for life for millions of years before the first organisms actually emerged. Chemistry alone doesn't equal biology. But the case for Enceladus keeps getting stronger in ways that go beyond simple chemical inventories. The plume composition varies over time, suggesting active processes in the ocean below rather than just static chemistry. The heat distribution across the South Pole shows patterns consistent with active hydrothermal systems. The very existence of those tiger stripe fractures, the cracks from which the plumes emerge, implies ongoing geological activity driven by forces we're still trying to understand. Each observation adds another layer of complexity, another hint that something more than simple chemistry might be happening beneath that ice. What we're really talking about is probability. How likely is it that an environment with liquid water, organic molecules, energy sources, minerals, and chemical disequilibrium would remain sterile? On Earth, life appears almost immediately wherever these conditions exist. We find organisms in boiling acid pools, in frozen Antarctic lakes, kilometers deep in solid rock, anywhere liquid water touches chemistry and energy. The assumption that Earth is somehow special, that life only emerged here despite similar conditions existing elsewhere, requires increasingly creative explanations. The scientific community is being careful, as it should be. Extraordinary claims require extraordinary evidence, and detecting actual life on Enceladus would be the most extraordinary claim in human history. We need direct measurements, not just circumstantial evidence. We need to actually sample that ocean in ways that can definitively distinguish biological from non-biological chemistry. We need missions specifically designed to search for life, not just general reconnaissance flybys analyzing whatever happens to drift through the plumes. Those missions are coming. NASA and ESA both have proposals for Enceladus-focused spacecraft that would do more than just fly past. They'd orbit the Moon, sampling the plumes repeatedly with instruments specifically designed to detect biosignatures. They'd map the surface in detail, looking for the best places to eventually land. Some designs even include penetrators that could punch through the ice to sample the ocean directly, though that technology is still being developed. The political and funding challenges are real, but the scientific justification has never been stronger. Because here's what makes Enceladus different from every other target in the search for life. We don't need to dig. We don't need to drill through kilometers of ice or land in some specific location and hope we got lucky. The Moon is literally handing us samples, spraying its ocean into space where we can collect and analyze them. It's the easiest astrobiological target in the solar system, if we're willing to go there with the right tools. The implications extend far beyond one small moon. If we find life on Enceladus, if we confirm that biology emerged independently in a completely different environment from Earth, it changes everything we think we know about life's prevalence in the universe. It would mean that given the right conditions, life isn't rare. It isn't a miracle. It's almost inevitable, a natural consequence of chemistry and physics operating under certain parameters. And if those conditions exist on a tiny moon orbiting Saturn, how many other places in our galaxy might harbor life we never imagined possible? the universe suddenly becomes less lonely, less special, less centered on Earth as the one place where the cosmic lottery paid off. And that realization, that fundamental shift in perspective, might matter more than any individual discovery. But before we get too far ahead, we need to understand what we're actually looking for. The word life gets thrown around as if it has one clear meaning, but the reality is far more complex. When scientists talk about detecting life on Enceladus, they're not imagining fish swimming in dark oceans or forests of alien kelp. They're thinking about chemistry that behaves in ways non-living chemistry doesn't. They're looking for patterns, disequilibrium, organization that requires explanation beyond simple physics. The challenge is distinguishing biological signatures from clever geology. Earth's history is full of examples where scientists thought they'd found ancient life, only to discover later that minerals and chemical reactions could produce similar structures. Meteorites have arrived on Earth containing organic molecules that looked suspiciously biological until detailed analysis revealed purely abiotic origins. Mars has rock formations that resemble stromatolites, those layered structures built by microbes, except they're probably just weathered sediment. The universe is surprisingly good at mimicking life without actually being alive. So what would constitute genuine evidence? The gold standard would be finding cells, actual membrane-bound structures with internal organization distinct from their environment. But even that has complications. Would alien cells look like Earth cells? Would they have membranes made of lipids, or something completely different? Would they be large enough to see, or so small they'd slip through our filters? We're designing detection systems based on the one example of life we know, which might be like trying to recognize all possible music by only ever hearing jazz. Chemical signatures offer more immediate possibilities. Life on Earth produces characteristic ratios of molecules that don't occur in non-living systems. Amino acids, the building blocks of proteins, come in left-handed and right-handed versions that are mirror images of each other. Non-biological processes produce equal amounts of both. Earth life uses exclusively left-handed amino acids, a quirk of our biochemistry that creates an obvious signature. If Enceladus samples show similar homochirality, similar preference for one molecular form over another, that's hard to explain without biology. Isotope ratios provide another line of evidence. Living organisms preferentially use lighter isotopes in their metabolism because they're slightly easier to move around chemically. This creates distinctive patterns in carbon, nitrogen, and sulfur isotopes that differ from non-biological distributions. We can measure these ratios with extraordinary precision, looking for the subtle skewing that life produces. The trick is proving that what we're measuring reflects biology rather than some unknown geological process that just happens to produce similar patterns. Energy metabolism leaves traces too. Life on Earth operates far from chemical equilibrium, constantly pumping electrons through molecular machinery to extract energy. This creates specific chemical gradients, particular molecules that shouldn't coexist in stable environments but do because organisms are actively maintaining them. Finding these disequilibrium signatures in Enceladus samples would be suggestive, though not definitive without understanding the full geochemical context. The ideal scenario involves multiple independent lines of evidence converging. Not just one suspicious molecule, but a whole suite of measurements that collectively make more sense with biology than without. Homochiral amino acids plus isotope fractionation plus disequilibrium chemistry plus organized structures plus something we haven't even thought of yet. The more ways the evidence points in the same direction, the harder it becomes to construct alternative explanations. Current technology can't quite get us there. Cassini's instruments, remarkable as they were, weren't designed for life detection. They measured what was in the plumes at a general level, identifying broad chemical categories rather than the specific molecular details that would reveal biological signatures. It's like knowing a building contains organic compounds versus being able to read the individual books in its library. We need the equivalent of those books, the fine details that only purpose-built instruments can provide. Those instruments exist, at least in prototype form. Mass spectrometers sensitive enough to detect individual molecules at parts per trillion concentrations. Microscopes that can image structures smaller than the wavelength of light. DNA sequencers adapted for potential alien biochemistry, though that assumes alien life uses something like DNA, which is far from certain. The technology for rigorous life detection isn't science fiction. It's engineering and funding. The real constraint is getting that technology to Enceladus. Saturn orbits nearly 1.5 billion kilometers from Earth, requiring years of flight time even with gravitational assists from other planets. The radiation environment around Saturn is harsh, degrading electronics and requiring heavy shielding that adds mass and cost. Landing on Enceladus, or even achieving a stable orbit around it, demands propulsion systems and guidance precision we've only recently developed. Every kilogram of scientific instruments requires additional kilograms of fuel, structure, power, and communication equipment. Mission designers face brutal trade-offs. Do you maximize the number of plume passes with simpler instruments, or do you make fewer passes with more sophisticated equipment? Do you focus on chemical analysis, or invest in imaging systems that might spot surface features indicating subsurface activity? Do you design for flexibility, able to adapt to unexpected discoveries, or optimize for specific measurements with less margin for surprise? There's no perfect answer, only different gambles about what matters most. One proposed approach is the orbiter concept, a spacecraft that would spend years circling Enceladus, passing through the plumes dozens of times with instruments specifically designed for biosignature detection. Each pass samples different parts of the plume under different conditions, building up statistical confidence about what's actually there versus contamination or instrument error. The spacecraft would also map the surface in unprecedented detail, identifying patterns in the ice that might reveal circulation patterns in the ocean below. Another approach is more ambitious, a lander that would touch down near the tiger stripes, directly in the path of the plumes as they erupt. This eliminates the need for high-speed sampling, allowing more time for detailed analysis of fresh material before it's altered by space radiation or surface chemistry. The lander could also drill or melt through surface ice, looking for organic materials that might have frozen out of the plumes over time. The technical challenges are enormous, but the scientific payoff could be transformative. The most aggressive proposals involve actual ocean access, penetrators that would punch through the ice to sample the water directly. This is decades away technologically, requiring not just the ability to get through potentially 20 kilometers of ice, but to survive the pressure, communicate through that much solid material, and operate in a completely dark ocean with no GPS, no external power source, and no possibility of retrieval if something goes wrong. It's the kind of challenge that makes Mars sample return look straightforward. Cost is the other reality. Cassini's entire mission, from development through operations, ran about $3 billion. A dedicated Enceladus life detection mission would likely cost similar amounts or more, depending on complexity. That's money competing with dozens of other scientific priorities, from exoplanet observations to particle physics to climate monitoring. The political process of allocating those resources involves scientific merit but also timing, advocacy, public interest, and sometimes factors that have nothing to do with the science itself. Yet something is shifting in how seriously the scientific establishment takes Enceladus. When Cassini first revealed those plumes, the reaction was fascination but also caution. Now major space agencies are actively planning follow-up missions. Papers on Enceladus biology appear in top-tier journals. Astrobiologists who once focused exclusively on Mars are redirecting attention to icy moons. The evidence has reached a threshold where ignoring Enceladus feels harder to justify than investigating it. Part of this shift comes from parallel discoveries about life on Earth. The more we explore extreme environments here, the more we realize how adaptable life actually is. Organisms don't just tolerate extreme conditions, they thrive in them, sometimes growing better under pressure or in darkness than in supposedly optimal environments. We've found ecosystems kilometers underground powered entirely by chemical energy from rocks, no sunlight required. We've discovered microbes that reproduce in boiling acid, in Antarctic ice, in the cooling water of nuclear reactors. The boundaries of where life can exist keep expanding every time we look. These discoveries aren't just academic curiosities. They directly inform what we should expect on Enceladus. If Earth life can survive in environments that superficially resemble that subsurface ocean, environments with similar chemistry and energy sources and physical conditions, then the argument that Enceladus is too extreme for biology becomes much harder to sustain. The question shifts from whether life could exist there to whether it actually does, which is a very different kind of uncertainty. Human summary. One way to think about this is through probability layering. The chances that any single piece of evidence points to life might be small. But when you stack independent lines of evidence, the cumulative probability shifts. Enceladus has liquid water, confirmed. It has organic molecules, confirmed. It has energy sources in the form of chemical disequilibrium, strongly indicated. It has the right pH range based on plume chemistry. It has a rocky core providing minerals and catalytic surfaces. Each of these alone is interesting. Together, they describe an environment that looks increasingly similar to places on Earth where life not only exists but dominates. This doesn't prove biology. It does mean the null hypothesis, the assumption that Enceladus is sterile, requires stronger justification than it once did. You can't simply point to the cold or the darkness or the isolation and declare life impossible when we found terrestrial organisms thriving under all those conditions. The burden of proof hasn't fully reversed, but it's definitely shifting. There's also a philosophical dimension here that scientists don't always articulate but definitely think about. If we find life on Enceladus, it answers one of the oldest questions humans have ever asked. If we don't find it despite those conditions being right, that tells us something almost as important. It means the origin of life is harder than we thought, that even with all the right ingredients, biology doesn't just spontaneously emerge. Either outcome fundamentally changes how we understand our place in the universe. That's why some researchers push so hard for this mission despite the cost and complexity. It's not just curiosity, though that's part of it. It's the recognition that opportunities like this are rare. We've explored dozens of worlds and found exactly one with confirmed life. Now we have a second candidate that's actually accessible, that's literally spraying its ocean into space for us to sample. Walking away from that because it's expensive or difficult feels like a choice future generations would struggle to understand. The detection strategy matters as much as the mission design. What exactly would constitute proof of life in those plumes? This isn't as straightforward as it sounds. Organic molecules alone won't cut it. We've already found those, and they could easily form through non-biological chemistry. Simple amino acids, the building blocks of proteins, exist in meteorites that have never been near life. Even complex organics can emerge from purely chemical processes given the right conditions and enough time. The gold standard would be something that screams biology, something chemistry alone can't easily explain. Patterns in how molecules are arranged. Life on Earth shows strong preferences, or chirality, in molecular structure. Amino acids come in left-handed and right-handed forms that are mirror images. Terrestrial biology almost exclusively uses left-handed amino acids. If Enceladus samples showed a similar bias, that would be striking because random chemistry produces equal amounts of both. A strong imbalance suggests selection, and selection usually means biology. Another marker would be isotopic ratios. Living organisms preferentially use lighter isotopes of elements like carbon and nitrogen because they're slightly easier to incorporate into biochemical reactions. This leaves a measurable signature, a deviation from what you'd expect in non-biological material. Multiple measurements showing consistent isotopic fractionation across different molecules would be hard to explain without invoking life. Then there are structural signatures. Cell membranes, even if the cells themselves are destroyed during sampling, might leave behind lipid patterns or vesicular structures visible under microscopy. Polymers with repeating units, especially if they show sequence complexity rather than random repetition, would raise serious questions. Biology builds information-rich structures. Chemistry builds repetitive or random ones. Spotting that difference requires incredibly sensitive instruments and careful analysis, but it's doable. The challenge is that we're looking for life that might not resemble Earth biology at all. It probably uses the same basic chemistry, water and carbon and energy flow, but the details could be wildly different. Maybe it doesn't use DNA. Maybe its proteins are built from different amino acids. Maybe it has cell walls made of materials we've never seen in terrestrial organisms. Designing instruments that can detect life as we know it while remaining open to life as we don't know it is one of the hardest problems in astrobiology. This is where contamination becomes a nightmare scenario. If we send a spacecraft that isn't perfectly sterilized and it finds organic molecules, how do we know they're not hitchhikers from Earth? Cassini wasn't built with biology in mind, so its cleanliness standards were lower than what we'd need now. Any future mission would require planetary protection protocols far more stringent than anything Cassini followed. That means building the spacecraft in ultra-clean facilities, sterilizing components with heat or radiation, and proving through rigorous testing that contamination levels are below detection thresholds. Even then, interpretation will be contentious. Science operates on reproducibility and peer review. One measurement showing possible biosignatures won't be enough. We'd need multiple independent detections, ideally using different techniques. We'd need ground-based labs analyzing return samples, if we can manage that technologically. We'd need theoretical models explaining how those signatures could arise biologically and why they couldn't arise chemically. The bar for claiming discovery of extraterrestrial life is extraordinarily high, as it should be. But here's what makes Enceladus different from every other target in astrobiology. The ocean is sampling itself. We don't have to drill through kilometers of ice or land on a hostile surface or wait decades for a rover to reach the right location. The plumes bring ocean material directly to us, fresh and uncontaminated by surface processes. That accessibility is transformative. It means the first real test of whether a subsurface ocean harbors life could happen within our lifetimes, maybe within the next 20 years if funding and planning align. Compare that timeline to Europa, where we're still debating whether we can even confirm the ocean's existence from orbit before committing to a lander. Or Titan, where the interesting chemistry is happening in lakes of liquid methane at 90 degrees above absolute zero, requiring entirely different instrument designs. Or Mars, where we've been searching for decades and still haven't found anything definitive despite constant presence on the surface. Enceladus offers a shortcut, a way to answer the life question without solving all the intermediate technical problems first. The geopolitical aspect is less discussed but increasingly relevant. Space exploration has always involved international cooperation and competition in varying measures. Right now, NASA is leading most serious Enceladus mission concepts, but ESA, the European Space Agency, has expressed strong interest in collaborative efforts. China's space program is expanding rapidly and has mentioned icy moon missions in long-term planning documents. Russia, despite economic constraints, maintains significant expertise in planetary science. A discovery of life on Enceladus wouldn't belong to any single nation. The scientific data would be shared globally, analyzed by researchers across dozens of countries, debated in international conferences and journals. But the prestige of making that discovery, of being the agency that sent the spacecraft that found it, matters. It shapes funding priorities, inspires the next generation of scientists and engineers, and demonstrates technological capability. These factors influence which missions get approved and when. There's also the private sector angle. Companies like SpaceX have dramatically reduced launch costs, which directly affects mission feasibility. A mission that would have cost $5 billion a decade ago might now be achievable for $3 billion because getting the spacecraft off Earth is cheaper. If that trend continues, the financial barrier to ambitious outer solar system missions drops steadily. That could accelerate timelines or allow more complex mission designs than traditional budgets would support. Would commercial interests ever target Enceladus directly? Probably not for resource extraction. The distances are too vast and the energy requirements too high for any conceivable economic return. But privately funded science missions aren't impossible. Several wealthy individuals have expressed interest in astrobiology. If someone wanted to fund an Enceladus life detection mission as a legacy project, the technical capability exists. It hasn't happened yet, but the landscape is shifting in ways that make it less unthinkable than it was even 10 years ago. Public perception plays into this as well. Missions require sustained political support, which means maintaining public interest across administrations and budget cycles. Enceladus has an advantage here because the concept is simple to communicate. There's an ocean under the ice. We can sample it. We might find life. That narrative resonates in ways that more abstract science goals sometimes don't. It's concrete, visual, and tied to a fundamental question people actually care about. The media coverage of Cassini's discoveries demonstrated this. Every major plume finding made headlines. The hydrothermal vent evidence was covered by mainstream news outlets, not just science publications. That visibility matters when Congress or parliaments are deciding which missions to fund. A mission to Enceladus isn't just scientifically justified, it's a story people want to follow. Human summary. And what happens if we actually find something? Not just organic molecules or chemical signatures, but clear, unambiguous evidence of biological activity. Something living, or something that was alive recently enough that we can study its biochemistry. The protocols for that scenario exist but have never been tested in practice. NASA established planetary protection guidelines decades ago, rules designed to prevent contamination in both directions. We don't want to accidentally bring Earth microbes to another world and mistake them for indigenous life. We also don't want to bring back something from another world without proper containment, though that's less of a concern for a mission that won't return samples directly to Earth. But the scientific protocols are just the beginning. The confirmation process would be rigorous to the point of paranoia. One instrument reading wouldn't be enough. One type of measurement wouldn't be enough. The standard would require multiple independent lines of evidence, preferably from different analytical techniques, all pointing toward the same conclusion. And even then, the debate would be intense. When Viking landers conducted experiments on Mars in the 70s, one test produced results that could have indicated microbial life. Decades of argument followed. Most scientists now think it was a chemical reaction, not biology, but consensus took years to establish. With Enceladus, the scrutiny would be even more severe because the implications are so profound. Teams would need to rule out every conceivable non-biological explanation before claiming a detection. That takes time, careful analysis, and probably follow-up observations if possible. The spacecraft might need to sample the plume multiple times, looking for consistency. Are the same signatures present in different plume particles? Do they show up in the same ratios? Are there seasonal variations or do the readings remain stable? Repeatability matters enormously in science, especially when making extraordinary claims. One intriguing reading is a puzzle. 3 identical readings from separate samples start looking like a pattern. 10 readings, all consistent, all showing the same biological markers, that becomes very difficult to explain through non-biological chemistry alone. Then there's the question of announcing it. How do you tell the world that we found life beyond Earth? The process would likely involve peer review first. Scientists would prepare papers documenting every measurement, every analysis, every alternative explanation they considered and rejected. Those papers would be submitted to journals like Science or Nature, where other experts would examine the methodology in detail. Only after passing that review would there be an official announcement. That's the ideal sequence anyway. In reality, information leaks. Mission teams involve hundreds of people. Data gets shared among collaborators. Someone always talks to someone else who talks to someone else. The discovery might become an open secret in the scientific community weeks or months before any public statement. Managing that information flow, preventing premature claims while maintaining scientific integrity, would be a challenge unlike anything space agencies have faced before. Because once the news breaks, the response would be immediate and global. Media coverage would be overwhelming. Every news organization on the planet would want the story. Scientists involved in the mission would face endless interview requests. Social media would explode with speculation, excitement, skepticism, and probably a fair amount of conspiracy theorizing. Religious institutions would need to grapple with theological implications. Philosophers would debate what it means for humanity's place in the universe. Artists and writers would produce countless works inspired by the discovery. It would reshape conversations across every domain of human culture. Politically, the response would probably be unifying in the short term. Finding life elsewhere is the kind of news that transcends national boundaries and ideological divisions. It's bigger than any single country or political system. In that moment, humanity would have a shared experience, a collective realization that we're not alone, that life is a cosmic phenomenon rather than an Earth-specific accident. That could produce a brief period of global solidarity, a sense of common purpose and shared wonder. How long that lasts is another question. Humans are skilled at turning even unifying moments into sources of division. But initially, the reaction would likely be one of excitement and curiosity rather than fear or conflict. The practical questions would follow quickly though. What do we do next? One detection from one moon wouldn't tell us everything we need to know about life in the universe. It would tell us that life can exist in an ocean world beneath an ice shell. It would tell us something about the chemistry that supports it, the energy sources it uses, the environmental conditions it tolerates. But it wouldn't tell us how common life is, whether it arose independently on Enceladus or somehow shares an origin with Earth life, whether it's simple bacteria-like organisms or something more complex, whether it's currently active or represents an extinct biosphere we're detecting through molecular fossils. Each answer would generate 10 new questions. The logical next step would be more missions. If we find life on Enceladus, Europa immediately becomes a higher priority target. Does it also harbor life? Is it the same kind of life or something different? What about the other ocean worlds, Ganymede, Callisto, Titan? Do they show similar biosignatures, or is Enceladus unique? Sample return missions would move from theoretical to essential. We'd need to bring material back to Earth where it could be analyzed with laboratory equipment far more sophisticated than anything we can fit on a spacecraft. That introduces the contamination concerns again, requiring quarantine facilities and handling protocols, but the scientific payoff would be enormous. We'd want to sequence genomes if the life is DNA-based, or understand whatever information storage system it uses if it's not. We'd want to study its metabolism in detail, grow it in culture if possible, understand its evolutionary history. All of that requires samples and equipment that won't fit through space anytime soon. Then there's the long-term question of protecting these environments. If Enceladus has life, do we have an obligation to leave it undisturbed? Right now, the moon is pristine, or at least as pristine as something can be after we've flown a spacecraft through its plumes several times. But intensive study would mean more missions, more instruments, possibly even landers on the surface or submarines in the ocean eventually. Each mission carries some risk of contamination, some chance of introducing Earth organisms that could disrupt or destroy native ecosystems. Space agencies take this seriously. Spacecraft bound for potentially habitable worlds undergo intensive sterilization. Components are heated to kill any microbes. Surfaces are cleaned with harsh chemicals. Assembly happens in clean rooms with filtered air and strict protocols. Nothing is perfect though. Some microbes survive extreme conditions. Spores can endure heat, cold, radiation, and vacuum. The chance of introducing something isn't zero, it's just very low. But very low multiplied by multiple missions over decades starts to add up. International agreements would probably be necessary. Rules about how many missions are allowed, what kinds of instruments can be used, what areas are off-limits to protect pristine environments. These agreements exist in principle through the Outer Space Treaty and Planetary Protection Guidelines, but they'd need to be strengthened and made more specific if we're dealing with confirmed biospheres rather than potential ones. And enforcement becomes tricky. Who decides what's acceptable? Who has the authority to prevent a nation or organization from launching a mission if they disagree with the restrictions? Space law is still developing in these areas. We're writing the rules as we go, usually staying one step behind the technology. The other long-term consideration is what finding life on Enceladus would mean for the search elsewhere. Does it make life seem more common or less? If two places in our solar system have life, that suggests biology emerges readily when conditions are right. Water, chemistry, and energy seem to be enough. That would be encouraging for the search around other stars. Every planetary system probably has multiple ocean worlds. If ocean worlds reliably develop life, then life might be everywhere. But there's a counterargument. What if Earth life and Enceladus life share a common origin? Panspermia, the idea that life can spread between worlds through meteorite impacts, isn't impossible. Mars and Earth have exchanged rocks throughout history. Material from Earth has reached the outer solar system, though rarely. If Enceladus life turns out to be related to Earth life genetically, that would be fascinating but also disappointing in a way. It would mean we've only found one origin of life, not two. We still wouldn't know if life is common or rare. We just know it can survive in different environments once it gets started. Distinguishing between these possibilities would require detailed genetic or biochemical analysis. If Enceladus life uses the same DNA structure, the same amino acids, the same basic cellular machinery as Earth life, common origin becomes likely. If it uses different building blocks, different coding systems, different fundamental chemistry, then it almost certainly arose independently. That would be the more profound discovery, two separate origins of life in the same solar system. At that point, the universe would statistically be teeming with life. We'd just be early in the process of finding it. The discovery wouldn't just reshape our understanding of life's abundance. It would force us to confront uncomfortable questions about intelligence, consciousness, and our own significance. If simple life exists in multiple places within one solar system, what about complex life? What about intelligence? The gap between microbes and minds is enormous, but if biology gets started easily, maybe that gap gets crossed more often than we think. Or maybe not. Maybe microbial life is common but complex multicellular organisms are vanishingly rare. Birth took billions of years to move from single cells to animals. Most of that time was spent as a microbial world. Intelligence came even later, in just the last few million years, a blink in geological time. There's no guarantee that evolution trends toward complexity. Natural selection doesn't have goals. It doesn't push life up a ladder toward intelligence. It just favors whatever works in the moment. If simple works, simple persists. Enceladus might have had life for billions of years without ever developing anything more sophisticated than bacteria. That would still be remarkable, but it would also suggest intelligence is a fluke, a rare accident that happened once on Earth and maybe nowhere else nearby. Then again, we don't really know what intelligence requires. We assume it needs complexity, nervous systems, brains. But what if there are other forms of cognition we wouldn't recognize? What if microbial communities develop collective behaviors that look simple individually but show emergent properties we'd classify as intelligent if we knew what to look for? We've seen hints of this on Earth. Bacterial colonies communicate chemically, coordinate responses, make collective decisions about growth and movement. Slime molds, not even animals, can solve maze puzzles and optimize network structures. Intelligence might not require neurons. It might just require information processing, feedback loops, and environmental interaction. Enceladus life could surprise us in ways we haven't imagined yet. But even if it's just microbes, the philosophical implications cut deep. For all of human history, we've wondered if we're alone. Every religion, every culture has grappled with our place in the cosmos. Finding life elsewhere, even simple life, even in our own solar system, changes that conversation permanently. We're not unique. We're not central. We're one example among potentially countless others. Some people find that thought liberating. It removes the burden of cosmic specialness, the pressure to justify our existence through uniqueness. We're just part of a larger pattern, one thread in a vast biological tapestry. Others find it unsettling. If we're not special, what's the point? If life is common, does individual life matter less? These are emotional reactions, not logical ones, but they're real and they'll shape how society processes the discovery. The scientific community would adapt quickly. Researchers would pivot to new questions, new investigations, new grant proposals. But the general public moves slower. Cultural narratives take time to shift. Education systems would need to incorporate this new reality. Textbooks would be rewritten. Documentaries would be produced. Children would grow up in a world where extraterrestrial life isn't science fiction, it's established fact. That generation would see the universe differently than any before them. They'd assume life is normal, expected, something to be studied and understood rather than wondered about in the abstract. Their questions would be different. Not whether life exists elsewhere, but what forms it takes, how it works, whether we can communicate with it. The long-term trajectory of human civilization might shift. If we know life is out there, space exploration becomes more urgent. Not just for scientific curiosity, but for connection, for understanding, for the possibility of finding something beyond microbes. Investment in telescopes, spacecraft, and astrobiology would increase. Private companies would get involved, not just governments. The economics of space would change if there's something genuinely valuable to find, not just rocks and radiation. And valuable doesn't necessarily mean resources. Knowledge is valuable. Contact is valuable. Understanding our place in a living universe is valuable in ways that don't show up on a balance sheet. But practical concerns would emerge too. If we're visiting Enceladus regularly, someone will eventually propose mining it. The moon has water, organic compounds, energy. Those are useful. Future spacecraft could refuel there. Habitats could be built. Humans might want to establish a presence. That raises a collision between exploration and exploitation. Do we have the right to industrialize a world with native life? Even if that life is microbial? Even if it's confined to subsurface oceans we're not directly disturbing? There's no clear answer. Earth's history offers cautionary tales. Every time humans encountered new ecosystems, we disrupted them. Sometimes deliberately, sometimes accidentally, but always irreversibly. Islands, forests, oceans, we've left marks everywhere. Doing the same thing on Enceladus would be consistent with our track record, but knowing we're doing it makes the choice more conscious, more morally weighted. Some would argue life anywhere is precious and must be protected absolutely. Enceladus should be declared a nature preserve, off limits to anything beyond minimal scientific study. Others would argue humans have needs too, and resources in space should be available for our use, especially if that use enables further exploration and the spread of Earth life to new environments. Finding balance between these positions would require international cooperation on a scale we've rarely achieved. The Antarctic Treaty System provides a model. Antarctica is protected for scientific research, with resource extraction banned. Military activity is prohibited. Multiple nations cooperate to maintain the agreement despite political differences elsewhere. Something similar could work for Enceladus, but enforcement is harder when you're dealing with space. There's no border patrol in the outer solar system. Monitoring compliance would require trust, transparency, and shared oversight. Violations would be difficult to detect and even harder to punish. Still, the attempt would be worth making. Establishing norms now, before economic interests become entrenched, is easier than trying to impose rules later. The discovery of life on Enceladus would give us that narrow window, that brief moment when everyone agrees protection matters before anyone has invested billions in infrastructure. That window wouldn't stay open long. Within a decade, maybe two, commercial interests would start pushing boundaries. Technology would advance. Costs would drop. The moon would go from distant curiosity to accessible destination. Once that transition happens, preservation becomes much harder. So the discovery itself would trigger a race, not just to study Enceladus, but to define its future. Scientists racing to gather data before environments are disturbed. Ethicists racing to establish guidelines before they're needed. Policymakers racing to create enforceable frameworks before violations occur. And entrepreneurs racing to figure out what can be extracted, sold, or utilized before regulations lock things down. All of this would unfold in parallel with continued searching elsewhere. Because if Enceladus has life, the next question is immediate. What about Europa? What about Titan? What about the moons of Uranus and Neptune we've barely studied? What about ocean worlds around other stars? The search would expand rapidly, driven by the knowledge that we're not looking for something hypothetical anymore. We're looking for variations on a theme we've already confirmed. That's a different kind of search, more concrete, more urgent. Funding would flow toward missions that can answer these questions. Public support would be strong. Media coverage would be intense. The field of astrobiology would transform from speculative science to mainstream research with clear goals and measurable progress. Universities would expand programs. Students would flood into the discipline. The best minds of a generation would focus on understanding life beyond Earth, how it starts, how it evolves, what forms it can take. Within a few decades, we might have a comparative biology that spans multiple worlds. We'd understand what features are universal and what varies. We'd know if DNA is inevitable or just one option among many. We'd see if evolution follows similar patterns everywhere or takes wildly different paths depending on environment. That knowledge would feed back into our understanding of Earth life. We'd see ourselves more clearly by seeing what we're not. The assumptions we make because we only have one example would become visible. We'd recognize which aspects of biology are fundamental and which are accidents of our particular history. And somewhere in that process, the question would surface again, sharper than before. If simple life is common, what about complex life? What about intelligence? What about civilizations? Are we alone in that category, or is the universe filled with minds looking outward just like we are, wondering the same things? the question shifts from whether we're alone to whether we're typical. And that distinction matters more than it seems at first. Being alone means something fundamentally different than being rare. Being rare but not unique means the path we took, from chemistry to cells to consciousness, can happen elsewhere given enough time and the right conditions. That would make intelligence not a miracle but a statistical outcome, improbable for any single world but inevitable across enough worlds. The discovery of microbes on Enceladus wouldn't answer that question directly. Simple life doesn't predict complex life. Earth had bacteria for billions of years before anything more complicated emerged. The jump from single cells to multicellular organisms required specific conditions, particular evolutionary pressures, maybe a dose of luck. Nothing about finding bacteria on Enceladus would tell us if that jump is easy or nearly impossible. But it would narrow the possibilities. Right now, we can't rule out the idea that life itself is the bottleneck, that getting from chemistry to biology is so difficult it only happened once in the observable universe. Finding life on Enceladus would eliminate that possibility. The bottleneck would have to be somewhere else, either in the transition to complexity or in the emergence of intelligence or in the development of technology. Each of those could be common or vanishingly rare. We don't know. We can't know from a single data point. But we'd know more than we do now. We'd know the first step isn't impossibly hard. And that would shift how we think about the rest of the ladder. If microbial life turns out to be common, scattered across ocean worlds and buried in subsurface environments throughout the galaxy, then the question becomes duration. How long does life need to develop complexity? Earth took about 3.5 billion years to go from bacteria to animals. Is that typical? Could it happen faster under different conditions, or does it require that much time regardless of environment? We don't know because we've only watched it happen once, on a planet with a stable sun, a large moon, plate tectonics, and a magnetic field. Change any of those variables and the timeline might compress or stretch or terminate entirely. Complexity might require not just life but stable life, ecosystems that persist long enough for evolutionary experiments to compound. On Enceladus, environmental stability is questionable. The moon's interior ocean might have existed for billions of years, or it might cycle between frozen and liquid states depending on orbital dynamics and tidal heating. If conditions fluctuate too much, life might restart repeatedly, never accumulating the layers of complexity that lead to multicellular organisms. That would make Enceladus a place where life exists but doesn't progress, trapped in a loop of survival without advancement. Earth avoided that trap through a combination of factors we're still trying to fully understand. Our planet stayed in the habitable zone. Our atmosphere thickened at the right rate. Our magnetic field shielded the surface from sterilizing radiation. None of those things were guaranteed. All of them were necessary. Other worlds might have one or two of those factors, but not all. And without all of them, complexity might be impossible no matter how long life has to try. So the discovery of Enceladus life would open up a second wave of questions about what conditions allow life to move beyond survival. We'd start looking not just for life, but for environments stable enough to support its evolution. That's a harder search because stability is difficult to measure from a distance. You can detect water. You can detect chemistry. But assessing whether a world has been stable for billions of years requires indirect inference, modeling, long-term observation. Still, the effort would be worth it. Because if we find complex life, even just multicellular organisms, the implications escalate dramatically. Complex life means evolution found a path forward. It means the transition is possible, not just theoretically but practically, in environments that actually exist. And if it happened twice, once on Earth and once elsewhere, the probability shifts again. Two independent origins of complexity would suggest it's not a fluke. It would suggest that given life and time, complexity emerges reliably. That wouldn't guarantee intelligence. Evolution doesn't aim for intelligence. It optimizes for survival and reproduction. Intelligence is one strategy among many, useful in certain contexts, irrelevant or even counterproductive in others. Most complex life on Earth never developed anything resembling human cognition. Trees, fungi, jellyfish, insects, all complex, none intelligent in the way we use the term. So even if complex life is common, intelligence might not be. The evolutionary pressures that favor big brains, social cooperation, language, and abstract thinking might be rare. They might require specific ecological niches, particular kinds of predation pressure, or competition dynamics that don't exist on most worlds. Earth had those conditions in certain regions at certain times, and intelligence emerged multiple times independently. Dolphins, elephants, corvids, octopuses, primates, all developed advanced cognition through different evolutionary paths. That suggests intelligence isn't a one-time accident. But it also shows it's not universal. Most lineages don't go that direction. Most complex life remains non-intelligent. If intelligence is rare even among complex life, then finding it elsewhere becomes exponentially more difficult. We need to identify not just worlds with life, not just worlds with complex life, but worlds where evolution happened to push organisms toward cognition. And then we'd need to catch them during the narrow window when they're technologically active. Because intelligence without technology is invisible from interstellar distances. We can't detect thought. We can't sense consciousness. We can only look for the side effects of technology, radio signals, atmospheric pollution, megastructures, things that leak evidence into space. A planet full of intelligent but pre-technological beings would look lifeless to us. We'd scan it, see biosignatures maybe, but miss the minds entirely. That asymmetry matters. It means the search for intelligence is really a search for technological civilizations, not intelligence itself. And technology adds another filter, another step in the chain that might be common or might be nearly impossible. Earth's path to technology depended on fire, which requires oxygen-rich atmosphere and dry land. It depended on manipulative appendages, hands or tentacles capable of fine motor control. It depended on social structures that allowed knowledge to accumulate across generations. Remove any of those and technology might never develop no matter how intelligent the species becomes. Dolphins are smart, arguably as smart as early humans, but they can't build tools underwater. Octopuses are brilliant problem solvers, but they're solitary and short-lived, so their knowledge dies with each individual. Intelligence alone isn't enough. The environment has to permit its expression in ways that lead to technology. And even if all those conditions align, there's still the question of whether technological civilizations survive long enough to be detected. Humans have been radio-capable for roughly a century. In cosmic terms, that's nothing, a flicker. If most civilizations last only a few centuries before collapsing or transcending or going silent, then the galaxy could be full of intelligence that we'll never notice because we're not looking at the right moment. The timing would have to overlap, our search coinciding with their broadcast, our receiver pointed at their transmitter during the brief window when both exist. That's a narrow target. It's possible we're surrounded by civilizations that existed 1 million years ago or will exist 1 million years from now, and we'll never know because we're scanning the wrong era. The Fermi paradox captures this tension. If life is common, if intelligence emerges reliably, if technology develops naturally, then where is everyone? Why haven't we seen evidence? The silence could mean we're alone. Or it could mean intelligence is common but short-lived. Or it could mean we're looking in the wrong places, listening for the wrong signals, missing evidence that's right in front of us because we don't recognize what we're seeing. Finding life on Enceladus wouldn't resolve the paradox, but it would shift the probabilities. It would confirm the first step is achievable. It would give us a second data point, a comparison that lets us start building models. And models, even rough ones, are better than pure speculation. They let us make predictions, design experiments, focus resources. Right now, the search for extraterrestrial intelligence operates in a fog of uncertainty. We don't know what we're looking for, where to look, or how common the target is. Every assumption is provisional. Every strategy is a guess. But if we find life nearby, if we confirm biology can start independently in multiple places within our own solar system, that fog lifts slightly. We'd know the universe is biologically active. We'd know the ingredients for life aren't unique to Earth. We'd know that wherever conditions permit, chemistry tries to become biology. And once we know that, the question isn't whether life exists elsewhere. The question becomes how far it gets. Right now, Enceladus is still just a candidate. We have chemistry, energy, water, everything life needs assembled in one place. But chemistry isn't biology. Potential isn't proof. The next decade will determine whether this tiny frozen moon becomes the most important place humanity has ever studied or just another interesting dead world with the right ingredients but no spark. The missions are being designed. Engineers are solving problems. Budgets are being negotiated. Within 15 years, maybe 20, we'll have spacecraft specifically built to answer this question. They'll fly through those plumes with instruments sensitive enough to detect molecular patterns that chemistry alone can't explain. They'll measure isotope ratios, search for homochirality, look for the subtle fingerprints that biology leaves on its environment. And when those results come back, transmitted across 1 billion kilometers of empty space, we'll be standing at one of two doorways. Either we'll have confirmation that life exists beyond Earth, that the universe is biologically active, that we're not alone. Or we'll have even deeper questions about why a world with every necessary ingredient remains sterile, what invisible barrier prevents chemistry from crossing into biology, what Earth had that Enceladus lacks. Both answers change everything. Finding life means rewriting our place in the cosmos, understanding that consciousness emerged not through cosmic accident but through processes that operate wherever conditions allow. Not finding life despite perfect conditions means the origin of life is harder than we thought, that some crucial factor we haven't identified makes the difference between chemistry and biology. The strange thing is we're living through the moment when this stops being philosophy and becomes data. For all of human history, the question of life beyond Earth was abstract, something to wonder about but never answer. Now we're maybe 2 decades from knowing. That's nothing. That's immediate. Anyone reading this will likely be alive when we get the answer, at least for Enceladus. What happens after depends entirely on what we find. Discovery of life triggers one future, a rapid expansion of astrobiology, missions to every ocean world we can reach, a fundamental recalculation of how common biology is. Not finding life triggers a different future, deeper investigation into what makes Earth special, more careful study of how life begins, perhaps lowered expectations about finding it elsewhere. Either way, we're approaching a boundary. The search for life has been theoretical for centuries, based on speculation and probability arguments. Soon it becomes empirical, based on measurements and data from actual environments where life might exist. That transition from wondering to knowing is rare in science. It usually happens quietly, through accumulated evidence rather than single dramatic discoveries. This might be different. If those instruments detect biosignatures in Enceladus's plumes, if multiple independent measurements converge on the same conclusion, if peer review confirms what the initial data suggests, then the announcement won't be quiet. It'll be the biggest news in human history, the moment we confirm we're not the only biology in the universe. And if the measurements come back negative, if the plumes contain only chemistry, if every test shows an ocean that's rich in potential but empty of life, that's also news. Profound news. It means we're rarer than we thought, that the universe doesn't automatically fill every habitable niche with biology, that Earth represents something unusual. We don't know which future we're heading toward. The data doesn't exist yet. But the fact that we're about to get that data, that we're living in the generation that will finally have an answer instead of just better questions, that matters. It changes how we think about exploration, about our priorities, about what we're actually searching for when we build spacecraft and point them at distant worlds. Enceladus is small. It's cold. It's unimaginably far from anything humans have ever touched. But right now, as you're reading this, it's spraying its ocean into space, offering samples of its hidden depths to anyone capable of reaching them. That opportunity won't wait forever. Eventually, mission windows close, funding priorities shift, political will fades. The moment to answer this question is now, not someday, not when it's more convenient. Because if there's life in that ocean, every day we wait is another day we remain ignorant of the most important discovery our species could make. And if there's not, every day we wait is another day we avoid learning something equally important about how rare and precious life actually is. Either answer justifies the effort, the cost, the decades of work required to get there and find out. The universe isn't going to hand us easy answers. It never has. But occasionally it offers shortcuts, places where the barriers between us and knowledge are thinner than usual. Enceladus is one of those places. A world that's literally giving us samples, that's making the search for life easier than it has any right to be, that's accessible with current technology if we choose to go. That choice is being made right now in budget meetings and conference rooms, in engineering labs and planning sessions. The decision about whether to prioritize Enceladus, whether to fund the missions that could actually answer this question, that's happening in the present tense. And once those decisions are made, once the spacecraft are built and launched, once they're committed to 7-year journeys across the solar system, we're locked in. We'll get our answer. Whatever it is. Human history will split at that point into before and after. Before we knew if life existed beyond Earth. After we confirmed it or ruled it out, at least in the most accessible place we found. That boundary is approaching faster than most people realize. We're not talking about science fiction futures or distant possibilities. We're talking about missions that could launch in the next decade, that could return data before 2040, that could answer humanity's oldest question within the lifetime of everyone currently alive. The clock is running. Enceladus is still there, still spraying its secrets into space, still waiting for us to show up with the right instruments and ask the right questions. Whether we do that, whether we commit the resources and attention required to actually find out what's in that ocean, that's on us. The universe has done its part. It's put the evidence right where we can reach it. What we do with that opportunity will define this era of human civilization more than almost anything else we're capable of achieving. Because this isn't about one moon or one mission or one scientific question. It's about whether we're alone. And that answer, whatever it turns out to be, changes everything that comes after. A telescope floating 1 million miles from Earth just detected something that shouldn't exist. 2 chemicals in the atmosphere of a distant planet, chemicals that on Earth are produced exclusively by living organisms. This isn't speculation or wishful thinking. It's data from the most powerful observatory humanity has ever built, pointing at a world 124 light-years away where something might be alive right now. The planet is called K2-18b. You've never heard of it, but scientists have been watching it obsessively since the James Webb Space Telescope turned its instruments toward this particular corner of the constellation Leo. What they found there has divided the scientific community into camps of cautious optimism and aggressive skepticism, because the implications are staggering. We might be looking at the first real evidence of life beyond Earth. Here's what makes this so compelling. The chemicals detected, dimethyl sulfide and dimethyl disulfide, don't just sometimes get produced by life on Earth. They're made almost exclusively by microscopic marine organisms like phytoplankton and algae. These aren't gases that form easily through random chemistry. They require specific biological processes, the kind that happen when living cells metabolize sulfur compounds in ocean water. Finding them in another planet's atmosphere is like walking into an empty house and smelling fresh coffee. Something made that. But let's pump the brakes for a second, because this story is way more complicated than the headlines suggest. K2-18b sits in what astronomers call the habitable zone of its star, a red dwarf much smaller and cooler than our Sun. The planet orbits this star every 33 days, receiving roughly the same amount of light Earth gets from the Sun. It's massive compared to our world, about 8.6 times Earth's mass and 2.6 times larger in diameter. That makes it what scientists call a sub-Neptune, somewhere between Earth and the ice giants in our own solar system. The distance? 124 light-years. That means the light the Webb telescope is analyzing left that planet during the late 1800s on Earth. We're looking at old light, ancient photons that traveled through space for more than a century before hitting Webb's mirrors and revealing their secrets. What makes K2-18b particularly interesting is that it might be what's called a Hycean world. The term was coined in 2021, a mashup of hydrogen and ocean, describing planets that weren't supposed to be habitable but might actually be perfect for life. These are worlds covered in deep oceans, potentially hundreds or even 1,000 kilometers deep in some places, with thick hydrogen-rich atmospheres above them. Before the concept of Hycean planets, scientists assumed hydrogen atmospheres would make worlds too hot for life. K2-18b is forcing a rethink of that assumption. The detection itself happened through a method called transit spectroscopy. When K2-18b passes in front of its star from our perspective, starlight filters through the planet's atmosphere before reaching us. Different molecules absorb specific wavelengths of light, creating a kind of chemical fingerprint. Webb's instruments are so sensitive they can detect these tiny variations in brightness and identify what gases are present millions of miles away. In 2023, the telescope detected methane and carbon dioxide in K2-18b's atmosphere. That was already significant, the first time such carbon-based molecules were spotted on an exoplanet in a habitable zone. But there was something else in the data, a weak signal that looked like it might be dimethyl sulfide. So the research team, led by scientists from the University of Cambridge, pointed Webb back at the planet using a different instrument, the mid-infrared instrument, designed to detect different wavelengths of light. What they found was stronger evidence for both dimethyl sulfide and dimethyl disulfide. The concentrations are wild compared to Earth. On our planet, these chemicals exist at levels below 1 part per billion in the atmosphere. On K2-18b, the estimated concentrations are over 10 parts per million. That's thousands of times stronger. If these readings are accurate, something is pumping these gases into that atmosphere at an extraordinary rate. On Earth, the process works like this. Marine phytoplankton, particularly dinoflagellates and coccolithophores, produce a compound called DMSP as part of their cellular processes. When these organisms die or get eaten, or when viruses attack them, DMSP breaks down into dimethyl sulfide. It's a complex chain involving bacteria, enzymes, and photochemical reactions. The DMS then escapes into the atmosphere where it plays a role in cloud formation and the planet's sulfur cycle. It's a signature of biological activity in ocean ecosystems. Finding it on another world should mean something similar is happening there. Should. That's where the controversy explodes. The statistical confidence level of this detection sits at 3 sigma. In scientific terms, that means there's a 0.3% chance the signal is just random noise, a fluke in the data. Sounds pretty good, right? Except the gold standard for scientific discovery is 5 sigma, which represents a probability of less than 0.00006% that the result is random. The difference between 3 sigma and 5 sigma is the difference between interesting and definitive. The research team knows this. They've been careful not to claim they've discovered life. What they're saying is that the data they have is consistent with the presence of these biosignature molecules, and that more observations are needed. They estimate another 16 to 24 hours of observation time with Webb could push them to that 5 sigma threshold. But here's where it gets messy. Other scientists have analyzed the same data and reached different conclusions. Some argue that the DMS signal isn't actually there at all, that it overlaps too much with methane signals and the analysis methods aren't robust enough to separate them. The data, they say, is consistent with a flat line, no bump or wiggle indicating a real detection. It's a classic case of seeing what you want to see in noisy data. Then there's the magma planet hypothesis. Some researchers think K2-18b isn't a water world at all, but a hellscape with oceans of molten rock instead of liquid water. Raymond Peary Humbert, a planetary physicist at Oxford, has run models suggesting the planet is too hot for life, that any water present would be superheated and the surface would be volcanic. If that's true, the entire Hycean world concept for K2-18b collapses. There's also the non-biological production problem. DMS has been detected on comets and in interstellar space, proving it can form without life through purely chemical processes we don't fully understand. Just because we only know biological sources on Earth doesn't mean that's the only way it forms in the universe. The cosmos is creative with chemistry in ways we're still discovering. The debate reveals something fundamental about where we are in the search for extraterrestrial life. The James Webb Space Telescope has given us capabilities we've never had before. In just a few years of operation, we've learned more about exoplanet atmospheres than in the previous 3 decades combined. But we're operating at the absolute edge of what's possible with current technology. We're trying to detect faint signals from small planets incredibly far away, and interpretation of that data requires making assumptions about alien environments we can't directly observe. This is the paradox. We finally have tools powerful enough to potentially detect life on other worlds, but we might not have tools precise enough to prove it definitively. Webb might identify the most promising candidates, the worlds worth watching more closely, but confirming life could require the next generation of telescopes that haven't been built yet. What we do know is this: K2-18b is real. It's sitting in its star's habitable zone. It has an atmosphere with carbon-based molecules. The chemistry is complex and active. Whether that chemistry includes life remains an open question, one that's driving some of the most heated scientific debates happening right now. The lead researcher, Niku Midhusudhan from Cambridge, has said that the only scenario currently explaining all the Webb data, past and present, is one where K2-18b is a Hycean world teeming with life. But he's also emphasized the need for deep skepticism, for testing and retesting, because that's how science works. No one wants to claim prematurely that we found alien life. What kind of life are we talking about if it exists? Not little green men or intelligent civilizations. The conditions on K2-18b, assuming it's habitable at all, would support microbial life at best. Think of Earth's oceans billions of years ago, filled with single-celled organisms producing gases as metabolic waste. That's the kind of biology we might be detecting, primitive but profound. The hydrogen-rich atmosphere adds another layer of complexity. Laboratory experiments show that Earth microorganisms can survive in hydrogen atmospheres, so hydrogen itself isn't a deal-breaker for life. But it changes which biosignature gases are reliable indicators. Some gases that scream life on Earth might form through non-biological processes in hydrogen-rich environments, making interpretation even trickier. The temperature is estimated between 250 and 300 Kelvin in the absence of greenhouse effects. That's cold, but not impossibly so. With the right atmospheric composition and pressure, liquid water could exist. The deeper layers of the atmosphere might be too hot, crushing, but there could be a zone, a sweet spot where conditions align. Ocean depths on Hycean worlds are staggering to contemplate. Depending on surface gravity and temperature, these oceans could extend tens to hundreds of kilometers deep, reaching pressures of 60,000 bar before the water transitions to high-pressure ice. That's an alien environment by any measure, but Earth's deep oceans host life in equally extreme conditions. Hydrothermal vents, crushing pressure, total darkness, yet life thrives. If K2-18b is producing dimethyl sulfide biologically, the implications extend far beyond one planet. Hycean worlds are thought to be common around red dwarf stars, the most numerous stars in the galaxy. Their larger sizes compared to rocky planets make them easier to observe and analyze. If these worlds can host life, the number of potentially habitable planets in the universe explodes. But we're getting ahead of ourselves. The data is preliminary, contested, requiring confirmation. The scientific process is working exactly as it should, with competing teams analyzing the same information and reaching different conclusions. That tension, that back and forth, is how we eventually arrive at truth. What happens next matters enormously. The full dataset became publicly available in late April, meaning more astronomers can now scrutinize every detail. Additional observation time with Webb has been requested. Other instruments on other telescopes might be able to contribute complementary data. The investigation is just beginning. Standing at this moment in history feels significant regardless of what we ultimately discover about K2-18b. We're asking questions about alien life not through speculation but through direct observation of another world's chemistry. We're analyzing gases in an atmosphere 124 light-years away and debating whether they come from living organisms. That alone represents a milestone. The search for life beyond Earth has entered a fundamentally new phase, one where we're no longer limited to searching for radio signals or planning distant missions to Mars. We're using infrared telescopes to sniff the air of alien worlds, looking for the chemical exhaust of biology. What we're learning is that the universe is far more complex than our models predicted, that every new observation raises new questions, and that the answer to whether we're alone might be closer than we ever imagined. But first, we need to understand what we're actually looking at. The challenge lies in how we actually detect these molecules from such an enormous distance. Light becomes the messenger. When K2-18b passes between its star and Webb's instruments, starlight filters through the planet's atmosphere. Different molecules absorb specific wavelengths, creating a unique spectral fingerprint. Each molecule has its own signature pattern, a barcode written in missing wavelengths. Webb's mid-infrared instrument was specifically designed to read these barcodes. The telescope doesn't see K2-18b directly. What it captures is the star's light after it's been altered by passing through alien air. The brightness drops slightly during transit, and that drop varies by wavelength depending on what's in the atmosphere. Methane absorbs certain infrared wavelengths. Carbon dioxide takes others. And dimethyl sulfide, if it's there, should create its own distinctive absorption pattern. The problem is that these signals are incredibly faint. We're talking about detecting changes in brightness of less than 1%, and then within that tiny signal, finding even smaller variations that indicate specific molecules. It's like trying to identify a single instrument in an orchestra from a mile away while standing next to a jet engine. The initial detection of dimethyl sulfide came from analyzing how starlight was absorbed at specific infrared wavelengths between about 3 and 5 microns. The researchers saw what they interpreted as the spectral signature of DMS, a pattern that shouldn't be there unless something is producing this molecule. But here's where it gets complicated. Dimethyl disulfide has overlapping spectral features in the same wavelength range. Both molecules belong to the same chemical family. Both are predicted biosignatures. The current data can't definitively distinguish between them. Think of it like hearing a muffled voice through a wall. You know someone's speaking, but you can't quite make out whether they said yes or yeah. both fit what you're hearing. Additional observations might sharpen the signal enough to tell them apart, but right now, we're stuck with one or the other, possibly both. The concentrations detected make the biological interpretation even more intriguing. On Earth, dimethyl sulfide exists in the atmosphere at concentrations below 1 part per billion by volume. It's produced by marine phytoplankton, released into the air, and quickly broken down by chemical reactions. The levels are low because production is balanced by destruction. On K2-18b, the estimated concentration is over 10 parts per million, thousands of times higher than Earth's levels. That's a massive difference requiring explanation. If biology is producing DMS on K2-18b, it must be doing so at extraordinary rates, or the molecule must be more stable in that environment, or both. A hydrogen-rich atmosphere might slow down the chemical reactions that normally destroy DMS on Earth. Alternatively, if there's a vast ocean teeming with DMS-producing organisms and the atmospheric chemistry is different, concentrations could build up to levels we never see in our own atmosphere. But the skeptics have compelling counterarguments. The DMS signal isn't strong. It appears in some data analysis methods but not others. When you're working at the absolute limits of instrumental sensitivity, noise becomes a serious concern. Random fluctuations can create patterns that look like signals. Statistical tools help separate real detections from false positives, but they're not perfect, especially when the signal is weak. The 3 sigma confidence level means there's a 0.3% chance the signal is just noise, a random fluctuation that happens to look like dimethyl sulfide. That might sound pretty good, but science has a higher bar. 5 sigma confidence, the gold standard for claiming a discovery, means less than a 0.00006% chance of being wrong. We're not there yet. Not even close. Then there's the methane problem. Computer models accounting for the physics and chemistry of both DMS and methane in a hydrogen-based atmosphere suggest the signal might actually be methane, not dimethyl sulfide at all. Methane was already detected on K2-18b in previous observations. If methane absorption overlaps with where we expect to see DMS, we might be mistaking one for the other. It's like trying to hear a whisper next to someone shouting. The loud signal can mask or mimic the quiet one. Raymond Peary-Humbert from Oxford University published research suggesting K2-18b might not be a water world at all. His models indicate the planet could be too hot for liquid water oceans, that what we're seeing might be better explained by a magma planet with oceans of molten rock instead of water. If that's true, the entire Hiccean interpretation collapses. No water ocean means no marine phytoplankton-like organisms producing DMS. The whole biological hypothesis falls apart. A magma planet sounds outlandish, but it's not impossible. The temperature estimates for K2-18b range from about 250 to 300 Kelvin without greenhouse effects, but greenhouse warming from a thick hydrogen atmosphere could push surface temperatures much higher. If the deeper atmosphere is hot enough to melt rock, we're not looking at a potentially habitable world but a hellscape wrapped in hydrogen. The recent reanalysis added more fuel to the skeptical fire. Independent researchers examined the same web data and found that instead of a clear signal, a bump or wiggle in the spectrum, the data looks consistent with a flat line. No dimethyl sulfide. No dimethyl disulfide. Just noise and uncertainty. They argue that more observations are absolutely necessary before making any claims about biosignature gases. This is where science gets messy and beautiful at the same time. Multiple teams analyzing identical data reach different conclusions. It's not that anyone's being dishonest or incompetent. It's that interpretation at the edge of detectability involves judgment calls about data processing, statistical methods, and modeling assumptions. Different choices lead to different results. Some researchers emphasize the signal they see and argue it's worth taking seriously even if it needs confirmation. Others emphasize the uncertainty and argue the evidence doesn't meet the threshold for a credible detection. Both perspectives are valuable. The optimists push the field forward by exploring possibilities. The skeptics keep everyone honest by demanding rigorous proof. The Discovery team, led by researchers including Miku Madhusudhan, maintains that the only scenario currently explaining all the Webb data, including past and present observations, is a Hycean world potentially hosting life. They're not claiming definitive proof. They're saying this interpretation fits the evidence better than alternatives. But they also emphasize the need for deep skepticism of their own results. Science advances through testing and retesting, not through accepting the first interesting result that comes along. What's undeniable is that we're operating at the absolute limit of current technology. Webb was designed to do exactly this kind of observation, but even Webb has constraints. The signals from small planet atmospheres are weak. The number of suitable targets is limited. The inference methods, the mathematical tools we use to extract meaning from noisy data, have inherent limitations. We're pushing against fundamental barriers of what's possible with this generation of instruments. So what comes next determines everything. The full dataset became publicly available in late April, opening the door for more astronomers to scrutinize every detail. Fresh eyes might spot something previous analyses missed. Alternative interpretations will be proposed and tested. The scientific community will collectively work through the data, and consensus will eventually emerge, though it might take months or years. Additional observation time with Webb is crucial. The team estimates that 16 to 24 hours of follow-up observations could push the statistical significance from 3 sigma to 5 sigma, crossing the threshold from intriguing possibility to credible detection. That's not a guarantee, though. More data might strengthen the signal, or it might make the whole thing disappear, revealing it was noise all along. Other instruments might contribute complementary information. Ground-based telescopes, future space missions, different observational techniques could all add pieces to the puzzle. The detection of biosignature gases isn't a one-shot deal. It's a gradual accumulation of evidence from multiple independent sources until the weight of data becomes overwhelming. But there's a deeper question lurking beneath all the technical debate. Even if we confirm dimethyl sulfide is present, even if we reach 5 sigma confidence, even if alternative explanations are ruled out, we still face the interpretation problem. Does the presence of DMS necessarily mean life? On Earth, yes. Dimethyl sulfide is produced by biological processes, mainly marine phytoplankton breaking down a precursor molecule called dimethylsulfoniopropanate. The biochemical pathways are complex, involving enzymes, bacteria, viruses, and photochemical reactions. It's deeply integrated into Earth's sulfur cycle and plays a role in cloud formation by contributing to sulfate aerosol production. The connection to life is ironclad here. But K2-18b isn't Earth. It's a world with a hydrogen atmosphere, potentially crushing pressures, temperatures, and chemistry completely unlike anything in our experience. Could there be non-biological pathways producing DMS under those exotic conditions? We genuinely don't know. DMS has been detected on comets and in the interstellar medium, proving it can form without biology in certain environments. Those detections don't involve planetary atmospheres or the concentrations we're discussing, but they demonstrate that the molecule itself isn't exclusively biological. Unknown chemistry in unknown conditions could potentially generate biosignature gases through non-living processes. This is the challenge facing astrobiology. There's no silver bullet, no single molecule that unambiguously screams life regardless of context. Everything depends on context. The molecule matters less than the environment producing it. What we need is a comprehensive understanding of what K2-18b actually is, and right now, we're operating with incomplete information. Let's step back and examine what we actually know about this world. K2-18b orbits an M-dwarf star, a red dwarf, about 124 light-years away in the constellation Leo. The star itself is smaller and cooler than our Sun, with a surface temperature around 3,457 Kelvin compared to the Sun's 5,778 Kelvin. Its radius is only 45% of the Sun's. These details matter because they shape the radiation environment, the energy budget, and the habitability prospects of any planet in the system. K2-18b completes one orbit every 33 days, placing it squarely within what astronomers call the habitable zone, the region where temperatures theoretically allow liquid water to exist on a planet's surface. It receives roughly the same amount of stellar radiation that Earth receives from the Sun. That sounds promising until you remember that habitable zone is a crude first-order approximation. It doesn't account for atmospheric composition, greenhouse effects, albedo, or a dozen other factors that determine whether a planet is genuinely habitable or a hellscape dressed in optimistic terminology. The planet is massive. 8.6 times Earth's mass. 2.6 times Earth's radius. Those numbers push it into the category of sub-Neptunes, worlds larger than Earth but smaller than Neptune. In our solar system, there's nothing quite like it. We have rocky terrestrial planets and gas giants, but nothing in between. Sub-Neptunes are common throughout the galaxy, yet we have no local example to study. That's a problem because it means we're working from theory and distant observations rather than direct knowledge. Here's where things get interesting. K2-18b's size and mass suggest it shouldn't be a rocky planet like Earth. It's too big. Traditional models predicted that planets of this size would have thick hydrogen atmospheres, crushing pressures, and no solid surface to speak of. Mini Neptunes, basically. Not places where life as we understand it could take hold. But the data from Webb complicated that picture considerably. The detection of methane and carbon dioxide in 2023 was the first hint that K2-18b might be something different. These carbon-based molecules in a habitable zone planet suggested chemistry compatible with life. Then came the potential DMS detection, and suddenly we're looking at a planet that doesn't fit neatly into existing categories. This led researchers to propose that K2-18b might be a Hycean world. The term Hycean was coined in 2021 by Niku Madhusudhan and his team at Cambridge. It's a portmanteau of hydrogen and ocean. The concept describes planets with vast liquid water oceans beneath thick hydrogen-rich atmospheres. Before this idea emerged, astronomers assumed such planets would be too hot to harbor life. The hydrogen atmosphere would trap heat through a runaway greenhouse effect, turning any water into superheated steam. But detailed modeling suggested otherwise. Under certain conditions, a hydrogen atmosphere could actually be cool enough to allow liquid water oceans at the surface. Think about what that means. A planet with an ocean covering its entire surface, potentially hundreds or even thousands of kilometers deep depending on temperature and gravity. Pressures at the ocean floor could reach 60,000 bar before transitioning to high-pressure ice phases. Above this ocean, a thick blanket of hydrogen gas, completely unlike Earth's nitrogen-oxygen atmosphere. Temperatures at the surface might reach 200 degrees Celsius or higher, yet the ocean persists because the pressure is so immense. This is not an environment remotely similar to Earth. If life exists there, it's operating under physical and chemical rules we can barely imagine. The hydrogen atmosphere alone changes everything. On Earth, certain biosignature gases are considered reliable indicators of life because they react quickly and shouldn't persist without biological replenishment. But in a hydrogen-rich atmosphere, those same gases might be produced through non-biological chemistry or might be stable enough to accumulate without life. The rulebook we've been using doesn't apply. Laboratory experiments have shown that Earth microorganisms can survive in hydrogen-rich atmospheres. Hydrogen itself isn't toxic to life. But that's different from saying life could originate and thrive in such conditions. We're extrapolating from our limited understanding of terrestrial biochemistry into a regime where we have no data. The ocean depths on a Hycean world depend critically on surface temperature and gravity. Models suggest they could span anywhere from tens of kilometers to potentially 1,000 kilometers before the pressure becomes so extreme that water transitions into exotic ice phases that exist only under crushing force. These ice layers would be hot, not cold, because temperature increases with depth. The conventional image of ice doesn't apply here. Within such an ocean, where would life exist? Near the surface, where temperatures might be survivable but radiation from the star could be intense. Deeper down, where pressure is enormous but conditions might be more stable. Around hydrothermal vents on the ocean floor, if such things exist. We genuinely don't know. The possibilities are wide open, constrained only by physics and chemistry, not by any examples we can point to. But there's a competing interpretation that throws all of this into question. Some researchers, including Raymond Peary-Humbert at Oxford, argue that K2-18b isn't a water world at all. Their models suggest it's too hot for liquid water to exist at the surface. Instead, they propose it might be a magma planet, a world where the surface is molten rock, not ocean. If that's correct, the entire Hycean hypothesis collapses, and K2-18b becomes a completely different kind of object, one with no habitability potential whatsoever. The debate hinges on details. Atmospheric composition, cloud properties, albedo, internal heat flow. Small differences in these parameters lead to radically different conclusions. One model produces a temperate water world. Another produces an inferno. Both are consistent with the limited data we have, which tells you how uncertain the situation really is. What about the concentration of DMS itself? On Earth, dimethyl sulfide exists in the atmosphere at levels below 1 part per billion by volume. Trace amounts. On K2-18b, the estimated concentration is over 10 parts per million. Thousands of times stronger. That's a massive difference. If the detection is real, it demands an explanation. Biological processes on Earth don't produce DMS at those concentrations in the atmosphere. So either K2-18b has vastly more biological activity than Earth, or the chemistry producing DMS there operates through entirely different mechanisms. Perhaps life on a Hycean world, if it exists, is incredibly productive. An ocean teeming with microorganisms, all releasing DMS as a metabolic byproduct, could potentially generate those high concentrations. Or maybe the atmospheric dynamics are different. On Earth, DMS gets oxidized quickly. On K2-18b, maybe it persists longer, accumulating to higher levels. We're speculating now, stacking assumptions on top of incomplete data. The alternative is that we're misinterpreting the signal entirely. Recent reanalyses of the web data suggest the DMS feature might not be there at all. Instead of a clear spectral bump indicating the molecule's presence, the data might be consistent with a flat line, just noise. The strength of the claimed detection depends entirely on how you process and interpret the raw data. Different analysis techniques produce different results. That's not ideal when you're trying to make the most important discovery in human history. Both DMS and DMDS are from the same chemical family, and their spectral features overlap in the wavelength range Webb observed. The current data can't definitively tell them apart. Future observations might resolve that ambiguity, or they might reveal that neither molecule is actually present. We're waiting on more data to settle questions that can't be answered with what we have now. The scientific process is working exactly as it should. Initial detection, public announcement, independent verification attempts, debate, more observations, gradual convergence toward truth. It's messy. It's slow. It involves false starts and dead ends. But it's the only method we have for reliably understanding reality. The alternative is wishful thinking dressed up as certainty. What happens if follow-up observations confirm DMS is really there? We're still left with the interpretation problem. Does its presence mean life, or does it mean we discovered an exotic chemical pathway that operates under conditions we've never studied? The problem runs deeper than just identifying molecules. It's about understanding what they mean in an environment nothing like Earth. When we detect DMS in our atmosphere, we know where it comes from because we've studied the organisms that produce it. We've mapped the pathways, measured the rates, understood the seasonal variations. We have context. On K2-18b, we have none of that. We're looking at a hydrogen-rich atmosphere wrapped around a planet that might be ocean, might be magma, circling a star completely different from our sun. Every assumption we make is built on Earth-based chemistry, and there's no guarantee those rules apply. Consider how DMS behaves here. Marine phytoplankton, specifically dinoflagellates and coccolithophores, synthesize it through complex biochemical pathways involving enzymes, viral interactions, and photochemical processes. Once released into the atmosphere, it oxidizes quickly, gets broken down, contributes to cloud formation. The whole cycle is tightly regulated by temperature, light availability, nutrient concentration. Change any of those parameters, and the entire system responds differently. Now imagine trying to predict what happens on a world where the atmospheric pressure is different, the radiation environment is different, the ocean chemistry is different. You can't just transplant Earth biology there and expect it to work the same way. That's why the concentration issue is so puzzling. Over 10 parts per million compared to less than 1 part per billion on Earth. If biology is producing it, the ecosystem would need to be extraordinarily productive, generating DMS at rates we've never observed. Or the atmospheric chemistry could be preventing its breakdown, allowing it to accumulate. Both scenarios require mechanisms we don't fully understand. You're trying to reverse engineer an alien biosphere from a single data point. It's like finding a footprint and trying to reconstruct the entire animal. Then there's the non-biological possibility. DMS has been detected on comets and in interstellar space, proving it can form without life. The processes aren't well understood, but they exist. Maybe K2-18b has some exotic chemistry driven by radiation, temperature gradients, or mineral interactions that produces DMS abiotically. We can't rule that out. The challenge is that we don't know all the ways molecules can form under extreme conditions. Chemistry textbooks are written based on laboratory experiments and Earth observations. We're constantly discovering that nature is more creative than our models predict. Statistical significance matters here more than in most fields. The current detection sits at 3 sigma. In particle physics, you need 5 sigma before claiming a discovery. That's because the stakes are enormous and the risk of false positives is real. Free sigma means there's a 0.3% chance the signal is just noise, random fluctuations masquerading as a real feature. Sounds pretty good until you consider how many observations astronomers make, how many wavelengths they examine, how many molecules they search for. With enough attempts, even unlikely coincidences become probable. The researchers know this. They're calling for more observation time, specifically 16 to 24 hours with Webb, to push the detection past 5 sigma. That would reduce the false positive probability to essentially 0. But getting that telescope time isn't trivial. Webb is the most in-demand observatory in history. Every astronomer wants a piece of it. Time is allocated through competitive proposals, peer review, priority ranking. K2-18b is important, but so are hundreds of other targets. Distant galaxies, forming stars, other exoplanets. The telescope can only point in one direction at a time. Patience is required, which is frustrating when you're sitting on potentially the biggest discovery ever made. While we wait, other teams are analyzing the same data independently. That's how science progresses. Multiple groups, different analysis techniques, various assumptions. If everyone reaches the same conclusion, confidence grows. If results diverge, that tells you the signal is ambiguous and more data is essential. Recent reanalyses have been less optimistic than the original announcement. Some researchers see a flat line where others see a spectral feature. The difference comes down to data processing choices. How you remove noise, how you model the baseline, which wavelength ranges you emphasize. None of these choices are obviously right or wrong. They're judgment calls, and different judgments lead to different conclusions. This is normal in cutting-edge science, especially when working at the limits of instrumental capability. Webb is astonishingly sensitive, but it's not magic. Detecting molecules in the atmosphere of a planet 124 light-years away requires extracting incredibly faint signals from noisy data. Think about what's actually happening. Light from a distant star passes through a thin layer of gas surrounding a planet. That gas absorbs specific wavelengths corresponding to molecular vibrations. Webb measures the spectrum, looking for those absorption features. But the planet's atmosphere is tiny compared to the star. The signal you're trying to detect is a fractional change in brightness at specific wavelengths. Any instrumental artifact, any calibration error, any unaccounted variable can produce false features. The overlap between DMS and DMDS complicates things further. Both molecules are sulfur-containing organics with similar structures. Their spectral signatures overlap in the wavelength range Webb observed. You can't definitively say which one you're seeing, or if it's a combination, or if it's something else entirely producing a similar-looking feature. Future observations at different wavelengths might resolve the ambiguity, but right now, the data is consistent with multiple interpretations. What about the hydrogen-rich atmosphere itself? On Earth, biosignature gases are identified based on their behavior in our nitrogen-oxygen atmosphere. Certain molecules, like oxygen or methane in large quantities, are considered strong biosignatures because they shouldn't coexist without biology constantly replenishing them. But in a hydrogen-dominated atmosphere, those rules change. Some gases that would be biosignatures on Earth become unreliable indicators elsewhere. The chemistry is just different. Hydrogen reacts with things nitrogen doesn't. It changes oxidation states, reaction rates, equilibrium concentrations. You need entirely new models to predict what a biosignature would even look like in that environment. Microorganisms from Earth can survive in hydrogen-rich conditions. Lab experiments have demonstrated that. So hydrogen itself isn't toxic to life. But would life that evolved in a hydrogen atmosphere produce the same waste gases as Earth life? Maybe. Maybe not. Evolution is contingent. Life adapts to its environment using whatever chemistry is available. On Earth, photosynthesis uses water and produces oxygen. On a Hycean world, maybe life uses different molecules, produces different byproducts. We're looking for Earth-like biosignatures because those are the ones we know how to recognize. That doesn't mean they're the only ones that exist. The bigger question is whether we're even asking the right questions. For nearly a century, astronomers have focused on atmospheric disequilibrium as a sign of life. The idea is that certain gas mixtures shouldn't exist together unless something is actively maintaining them. Oxygen and methane, for example. They react with each other, so if you detect both in significant quantities, something must be replenishing them faster than they can react away. Life is one explanation. But it's not the only one. Geological processes, photochemistry, exotic atmospheric dynamics could also create disequilibrium. The challenge is distinguishing between biological and non-biological sources from light years away. K2-18b represents a test case for this entire approach. If the DMS detection holds up, if follow-up observations confirm it, we're forced to interpret what it means. Is it life? Is it chemistry we don't understand? Is it an instrumental artifact we haven't identified? Each answer has profound implications. If it's life, everything changes. If it's not, we've learned something important about planetary atmospheres and the limits of remote sensing. Either way, the process of figuring it out advances our understanding. The lead researchers have been careful with their language. They're not claiming definitive proof of life. They're presenting data, proposing interpretations, calling for verification. That's exactly how it should work. The temptation to overhype is enormous. This is the kind of discovery that makes careers, wins awards, gets remembered in textbooks. But claiming life prematurely, only to be proven wrong later, damages credibility and sets the field back. Better to be cautious, methodical, rigorous. Let the data accumulate. Let the debate unfold. Let the truth emerge through the messy, slow process of scientific consensus building. What happens if 5 sigma is reached and DMS is confirmed beyond doubt? We're still left with interpretation. The molecule is there, but what's producing it? That question can't be answered with spectroscopy alone. You'd need detailed atmospheric models, laboratory experiments simulating Hycean conditions, maybe even direct detection of other biosignatures to build a coherent case. One molecule isn't enough. You need a pattern, a suite of indicators that together point toward biology as the most likely explanation. That's the standard. Anything less leaves too much room for doubt. And that's where the timeline becomes critical. The researchers estimate they need between 16 and 24 additional hours of observation time with Webb. That might not sound like much, but telescope time is precious. Webb isn't dedicated to a single target. Hundreds of research teams compete for every available hour. Scheduling those observations, collecting the data, processing it, that takes months. Maybe longer. During that waiting period, the scientific community doesn't sit idle. The original dataset became publicly available in April, which means independent researchers can now access the same raw data. They'll run their own analyses, apply different statistical methods, test alternative interpretations. Some will support the findings. Others will challenge them. That's the messy reality of frontier science. Consensus doesn't emerge from a single paper, but from a community-wide process of scrutiny and verification. Here's what makes this particularly complicated. The signal isn't clean. It's buried in noise, overlapping with methane spectral features, sitting right at the edge of what current instruments can reliably detect. Different teams using different data reduction techniques might reach different conclusions about whether the bump in the spectrum is real or an artifact. That's already happening. Recent reanalyses of the same data suggest the signal might be consistent with a flat line, no feature at all. The original team stands by their results, pointing to specific wavelengths and modeling approaches that support the detection. Who's right? More data will tell. But even if the 5 sigma threshold is eventually reached, even if DMS is confirmed with statistical certainty, we're not done. Confirmation means the molecule is there. It doesn't automatically mean life put it there. You'd still need to rule out non-biological explanations. And here's where it gets tricky. DMS has been detected in comets. It's been found in the interstellar medium, the diffuse gas and dust between stars. That proves it can form through purely chemical processes, no biology required. So how do you distinguish between biological and non-biological sources from 124 light-years away? Context matters. On Earth, DMS concentrations in the atmosphere are typically below 1 part per billion. On K2-18b, the estimated concentration is over 10 parts per million, thousands of times higher. That difference is significant. Non-biological processes tend to produce trace amounts, not massive concentrations. But that's an assumption based on Earth chemistry. Maybe Hycean world chemistry operates differently. Maybe there's some exotic photochemical pathway we haven't considered that churns out DMS at high concentrations without biology. You can't rule it out just because we haven't thought of it yet. This is why the search for biosignatures requires multiple lines of evidence. One molecule, however compelling, isn't enough. You need a pattern. A suite of gases that together make biological production the most parsimonious explanation. The researchers detected methane and carbon dioxide in earlier observations. Those are consistent with biological activity, but they're also produced by geological processes. Add DMS to the mix and the biological hypothesis becomes stronger. But it's still not definitive. You'd want to see other sulfur compounds, maybe organic molecules, signatures of metabolic waste products that don't have obvious non-biological sources. The challenge is that we're working with incredibly limited information. Transmission spectroscopy gives you atmospheric composition, nothing more. You can't see the surface. You can't measure ocean salinity or pH. You can't detect cellular structures or DNA. All you have is the chemical fingerprint of gases backlit by starlight. From that, you're trying to infer the presence of life. It's like trying to diagnose a patient's illness by analyzing their breath, except the patient is 124 light-years away and you only get to sample their breath once every few years. What would make the case stronger? Detection of gases that have no known non-biological source. On Earth, certain compounds are produced exclusively by life. Methyl chloride, for example, is made by marine algae and has no significant geological source. If you detected that on K2-18b, alongside DMS and other biosignatures, the biological hypothesis would become much harder to dismiss. But even then, you'd need to demonstrate that no unknown chemistry could produce those gases. That's a high bar. Some researchers argue we're approaching this wrong. Instead of looking for individual molecules, we should be looking for chemical disequilibrium patterns that only life can sustain. The idea is that life constantly pushes chemistry away from equilibrium, maintaining gas mixtures that would otherwise react away. On Earth, oxygen and methane coexist because life produces both faster than they can react with each other. Without life, that disequilibrium would collapse within a few million years. If you can demonstrate that K2-18b's atmosphere is in persistent disequilibrium, and that no geological or photochemical process can maintain it, that's stronger evidence than any single molecule. The problem is proving the negative. How do you demonstrate that no unknown process exists? You can't. You can only show that all known processes fail to explain the data. That's weaker than it sounds because planetary science is full of surprises. Venus has phosphine in its atmosphere, and nobody's entirely sure why. It could be life, or it could be some weird atmospheric chemistry we don't understand yet. The same uncertainty applies here. There's also the question of what kind of life we're talking about. If K2-18b hosts biology, it's almost certainly microbial. Single-celled organisms living in the ocean, metabolizing whatever chemistry is available. Not fish, not plants, nothing complex. The conditions wouldn't support it. High atmospheric pressure, crushing ocean depths, temperatures that might reach 200 degrees Celsius in deeper layers. Surface waters might be cooler, more temperate, but you're still looking at an alien environment fundamentally different from Earth. Life adapted to that environment wouldn't look like Earth life. It would be something else entirely. Which raises an interesting philosophical point. When we search for biosignatures, we're inherently biased toward Earth-like life because that's the only reference we have. We know what Earth life produces, what gases it releases, what chemical disequilibria it creates. But life elsewhere might operate on completely different principles. Silicon-based instead of carbon-based. Using ammonia instead of water. Metabolizing in ways we haven't imagined. If that's the case, we might be looking right at biosignatures without recognizing them because they don't match our preconceptions. This is the central tension in astrobiology. You want to cast a wide net, consider all possibilities, but you also need specific, testable predictions. You can't search for everything simultaneously. You have to prioritize, make educated guesses about what's most likely. That inevitably introduces bias. The challenge is being aware of that bias and not letting it blind you to unexpected findings. K2-18b might not be the planet where we finally confirm extraterrestrial life. The signal might not hold up. The interpretation might shift as more data comes in. But it's pushing the field forward regardless. It's forcing researchers to refine their models, develop better detection methods, think harder about what constitutes convincing evidence. Each attempt, whether successful or not, teaches us something about planetary atmospheres, about the limits of our instruments, about the complexity of distinguishing biology from chemistry. The next generation of telescopes will help. Instruments currently being designed will have higher sensitivity, better spectral resolution, the ability to study smaller, rockier planets. They'll be able to detect fainter signals, resolve overlapping spectral features, measure atmospheric composition with greater precision. That will narrow the uncertainty, make the interpretations less ambiguous. But it won't eliminate the fundamental challenge. We're still doing remote sensing across vast distances, inferring complex phenomena from limited data. What happens if we do eventually confirm life on K2-18b or another exoplanet? The implications extend far beyond astronomy. It would reshape our understanding of biology, showing that life isn't unique to Earth but emerges wherever conditions allow. It would raise questions about how common life is, whether intelligence is inevitable, what our place is in a universe teeming with biology. Those aren't just scientific questions. They're philosophical, even existential. But we're not there yet. Right now, we're in the uncertain middle ground between detection and confirmation. The data is suggestive but not conclusive. The interpretations are plausible but not proven. The path forward requires patience, rigor, and a willingness to accept that we might not get definitive answers as quickly as we'd like. The scientific process doesn't move in straight lines. It advances through incremental steps, occasional breakthroughs, and frequent setbacks. Each observation adds a data point, constrains the possibilities, eliminates hypotheses that don't fit. That's how knowledge builds. Not through sudden revelations, but through accumulated evidence slowly shifting the balance of probability. K2-18b represents one data point in that larger process. A significant one, certainly. The detection of potential biosignature gases on a habitable zone planet using our most advanced telescope is noteworthy regardless of how the interpretation ultimately resolves. But it's still just one planet among thousands we've discovered, millions that likely exist in our galaxy alone. Consider the broader landscape. Webb has only been operational since 2022. In that short time, it's already transformed exoplanet science, providing atmospheric data that was impossible to obtain before. WASP-39b, a hot Jupiter, revealed a complete chemical inventory including water vapor, carbon dioxide, carbon monoxide, sodium, potassium, and sulfur dioxide. That last one was particularly interesting because it requires photochemistry, chemical reactions driven by starlight in the upper atmosphere. It showed that planets far different from Earth still have complex atmospheric processes. TOI 561b, an ultra-hot super-Earth, challenged assumptions about small planets close to their stars. Conventional wisdom said they shouldn't retain atmospheres. The stellar radiation should strip away any gases. Yet Webb detected what appears to be a thick atmosphere, possibly above a global magma ocean. The planet's unusually low density suddenly made sense. It's not just rock. It has a substantial gaseous envelope despite the harsh environment. These discoveries matter because they expand our understanding of what's possible. Each unexpected finding forces researchers to reconsider their models, question their assumptions, think more creatively about planetary formation and evolution. That's the value of exploration. Not just confirming what you already believe, but discovering what you didn't anticipate. Which brings us back to the central question. How do we know when we've actually found life? The answer isn't simple because life itself isn't simple. On Earth, biology produces countless chemical signatures. Oxygen, methane, phosphine, nitrous oxide, dimethyl sulfide, hundreds of volatile organic compounds. But almost all of these can also be produced through non-biological processes under the right conditions. Oxygen comes from photosynthesis, but also from water vapor breaking apart in the upper atmosphere. Methane comes from microbes, but also from volcanic activity and geochemical reactions. Phosphine might come from life, but it's also produced in gas giants through atmospheric chemistry. The challenge is distinguishing biological production from geological or atmospheric production when you can't visit the planet to investigate directly. You're limited to remote observations, spectral analysis, statistical inference. That means looking for combinations of gases that are unlikely to coexist without biology. On Earth, oxygen and methane together create a disequilibrium signature. They react with each other chemically, so their simultaneous presence suggests continuous replenishment from different sources. Biology maintains that disequilibrium. Remove life, and the gases would equilibrate, disappearing over geological timescales. But what if alien biochemistry operates differently? What if life elsewhere uses different metabolic pathways, produces different waste products, creates different atmospheric signatures? We wouldn't necessarily recognize it because we're calibrated to Earth's patterns. This is the biosignature detection paradox. You need specific predictions to guide your search, but those predictions inevitably narrow your perspective. You might miss what you're not looking for. Some researchers argue for a more agnostic approach. Instead of searching for specific molecules, look for any chemical disequilibrium that can't be explained by known planetary processes. If you detect an atmosphere that's thermodynamically unstable, something must be maintaining it. That something could be biology. The problem with this approach is that planetary processes are complex and not fully understood. What looks like unexplained disequilibrium might just be physics we haven't modeled correctly. You risk false positives if you're not careful. The flip side is false negatives. If you're too conservative, demanding ironclad proof before accepting a biosignature, you might dismiss genuine detections because they don't fit your preconceptions. Science requires balancing these risks. Being open to unexpected findings while maintaining rigorous standards of evidence. It's not easy. The history of science is littered with premature announcements, overinterpreted data, claims that didn't hold up to scrutiny. That's why the K2-18b team is proceeding cautiously despite the suggestive signals. They've reached 3 sigma statistical significance, meaning there's a 0.3% chance the signal is random noise. That sounds compelling, but in scientific terms, it's not enough for confirmation. The standard for discovery claims is 5 sigma, which corresponds to a probability of less than 0.00006% that the signal is coincidental. Getting from 3 sigma to 5 sigma requires more observation time, better data, additional wavelength coverage. The team estimates they need 16 to 24 more hours of telescope time to reach that threshold. Whether they'll get it is uncertain. Telescope time is precious. Webb is oversubscribed by a factor of 10, meaning far more proposals than available observation slots. Every hour spent on K2-18b is an hour not spent on other targets. Committees evaluate proposals, prioritize based on scientific merit, feasibility, likely impact. K2-18b is compelling, but so are dozens of other candidates. The allocation process is competitive. Assuming they do get the time and the signal strengthens to 5 sigma, then what? Even definitive detection of dimethyl sulfide doesn't automatically mean life. It just means biology becomes the most likely explanation. You'd still need to rule out alternative scenarios. Could photochemistry in a hydrogen-rich atmosphere produce DMS through pathways we haven't considered? Could geological processes release sulfur compounds that combine in the atmosphere to form DMS? Are there reactions involving minerals, water, and atmospheric gases that might generate these molecules without biology? These questions need answers before claiming discovery. The scientific community would scrutinize every aspect. The data reduction methods, the spectral analysis techniques, the atmospheric models, the statistical treatments. Other teams would attempt independent analyses using the same data. They'd look for errors, biases, alternative interpretations. This process is essential. Extraordinary claims require extraordinary evidence, and the claim of extraterrestrial life is about as extraordinary as it gets. The evidence needs to withstand intense scrutiny from multiple independent groups before acceptance. Even if the biosignature interpretation holds up, you're still left with profound uncertainties. You know life probably exists on that planet, but you don't know what kind. You don't know how it originated, how it evolved, whether it's widespread or rare. You don't know if it's simple or complex, ancient or recent, thriving or barely surviving. All you have is a chemical signature in an atmosphere 124 light-years away. The rest is inference and speculation. That might seem frustrating, but it's actually remarkable. A century ago, we didn't know if other planets existed beyond our solar system. 50 years ago, we had no way to study their atmospheres. 20 years ago, we couldn't detect biosignature gases even if they were present. Now we're at the threshold of potentially confirming life elsewhere. The pace of progress has been extraordinary. The capabilities we've developed in just a few decades would have seemed like science fiction to earlier generations. Future telescopes will push further. The Habitable Worlds Observatory, currently in early planning stages, is designed specifically to characterize Earth-like planets around Sun-like stars. It will have a coronagraph to block starlight, allowing direct imaging of planets in reflected light rather than relying on transit spectroscopy. That opens up new possibilities. You could study planets that don't happen to pass in front of their stars from our perspective. You could analyze surface features, detect seasonal variations, maybe even observe geological activity. Ground-based extremely large telescopes coming online in the next decade will complement space-based observations. The 30-meter telescope, the Giant Magellan Telescope, and the Extremely Large Telescope will have collecting areas far larger than Webb, allowing them to gather more photons and achieve higher spectral resolution. They'll be limited by Earth's atmosphere, but adaptive optic systems are becoming sophisticated enough to compensate. Combined with space telescopes, they'll provide multiple independent measurements of planetary atmospheres, reducing uncertainties and strengthening conclusions. The technology is advancing rapidly. So is our theoretical understanding. Researchers are developing more sophisticated atmospheric models, accounting for complex chemistry, cloud formation, atmospheric circulation patterns. They're studying extreme environments on Earth, places where life persists under conditions previously thought uninhabitable. Deep ocean vents, acidic hot springs, Antarctic ice, high-altitude deserts. Each discovery of terrestrial extremophiles expands the envelope of possibility for where life might exist elsewhere. This is the context in which K2-18b should be understood. Not as a definitive answer, but as a milestone in an ongoing journey. We're learning how to ask the right questions, developing the tools to answer them, building the knowledge base to interpret what we find. Each planet we study teaches us something new about atmospheric processes, planetary formation, the conditions necessary for habitability. That knowledge accumulates, refining our search strategies and improving our detection capabilities. The question of whether we're alone in the universe won't be answered by a single observation. It will be answered through accumulated evidence from multiple planets, multiple biosignatures, multiple independent confirmation. K2-18b might be part of that answer. Or it might turn out to be a false alarm, teaching us about atmospheric chemistry instead of biology. Either way, it's moving the field forward. That's what matters. Not whether this particular planet hosts life, but whether we're getting better at finding out. And we are. The tools exist now. The methods are proven. The candidates are identified. What happens next depends on how we use them. The timeline stretches ahead with uncomfortable uncertainty. Those additional observation hours with Webb might happen next year. Or the year after. scheduling depends on committee decisions, competing priorities, telescope availability. Meanwhile, K2-18b keeps orbiting its red dwarf star, oblivious to our scrutiny, its atmosphere continuing whatever processes are producing those spectral features we're arguing about. What's clear is that we've crossed a threshold. Not necessarily the threshold of discovering life, but the threshold of being able to look. Webb represents a capability that didn't exist 5 years ago. We can now analyze the atmospheric composition of planets in habitable zones around other stars. That's not incremental progress. That's a fundamental shift in what's possible. Think about where we were just a generation ago. The first exoplanet around a Sun-like star was confirmed in 1995. Before that, we literally didn't know if planets existed beyond our solar system. We suspected they did, but suspicion isn't knowledge. Now we've cataloged thousands. We know that planets are common, that rocky worlds exist throughout the galaxy, that many orbit within their stars' habitable zones. The question has shifted from, are there other worlds? To, what are those worlds like? K2-18b forces us to confront how we answer that second question. The data is ambiguous. The interpretations diverge. But the fact that we're having this debate at all, arguing over spectral features in the atmosphere of a planet 124 light-years away, that's the real story. We're not just theorizing anymore. We're measuring, analyzing, testing hypotheses against actual observations. Whether dimethyl sulfide is really there, whether it's produced by life or exotic chemistry, those questions will get resolved through the normal scientific process. More data will arrive. Better models will be developed. Consensus will eventually form, though it might take years. That's fine. Science doesn't run on news cycle timelines. It runs on evidence accumulation, and evidence takes time. The broader trajectory is what matters. Each planet studied adds to our understanding of atmospheric processes, of what's normal and what's anomalous, of how to distinguish biology from geology from instrumental artifacts. We're building a reference library of planetary atmospheres, learning to read chemical signatures across an enormous range of conditions. That knowledge base will prove invaluable when the next candidate emerges, and the one after that. Because K2-18b won't be the last world where we detect potential biosignatures. Webb is just getting started. There are thousands of exoplanets within its observational range. Some will be too hot, too cold, too massive, too small. But some will sit in that narrow band of conditions where life as we understand it might exist. Those are the targets. Those are where humanity's attention will focus over the coming decades. The search for life beyond Earth isn't a single moment of discovery. It's a process of gradually narrowing uncertainty, eliminating alternatives, building confidence through accumulated evidence. Maybe K2-18b is the first confirmed detection. Maybe it's a stepping stone toward understanding Hycean worlds that don't actually host life. Maybe it's teaching us that our biosignature detection methods need refinement before we can trust them. All of those outcomes advance the field. What we're witnessing is the maturation of a scientific discipline. Astrobiology is transitioning from speculation to observation, from hypothetical arguments to data-driven conclusions. The tools exist now to test ideas that were purely theoretical a decade ago. That's transformative. It changes the conversation from, could there be life? To, here's what we measured, here's what it might mean, here's how we find out for sure. The philosophical implications lurk beneath the technical debates. If life exists on K2-18b, even simple microbial life, it would demonstrate that biology isn't unique to Earth. It would show that the chemical and physical conditions necessary for life arise naturally when planets form in habitable zones. That has profound consequences for how common life might be throughout the galaxy, throughout the universe. If it happened twice in one small corner of space, it's probably happened billions of times elsewhere. But those implications only matter if the detection is real. And that's still uncertain. The data is suggestive. The interpretation is plausible. The statistical confidence is approaching significance. But we're not there yet. The gap between intriguing possibility and confirmed discovery remains, and crossing that gap requires rigor that can't be rushed. So we wait. For more telescope time. For additional observations. For independent verification. For the slow grinding process of science to either confirm or refute what the initial data suggested. That waiting is hard, especially when you're potentially sitting on the biggest discovery in human history. But it's necessary. Getting it right matters more than getting it first. Meanwhile, the search continues. Other planets. Other biosignatures. Other research teams developing new detection methods, new analytical techniques, new ways of interpreting atmospheric chemistry. The field is advancing on multiple fronts simultaneously. K2-18b is one thread in a much larger tapestry. Whether this particular thread leads to confirmation of life or not, the tapestry keeps growing, the picture keeps coming into focus. What happens over the next few years will determine whether K2-18b enters the history books as the first world where we found evidence of extraterrestrial life, or as a cautionary tale about the challenges of biosignature detection. Either way, it's already changed how we think about the search. It's demonstrated that we have the capability to detect potential biosignatures. It's revealed the complexity of interpretation. It's shown us both the promise and the pitfalls of this endeavor. The universe doesn't care about our timelines or our eagerness for answers. It will reveal its secrets at its own pace, through careful observation and rigorous analysis. Our job is to keep looking, keep measuring, keep questioning our interpretations until the weight of evidence becomes overwhelming. That's how we'll eventually answer the question of whether we're alone. Not through a single dramatic discovery, but through accumulated knowledge that makes the answer undeniable. K2-18b might be that answer. Or it might be a false start. The only way to know is to keep looking at the data, keep refining the models, keep pushing the instruments to their limits. Because somewhere out there, on some planet around some distant star, life probably exists. Finding it, proving it, understanding it, that's the challenge defining this era of astronomy. And we're closer now than we've ever been. "96% of all marine life vanished in less than 100,000 years. Earth became a graveyard. This wasn't the asteroid that killed the dinosaurs. This happened 200 million years earlier, and it was worse. Much worse. Welcome to the Permian-Triassic extinction event, the moment life on Earth nearly ended forever. Around 252 million years ago, something catastrophic unfolded. The planet didn't just experience a bad day or a rough century. It endured geological violence on a scale that makes every other extinction look modest by comparison. When paleontologists first began piecing together what happened at the boundary between the Permian and Triassic periods, they uncovered something deeply unsettling. The fossil record showed a world teeming with complexity and diversity, then suddenly, almost nothing. Scientists call it the Great Dying, and that name barely captures the horror. Imagine walking through a forest where every tree stands dead and silent. Picture oceans where fish float lifeless at the surface as far as the eye can see. Envision continents where the very soil turns toxic and rivers run thick with decay. That was Earth during this extinction. Life didn't just take a hit. It got obliterated from nearly every ecosystem simultaneously. The numbers tell a story that's hard to grasp emotionally. 81% of marine species disappeared. 70% of terrestrial vertebrates went extinct. Even insects, those notoriously resilient survivors, lost entire orders. Trilobites, ancient armored creatures that had crawled across ocean floors for over 250 million years, vanished completely. Coral reefs, which had built intricate underwater cities for eons, collapsed so thoroughly that reef building stopped for 14 million years. Forests died en masse. The planet's green lungs simply gave out. What makes this extinction truly terrifying isn't just the scale. It's the speed. Early estimates suggested the dying unfolded over millions of years, giving species time to adapt or migrate. As dating methods improved, that timeline collapsed. Recent research indicates the main pulse of extinction occurred in perhaps 60,000 years. Some scientists argue it happened even faster, in as little as 8,000 years. On geological timescales, that's instantaneous. Evolution, which needs time to work its magic, couldn't keep pace. But here's what really happened before everything fell apart. The Permian period was a time of strange beauty. All of Earth's continents had merged into a single massive landmass called Pangaea. This supercontinent stretched from pole to pole, creating climates and ecosystems unlike anything we see today. The interior regions, thousands of kilometers from any ocean, became vast deserts where temperatures swung between blistering heat and bone-chilling cold. Coastal areas supported diverse life, but the heart of Pangaea was brutal. Life had adapted remarkably well to these conditions. Synapsids, the ancestors of mammals, dominated the land. Creatures like Dimetrodon, with its distinctive sail-like back, prowled as apex predators. Massive herbivores such as mosechops grazed in herds. In the oceans, biodiversity exploded. Brachiopods, ancient shellfish unrelated to modern clams, covered the seafloor in dense colonies. Ammonoids, spiral-shelled relatives of squid, hunted through the water column. Sea scorpions, some reaching over 2 meters in length, ruled as marine predators. The planet hummed with activity. Forests of seed ferns and conifers spread across habitable zones. Insects had diversified into countless forms. Complex food webs connected every ecosystem. Earth wasn't paradise, but it was stable, functioning, alive. Species had found their niches and settled into patterns that could have continued indefinitely. Then Siberia began to tear itself apart. Deep beneath what is now central Russia, something unprecedented started happening. Magma from the mantle began forcing its way toward the surface on a scale the planet had rarely witnessed. This wasn't a single volcano preparing to erupt. This was the formation of what geologists call a large igneous province, specifically the Siberian Traps. The name sounds almost quaint until you understand what it represents. The Siberian Traps eruptions continued for roughly 2 million years, spanning the Permian-Triassic boundary itself. During that time, volcanic activity covered an area larger than modern Europe with layer upon layer of basaltic lava. In some regions, the lava piled up over 3 kilometers thick. The volume of material ejected makes Yellowstone look insignificant. One researcher described it perfectly: it's literally a singular event in Earth's history. It's a monster. It makes Yellowstone look like the head of a pin. But the lava itself wasn't the main killer. As magma rose through Earth's crust, it encountered vast deposits of coal, salt, and limestone. These sedimentary layers had accumulated over hundreds of millions of years, locking away enormous quantities of carbon and other elements. When superheated magma cooked through these deposits, it triggered chemical reactions that released catastrophic amounts of gases into the atmosphere. Carbon dioxide poured out in volumes that defy easy comprehension. Scientists estimate the eruptions released 100,000 billion metric tons of CO2. That number is so large it becomes abstract. To put it in perspective, modern human civilization releases about 10 billion metric tons of carbon dioxide annually. The Siberian traps, over their active period, released the equivalent of 10,000 years of current human emissions every single year. The atmospheric concentration of CO2 didn't just rise. It exploded to 13 times pre-extinction levels. The greenhouse effect kicked in with unprecedented force. Global temperatures began climbing rapidly, rising by 6 to 10 degrees Celsius within a geological eye blink. Polar ice disappeared. Tundra ecosystems vanished as temperate conditions spread to the highest latitudes. The tropics, already warm, became furnaces where few organisms could function. Along with carbon dioxide came sulfur dioxide. When released in massive quantities, SO2 reacts with water vapor to form sulfuric acid. Acid rain began falling across the planet. Soils turned toxic. Lakes and rivers acidified. Plants struggled to photosynthesize as their leaves became damaged. The very chemistry of rain, normally a source of life, turned destructive. Ocean chemistry shifted in equally devastating ways. As atmospheric CO2 levels soared, the oceans absorbed enormous quantities of it. When CO2 dissolves in seawater, it forms carbonic acid, lowering the pH and making the water more acidic. Organisms that built calcium carbonate shells and skeletons, including most reef-building corals, brachiopods, and many plankton species, found themselves in an impossible situation. The chemical reactions needed to construct their protective structures simply stopped working efficiently. Warming oceans created another lethal problem. Water holds less dissolved oxygen at higher temperatures. As the seas warmed, oxygen levels dropped. Simultaneously, the warming disrupted ocean circulation patterns. Normally, cold water sinks at the poles and travels along the ocean floor toward the equator, carrying oxygen to deep waters. As polar temperatures rose, this circulation weakened. Deep ocean zones became oxygen-starved, creating what scientists call anoxic conditions. In some regions, conditions deteriorated even further into euxinia, where waters become not just oxygen-poor but actively toxic with hydrogen sulfide. This nasty compound, which smells like rotten eggs, is produced by certain bacteria thriving in oxygen-free environments. Pulses of hydrogen sulfide-rich water may have risen to the surface, poisoning marine life and even releasing toxic gas into the atmosphere. Recent research has added another layer to this catastrophe. Scientists studying oxygen isotopes in fossilized conodont teeth, tiny extinct eel-like creatures, discovered something unexpected about temperature patterns. Normally, Earth maintains strong temperature gradients between the equator and poles. During the Permian-Triassic extinction, these gradients collapsed. Temperatures became remarkably uniform across latitudes. The culprit appears to be mega El Niño events. Today's El Niño brings temporary warming to the Pacific and disrupts weather globally for a year or two. During the extinction, similar but far more intense events persisted for decades or even centuries. These mega El Niños created positive feedback loops. Warming intensified, which triggered more warming, which caused more disruption. Species simply couldn't adapt to climate patterns that refused to stabilize. On land, vegetation began dying on a massive scale. The fossil record reveals something called a fungal spike, elevated levels of fungal remains in rock layers corresponding to the extinction boundary. When scientists find this pattern, it indicates forests dying faster than they can decompose normally. Fungi thrive on dead organic matter, and suddenly they had more food than they could process. Nearly all the world's trees died. Dense gymnosperm woodlands that had covered high-latitude regions disappeared. The Glossopteris flora, seed ferns that dominated southern continents, collapsed around 370,000 years before the actual boundary. Their demise shows up indirectly in geological records through an abrupt change in river patterns. When rooted plants die off, rivers transform from meandering systems with stable banks into braided systems that constantly shift and erode. As forests vanished, one of Earth's primary carbon sinks disappeared with them. Plants pull CO2 from the atmosphere through photosynthesis. When the plant biosphere collapsed, this crucial mechanism for regulating atmospheric carbon stopped functioning. The planet lost its ability to naturally counteract the ongoing volcanic emissions. Carbon dioxide accumulated even faster, accelerating warming in a vicious cycle. The extinction didn't happen uniformly. Geographic patterns reveal crucial information about the kill mechanisms. High-latitude species, particularly those in polar regions, suffered the worst losses. Near-complete extinction struck these areas. Tropical species fared somewhat better, though still experiencing catastrophic losses. This pattern makes sense when you consider the combined effects of temperature and oxygen. Marine organisms have specific thermal tolerances and oxygen requirements. As water warmed, their metabolism sped up, increasing their oxygen demands precisely when available oxygen was dropping. For species already living near their thermal limits, there was nowhere to go. Polar species couldn't migrate toward cooler waters because those waters no longer existed. The extinction unfolded in pulses. Evidence suggests 1 to 3 distinct phases when death rates spiked. Some species made it through the first wave only to succumb during subsequent crises. This pattern hints at multiple triggers or cascading effects where initial impacts created conditions that generated additional disasters. What happened next would determine whether life on Earth could recover, or whether the planet would remain a desolate wasteland for millions of years. The mechanisms of death were clear enough, but survival would prove far more mysterious. In the oceans, survivors faced a world transformed beyond recognition. The water itself had become hostile. Hydrogen sulfide, that rotten egg smell you associate with swamps and sewage, now permeated vast stretches of ocean. Under normal conditions, oxygen-breathing bacteria dominate marine ecosystems. When oxygen disappears, different microbes take over. Anaerobic bacteria produce hydrogen sulfide as a metabolic byproduct. During the Permian-Triassic extinction, hydrogen sulfide levels rose so high that the gas didn't just stay dissolved in deep water. It bubbled up to the surface and escaped into the atmosphere. Imagine swimming in an ocean that smells like 1,000 rotten eggs. That's not even the worst part. Hydrogen sulfide is toxic to most complex life. It interferes with cellular respiration, essentially poisoning organisms from the inside. Marine creatures that managed to survive warming and oxygen depletion now had to contend with water that was actively poisonous. The geological record preserves evidence of these euxinic conditions, oxygen-free waters rich in hydrogen sulfide, in rock formations worldwide. Black shales containing high concentrations of pyrite, fool's gold, mark these deadly zones. But certain organisms found ways to persist. The survivors weren't random. Research examining 4 critical factors revealed patterns in who lived and who died. Position in the water column mattered enormously. Bottom-dwelling organisms, those living directly in sediment, suffered catastrophic losses. Creatures called bioturbators, animals that burrow through seafloor sediments mixing nutrients and oxygen, nearly vanished. The loss of this mixed sedimentary layer appears in the geological record as a stark boundary. Below it, sediments show signs of active life. Above it, nothing but undisturbed layers. Shell mineralization determined survival odds. Organisms with heavily calcified shells, thick protective armor built from calcium carbonate, died in disproportionate numbers. The problem was ocean acidification. As atmospheric CO2 dissolved into seawater, it formed carbonic acid. This lowered ocean pH, making it harder for creatures to extract calcium carbonate from water and build shells. Organisms that relied on thick shells for protection found themselves unable to maintain their defenses. Their shells literally began dissolving. Creatures with lighter calcification fared better. Brachiopods, ancient shellfish that superficially resemble clams, experienced massive losses. But the species that survived generally had thinner shells and more active control over their internal circulation. They possessed elaborate gas exchange mechanisms that let them extract oxygen more efficiently from oxygen-poor water. Simple breathing apparatuses became death sentences. Species diversity within a genus also predicted survival. Groups with many closely related species distributed across different environments had better odds. If one species went extinct, related forms might survive elsewhere and later repopulate empty habitats. Groups with just 1 or 2 species in limited ranges disappeared completely when their specific environment collapsed. Sensitivity to acidification was the final critical factor. Some organisms simply couldn't tolerate pH changes. Others showed remarkable resilience. The survivors weren't necessarily stronger or more complex. They were flexible. Generalist species that could eat various foods, tolerate different temperatures, and adapt their behavior had advantages specialists lacked. On land, the picture was equally grim but followed different rules. 8 entire orders of insects vanished. Think about that. Not just species or genera, but whole orders, taxonomic groups representing millions of years of evolutionary divergence. Modern insects are incredibly diverse, with over 1 million described species. The Permian-Triassic extinction wiped out fundamental branches of the insect family tree that had existed since the Carboniferous. 2/3 of tetrapod families disappeared. These were the dominant land animals, the synapsids, and early reptiles that had ruled terrestrial ecosystems for tens of millions of years. Massive herbivores that browsed on vegetation, apex predators with saber-like teeth, all gone. The speed of this collapse is what makes it so striking. These weren't species teetering on the edge of extinction before the catastrophe hit. Many were abundant and widespread right up until the boundary. Trilobites, those iconic armored arthropods that had crawled across seafloors for over 250 million years, finally met their end. They'd survived 4 previous mass extinctions. They'd adapted to countless environmental changes. They'd diversified into thousands of species occupying every marine niche imaginable. None of it mattered. The Permian-Triassic extinction erased them completely. Reef building stopped. For 14 million years afterward, no significant reef structures formed in shallow seas. Reefs are biodiversity hotspots, complex three-dimensional habitats that support enormous numbers of species. When reefs disappeared, they took their dependent ecosystems with them. The absence of reefs in early Triassic oceans reveals just how thoroughly marine ecosystems had collapsed. The basic ecological architecture that supports diverse life was gone. Tabulate and rugose corals, the primary reef builders of the Paleozoic, went extinct. Modern coral reefs are built by scleractinian corals, a completely different group that evolved later. The reefs we know today aren't descendants of Permian reefs. They're new constructions built by new organisms that emerged millions of years after the catastrophe. So what actually survived? The list is surprisingly short and decidedly unglamorous. Small terrestrial vertebrates made it through, particularly those that could burrow underground or shelter in crevices. Body size had become a liability. Large animals need more food, more oxygen, more stable conditions. When all three became scarce simultaneously, being small and undemanding became the ultimate survival strategy. Certain fish lineages persisted, mostly small-bodied forms that could tolerate low oxygen. Sharks, surprisingly, came through relatively intact. Their cartilaginous skeletons required less calcium than bony fish, giving them an edge as ocean chemistry shifted. They could also regulate their internal salt balance more effectively, helping them cope with changing ocean chemistry. Amphibians showed remarkable resilience. Their ability to breathe through their skin supplemented lung capacity, allowing them to extract oxygen from both air and water. Many could also enter dormant states during harsh conditions, essentially waiting out the worst phases. Some amphibian lineages that made it through the extinction would later give rise to the ancestors of modern frogs and salamanders. Early reptiles, particularly small insectivorous forms, survived in decent numbers. Their scaly skin reduced water loss, crucial as climates became more arid. Their eggs with protective shells didn't require standing water, allowing them to reproduce away from increasingly hostile aquatic environments. These survivors formed the foundation for the later radiation of reptiles during the Triassic, eventually leading to dinosaurs. But the real story of survival belongs to organisms you wouldn't expect. Lystrosaurus, a dicynodont about the size of a pig, became the most abundant land vertebrate in the early Triassic. Before the extinction, Lystrosaurus was just one of many synapsid species. Afterward, it accounted for roughly 90% of terrestrial vertebrate fossils. Not because it was superior, but because almost everything else had died. This phenomenon has a name: disaster taxa. These are species that explode in abundance immediately after mass extinctions, not through competitive superiority but through sheer lack of competition. Lystrosaurus didn't outcompete other species. It simply inherited an empty planet. The fossil record from the early Triassic shows something bizarre. Before the extinction, communities varied dramatically from place to place. Different continents hosted different species adapted to local conditions. After the extinction, the same handful of species appeared everywhere. Fossils from South Africa look nearly identical to those from Antarctica, Russia, and China. The entire planet became monotonous. Scientists use a striking analogy. If someone asked you today where to find kangaroos, you'd say Australia. Now imagine a catastrophe occurred and suddenly kangaroos showed up in North America, Europe, Asia, everywhere. That's essentially what happened in the early Triassic. Geographic barriers that normally keep species separated became meaningless because so few species remained. Marine environments showed the same pattern. A few disaster taxa, mostly small bivalves and simple brachiopods, dominated oceans worldwide. Species that previously existed only in specific regions suddenly had global distributions. Not through dispersal or migration, but because everything else had vanished, leaving them alone on an empty stage. The survivors weren't thriving. They were persisting in a world that remained fundamentally unstable for millions of years after the initial extinction pulses. Atmospheric carbon dioxide levels continued climbing, driven by ongoing volcanic activity in Siberia. The Siberian traps weren't a single explosive event like later asteroid impacts. They represented sustained eruption over roughly 2 million years, with peak activity coinciding precisely with the extinction boundary. Magma punched through coal deposits and organic-rich sediments, essentially cooking them and releasing massive quantities of greenhouse gases. Not just carbon dioxide, but methane, a far more potent warming agent. Temperature records preserved in ancient tooth enamel tell a brutal story. Conodonts, tiny eel-like creatures with mineralized teeth, left behind a thermal archive. Their oxygen isotope ratios reveal that global temperatures spiked by 6 to 10 degrees Celsius. That doesn't sound catastrophic until you realize this warming happened over perhaps 100,000 years. For evolution, that's instantaneous. Species adapted to stable climates over millions of years suddenly faced conditions that changed faster than genetics could respond. But raw temperature increase wasn't the primary killer. Recent research identified something far more insidious. The warming triggered mega El Niño events, climate oscillations that make modern El Niños look gentle. These weren't temporary disruptions lasting months. They persisted for years, possibly decades at a time. Temperature gradients between equator and poles collapsed. Tropical heat spread toward high latitudes. Species that had evolved for cooler environments found themselves trapped in zones that became lethally hot. Here's where the cascade accelerated. Plants began dying on a massive scale. Not just individual trees, but entire forest ecosystems. The fossil record shows something called a fungal spike, elevated levels of fungal spores appearing globally in sediments right at the extinction boundary. Fungi decompose dead organic matter. Their explosion in abundance means one thing. The world's forests were dying faster than normal decay processes could handle. When forests collapsed, the planet lost its primary carbon sink. Living plants pull CO2 from the atmosphere through photosynthesis, storing it in wood and soil. Dead plants stop removing carbon. Worse, their decomposition releases stored carbon back into the atmosphere. The Siberian traps kept pumping out greenhouse gases while Earth's biological systems for removing those gases shut down. Positive feedback loop. Warming killed plants, dead plants meant more atmospheric carbon meant more warming. On land, river systems changed character completely. Meandering rivers, which require vegetation to stabilize their banks, transformed into braided channels that shifted and wandered. Without plant roots holding soil in place, erosion accelerated. Sediments clogged waterways. The landscape itself became unstable. Marine environments faced a different horror. Warmer water holds less dissolved oxygen. Basic chemistry. As ocean temperatures climbed, oxygen concentrations dropped. Simultaneously, marine animals' metabolism sped up in the heat, requiring more oxygen precisely when less became available. Researchers modeled this lethal combination using modern species' oxygen requirements. The results matched fossil distributions with eerie precision. Species living in cold polar waters, where oxygen demands were already high, went extinct almost completely. Tropical species fared slightly better, already adapted to warmer, lower oxygen conditions. But even tropical oceans reached thresholds where metabolism outpaced oxygen supply. Animals essentially suffocated in warming water. The ocean circulation system began breaking down. Normally, cold polar water sinks and flows toward the equator along the seafloor, while warm tropical water moves poleward at the surface. This thermohaline circulation acts like a planetary conveyor belt, distributing heat and nutrients. As temperature gradients flattened, circulation weakened. Deep ocean zones became stagnant. Stagnant water turns anoxic. Without circulation bringing fresh oxygen to depth, deep ocean zones essentially died. Anaerobic bacteria took over, producing hydrogen sulfide, the same compound that gives rotten eggs their smell. Except this wasn't a small pocket of decay. Vast stretches of deep ocean became toxic. Some researchers suggest hydrogen sulfide periodically bubbled to the surface, poisoning shallow waters and even escaping into the atmosphere. Ocean acidification compounded every other problem. When CO2 dissolves in seawater, it forms carbonic acid. The more atmospheric CO2, the more acidic oceans become. Calcium carbonate, the material organisms use to build shells and skeletons, dissolves in acidic water. Species that had protected themselves with armor for hundreds of millions of years suddenly found their defenses literally dissolving. Trilobites, ancient arthropods that had survived previous extinction events, vanished entirely. They'd existed for over 270 million years, weathering countless environmental changes. The Permian-Triassic event erased them permanently. Coral reefs, those complex ecosystems supporting thousands of species, collapsed globally. It would take 14 million years before reef-building organisms recovered enough to construct similar structures. On land, insects suffered their only known mass extinction. 8 entire orders disappeared. Insects typically survive environmental catastrophes because of their small size, rapid reproduction, and ability to exploit diverse food sources. Not this time. Even organisms at the base of food webs couldn't find refuge. What makes the Permian-Triassic extinction uniquely devastating isn't just the raw percentage of species lost. It's the complete disruption of ecological functioning. Previous mass extinctions removed many species but left ecosystem structures largely intact. This event shattered those structures. Food webs didn't just simplify. They collapsed to nearly nothing and had to rebuild from scratch. The aftermath persisted for an almost incomprehensible duration. 5 to 6 million years of continued environmental instability. Survivors that managed to establish populations faced repeated setbacks as climate continued oscillating wildly. Carbon cycle perturbations, oxygen fluctuations, temperature spikes. Communities would begin recovering, then get knocked back by another pulse of environmental chaos. This explains why disaster taxa dominated for so long. Normal competitive dynamics couldn't operate. Ecosystems never achieved enough stability for complex food webs to re-establish. Lystrosaurus wasn't necessarily better adapted than other organisms. It just happened to survive the initial extinction and could tolerate the ongoing instability. In a stable environment, more specialized species would have outcompeted. In perpetual crisis, being generalized and tolerant meant everything. Terrestrial ecosystems took 30 million years to fully recover. Not recover to pre-extinction diversity, that never happened. Many ecological roles that existed before the Permian-Triassic boundary remained empty permanently. But 30 million years before ecosystems achieved new stable configurations, before food webs became complex again, before the planet hosted comparable biodiversity. Marine ecosystems showed similar timescales. Shallow marine communities recovered somewhat faster, within 5 to 10 million years. But deep ocean ecosystems and complex trophic structures took up to 50 million years. The extinction didn't just reset Earth's biological clock. It fundamentally altered what was possible. Why did this extinction exceed all others in severity? The Siberian traps erupted at a uniquely vulnerable moment in Earth's history. Pangaea, the supercontinent, had just formed. Land masses concentrated in one area reduced total coastline length, eliminating shallow marine habitats that typically serve as refuges during environmental stress. Continental interiors, far from ocean's moderating influence, experienced extreme temperature swings. Species had fewer options for geographic escape. The volcanism itself reached unprecedented scales. Individual eruptions weren't necessarily larger than other volcanic events, but the duration and total volume exceeded anything else in the Phanerozoic record. 2 million years of sustained eruption. No previous volcanic episode approached that timescale. The planet's environmental systems couldn't buffer or absorb the continuous input of greenhouse gases. Most critically, the extinction occurred faster than recovery mechanisms could operate. Normal extinction events, even severe ones, happen slowly enough that evolution fills empty niches relatively quickly. The Permian-Triassic event compressed catastrophic environmental change into a window too narrow for adaptive responses. Species either possessed pre-existing traits allowing survival or they died. No time for evolution to generate new solutions. Understanding how thoroughly the planet broke during this event makes the survival of any lineage remarkable. Every living organism today, from bacteria to blue whales, descends from something that made it through those 5 million years of hell. Our own distant ancestors, small cynodont synapsids barely distinguishable from other survivors, persisted when 90% of life could not. The Permian-Triassic extinction wasn't merely Earth's deadliest event. It represented the closest life has come to complete extinction since complex organisms evolved. A few more degrees of warming, a bit more ocean acidification, slightly longer duration of environmental chaos, and perhaps nothing would have survived. Yet that scenario nearly happened. And the mechanisms that pushed Earth toward complete biosphere collapse reveal patterns that operate across all mass extinctions. The kill mechanisms during the Permian-Triassic weren't random. They formed an interconnected cascade where each environmental change amplified the next. Understanding this cascade explains why the extinction exceeded all others in lethality. Start with the oceans. Marine ecosystems experienced a transition from oxygenated to anoxic-euxinic conditions across most of the globe. Anoxic means depleted of oxygen. Euxinic means not only oxygen depleted but saturated with hydrogen sulfide. Seawater became actively toxic. This wasn't uniform. Different ocean basins showed regional variability in timing and intensity, but the overall pattern held. Decreased ocean ventilation coincided with rapidly rising temperatures. The combination proved lethal. Here's why. As water temperature increases, its capacity to hold dissolved oxygen decreases. Basic chemistry. Warmer water physically cannot contain as much oxygen as cooler water. At the same time, warmer temperatures increase metabolic rates in marine animals. Their bodies require more oxygen precisely when less is available. Research combining models of ocean conditions with animal metabolism and paleoceanographic records demonstrates this thermal oxygen squeeze killed most marine life. Species couldn't breathe. Their physiology demanded oxygen levels the environment could no longer provide. The geographic pattern of extinction confirms this mechanism. High-latitude species, especially those with high oxygen demands, were nearly completely wiped out. Polar regions, traditionally cooler and oxygen-rich, warmed dramatically. Animals evolved for cold, oxygen-rich water suddenly faced warm, oxygen-poor conditions. No evolutionary pathway existed from their current physiology to what survival required. Tropical species fared slightly better. Not because tropical waters remained cool or oxygen-rich, they didn't. But tropical organisms already possessed adaptations for warmer temperatures and lower oxygen. Their baseline tolerance was higher. Many still died, but survival rates exceeded those at higher latitudes. The fossil record shows species distribution before the extinction. Then, at the boundary, diversity collapses. The geographic signature matches what models predict for warming-driven oxygen loss. Theory and evidence align perfectly. But oxygen depletion alone doesn't explain the hydrogen sulfide. That requires understanding what happens when oceans stop mixing properly. Normal ocean circulation brings oxygen-rich surface water down into the depths. Upwelling brings nutrient-rich deep water to the surface. This constant mixing maintains oxygen levels throughout the water column and prevents toxic compounds from accumulating. During the Permian-Triassic, warming disrupted this circulation. Temperature differences between surface and deep water decreased. Without strong temperature gradients, the physical engine driving ocean mixing weakened. Stratification increased. Surface water stopped sinking. Deep water stopped rising. In stagnant deep waters, oxygen ran out quickly as bacteria consumed it, decomposing organic matter. Once oxygen disappeared, anaerobic bacteria took over. These organisms produce hydrogen sulfide as a metabolic byproduct. In oxygenated water, other bacteria consume hydrogen sulfide, keeping it in check. Without oxygen, nothing stops hydrogen sulfide accumulation. Concentrations built up over thousands of years. Eventually, hydrogen sulfide saturated deep waters. Then it began rising toward the surface. Some researchers suggest hydrogen sulfide breached the ocean surface, releasing toxic gas into the atmosphere. Others think it remained dissolved but created a toxic layer throughout much of the ocean's depth. Either scenario meant death for most marine organisms. Hydrogen sulfide is directly toxic. It interferes with cellular respiration. Animals swimming through euxinic waters were essentially swimming through poison. The loss of marine species reached 96%. Nearly everything died. The extinction rate wasn't gradual. Bioturbators, organisms that churn through seafloor sediments, were extremely severely affected. The loss of the sedimentary mixed layer in many marine facies shows that animals stopped disturbing the seafloor. Nothing was left to dig, burrow, or mix sediments. The ocean floor became a dead zone. Reef building stopped for 14 million years. Shallow seas, typically refuges during environmental stress, offered no protection. Marine animals in reefs and shallow waters were especially hard hit. The structures that had provided habitat for millions of years simply ceased to exist. Specific groups that had dominated oceans for hundreds of millions of years vanished entirely. Trilobites, survivors of multiple previous mass extinctions, finally went extinct. They'd existed for over 250 million years. The Permian-Triassic boundary ended them. Tabulate and rugose corals disappeared. Blastoid echinoderms, graptolites, eurypterids, acanthodians, placoderms, all gone. The extinction primarily affected organisms with calcium carbonate skeletons, especially those reliant on stable CO2 levels to produce their skeletons. Ocean acidification from dissolved atmospheric CO2 made skeleton construction impossible. Carbonate chemistry shifted beyond what these organisms could tolerate. Research identified 4 factors most essential to survival. Where in the water organisms lived, the mineralization of their shells, species diversity within their genus, sensitivity to acidification. Surviving groups generally had active control of circulation, elaborate gas exchange mechanisms, and light calcification. Heavily calcified organisms with simpler breathing apparatuses suffered the greatest loss of species diversity. This explains why certain groups survived while others didn't. Brachiopods, corals, bryozoans, foraminifers, ostracods, all heavily calcified benthic organisms, were devastated. Meanwhile, some less calcified, more physiologically flexible groups persisted at low levels. Planktonic populations underwent fundamental change. Eukaryotic algae, which had formed the base of marine food webs, were replaced by nitrogen-fixing bacteria, green sulfur bacteria, sulfate-reducing bacteria, and prasinophytes. The entire foundation of marine ecosystems shifted from complex eukaryotic organisms to simpler prokaryotic forms. On land, the devastation followed different pathways but achieved similar totality. Recent research published in Nature Communications identified the primary cause of prolonged post-extinction warming: the collapse of tropical forests. When global forests died, carbon sequestration essentially stopped. Photosynthesis removes CO2 from the atmosphere. Plants are Earth's natural mechanism for keeping CO2 in check. When nearly all trees died, that mechanism broke. The fungal spike in Permo-Triassic rocks worldwide confirms this. Elevated levels of fungal remains with few tree pollen grains. Fungi decompose dead organic matter. A planet-wide fungal spike means a planet-wide mass of dead vegetation being broken down. Nearly all the world's trees died en masse. The Glossopteris flora that characterized high-latitude Gondwana collapsed in Australia around 370,000 years before the Permian-Triassic boundary. In western Gondwana the timing is less constrained but still likely occurred hundreds of thousands of years before the boundary. This collapse is indirectly marked by abrupt changes in river morphology from meandering to braided river systems. Rivers change character when vegetation dies. Without root systems stabilizing banks, rivers spread out, become shallow and multi-channeled. The widespread demise of rooted plants restructured hydrology across continents. Pollen studies from East Greenland showed dense gymnosperm woodlands before the event. Then, at the boundary, those woodlands disappeared. Smaller herbaceous plants including Lycopodiophyta increased in diversity. The world shifted from forests to scrubland. From complex plant communities to pioneer species. This vegetation collapse created a feedback loop. Volcanic eruptions released CO2. Plants normally absorb that CO2. With plants dead or dying, atmospheric CO2 continued climbing unchecked. More CO2 meant more warming. More warming meant more plant death. The cycle amplified itself. This explains why super-greenhouse conditions persisted for 5 million years after the initial extinction. The planet's natural CO2 regulation system had broken. Volcanic inputs continued. Nothing removed the gas from the atmosphere at rates approaching volcanic output. Temperature climbed beyond anything Earth had experienced before or since. Scientists have used plant fossils to model a dramatic 10 degrees Celsius rise in global temperatures. On land, surface temperatures increased by 6 to 10 degrees Celsius. Too rapid for life to evolve and adapt. Evolution operates on timescales of hundreds of thousands to millions of years. The Permian-Triassic warming occurred in tens of thousands of years. No time for adaptive responses. Recent research on mega El Niño events adds another layer. The study, published in Science, shows that the collapse of temperature gradients in low and mid-latitudes was driven by prolonged, intense El Niño events far exceeding anything witnessed today. Researchers stated that essentially, it got too hot everywhere. Species were simply not equipped to adapt or evolve quickly enough. These mega El Niños created positive climate feedbacks. Incredibly warm conditions started in the tropics, then spread globally. Vegetation died back. With plants dying, Earth's CO2 removal mechanism failed, allowing continued volcanic CO2 buildup. This also explains why extinction on land occurred tens of thousands of years before extinction in the oceans. Terrestrial environments respond faster to atmospheric changes. Marine systems, with greater thermal inertia, lagged behind. But eventually, ocean warming caught up and exceeded what marine life could tolerate. Less than 1/3 of large animal species on land survived. The volcanic eruptions themselves caused immediate problems. Sulfur dioxide and carbon dioxide poisoned the air. Ash and soot blocked sunlight, preventing plant growth. Food chains collapsed from the bottom up. With little greenery to eat, herbivores died. Predators followed. The speed of terrestrial collapse reveals something crucial about ecosystem fragility. Complex food webs depend on continuous energy flow from primary producers. When photosynthesis stops, everything above that level has days to weeks before starvation. Large animals, with high energy demands, died first and fastest. Yet from this near-total devastation, life found pathways to persist. Understanding what survived and how sets up the recovery story, a process that would test the limits of biological resilience for millions of years to come. In the aftermath, what survived wasn't selected by strength or size. The winners were organisms whose traits happened to match a world nobody could have predicted. Lystrosaurus. A dicynodont therapsid about the size of a pig, with a blunt snout, barrel chest, and sprawling limbs. Before the extinction, it was one species among hundreds. After, it dominated. Fossil evidence shows that Lystrosaurus accounted for approximately 90% of terrestrial vertebrates in the earliest Triassic. 90%. Imagine walking across early Triassic landscapes and seeing nothing but variations of one creature, from horizon to horizon, across every continent. This wasn't evolutionary superiority. This was luck combined with specific physiological advantages. Lystrosaurus could dig burrows, escaping surface temperature extremes. It had a generalist diet, feeding on whatever sparse vegetation remained. Its metabolism was efficient enough to survive on minimal food. Most importantly, it reproduced quickly. Where larger predators needed years to reach maturity, Lystrosaurus populations could rebound within seasons. Scientists call these survivors disaster taxa. Species that explode in abundance immediately after catastrophic events, filling ecological space left vacant by extinctions. They're not the most advanced organisms. They're the most opportunistic. The most flexible. The most boringly successful in conditions where nothing else can compete because nothing else exists. The pattern repeated in the oceans. marine post-extinction faunas were dominated by a handful of forms. Bivalves like Chlorea, Unionites, and Eumorphotis spread across seafloors worldwide. Conodonts Clarkina and Hindiotis became ubiquitous. The inarticulate brachiopod Lingularia, with its simple body plan and minimal calcification needs, thrived in acidified waters that killed more complex relatives. What's striking isn't that these species survived, but where they appeared. Before the extinction, marine species showed clear geographic distributions. Certain organisms lived only in specific regions, adapted to local conditions. After the boundary, that geographic structure collapsed. Species previously found only in one ocean suddenly appeared globally. Researchers describe this as cosmopolitanism, a planetary homogenization of life. The modern analogy makes it visceral. If you asked where to find kangaroos today, the answer is Australia. Now imagine a catastrophe occurs, and afterward, kangaroos appear in massive numbers across every continent. Europe, Africa, Asia, the Americas. Everywhere you look, kangaroos. That's what early Triassic oceans resembled. Fossils show communities going from richly diverse assemblages to boringly alike groups, no matter where you sample. This homogenization reveals something profound about how ecosystems collapse and rebuild. Diversity doesn't return immediately. Complex food webs with specialized niches take millions of years to re-establish. In the immediate aftermath, life reverts to simpler patterns. Generalists dominate. Specialists vanish. Ecosystems flatten into crude approximations of what existed before. But even disaster taxa faced challenges. The extinction event itself lasted perhaps 60,000 years. The aftermath persisted for 5 million. Volcanic activity didn't stop at the boundary. The Siberian traps continued erupting. Carbon cycle disruptions continued through the entire early Triassic. Communities that survived the initial extinction and began rebuilding were hit again by recurring environmental shocks. Temperature spikes. Ocean anoxia returning impulses. Acidification events. Each wave of stress interrupted recovery. Terrestrial ecosystems endured boom and bust cycles for 8 million years before stabilizing. Populations would grow, ecosystems would begin complexifying, then another environmental shock would knock everything back to simpler states. 2 steps forward, 1 step back, repeated across geological time. The low diversity of surviving vertebrates created its own problems. With few species present, ecosystems lacked redundancy. If one disaster taxon declined, nothing filled its role. Food webs remained fragile, vulnerable to any perturbation. Contrast this with pre-extinction ecosystems where multiple species performed similar functions. Lose one, others compensate. In the early Triassic, there were no backup species. Marine ecosystems faced additional constraints. Research shows that taxonomic diversity at the community level didn't recover to pre-extinction levels during the early or middle Triassic. It reached only a low plateau after each extinction pulse and remained depressed well into the late Triassic. Full ecological recovery, where complex trophic structures and diverse guilds re-established, took roughly 50 million years. That's not a typo. 50 million years. While taxonomic diversity, the raw number of species, recovered in 5 to 10 million years, true ecosystem function took 5 times longer. Having species present doesn't mean having a functioning ecosystem. You need the right species in the right roles with the right interactions. Rebuilding that structure from near zero is astonishingly slow. Why did recovery take so long? several factors compounded. First, the carbon cycle remained disrupted. Atmospheric CO2 stayed elevated because volcanic inputs continued while biological carbon sequestration remained weak. Without dense vegetation or diverse marine plankton communities, CO2 removal rates stayed far below normal. This perpetuated greenhouse conditions, maintaining environmental stress. Second, ocean chemistry stayed hostile. Anoxic zones expanded during warm periods. Acidification events recurred whenever volcanic pulses added more CO2. Marine organisms trying to rebuild calcium carbonate structures faced repeated setbacks. Reefs didn't begin reforming until 14 million years into the Triassic. That's 14 million years without the ocean's most productive ecosystems. Third, evolutionary innovation takes time. After mass extinctions, surviving lineages diversify to fill empty niches. But evolution doesn't work instantly. Mutations arise randomly. Selection operates over generations. Beneficial traits must accumulate. New body plans must stabilize. For complex organisms with long generation times, meaningful evolutionary change requires hundreds of thousands to millions of years. The fossil record documents this slow rebuilding. Early Triassic communities show low gill diversity. Gill refers to groups of species that exploit resources in similar ways. Before the extinction, multiple species of filter feeders, multiple species of predators, multiple species of burrowers coexisted. After, each guild had one or two representatives at most. Ecosystems functioned, barely, but lacked the complexity and resilience of pre-extinction systems. Geographic patterns of recovery varied. Some regions rebounded faster than others. Studies of localized marine ecosystems, like the Paris biota in what's now Idaho, show recovery occurring in 1.3 million years. But that's recovery of basic ecosystem function, not restoration of pre-extinction diversity or complexity. These were simple communities compared to Permian reefs. On land, the situation was even more extreme. Terrestrial vertebrate fauna took 30 million years to fully recover numerically and ecologically. Not until the late Triassic did land ecosystems display the diversity and complexity seen in the Permian. That's when dinosaurs, pterosaurs, crocodiles, archosaurs, amphibians, and mammaliforms became abundant and diverse. The world we associate with the Mesozoic emerged only after 3 decades of millions of years rebuilding from near oblivion. What determined which lineages eventually diversified? The answer involves both chance and contingency. Survivors at the boundary had won the first lottery, possessing traits that allowed persistence through catastrophe. But recovery success required different traits. Flexibility in diet and habitat. Efficient reproduction. Ability to exploit newly available niches. Lineages that survived but couldn't adapt to changing conditions during recovery stagnated or went extinct in secondary waves. The early Triassic world was simultaneously empty and full. Empty of diversity, of complex ecosystems, of specialized organisms. Full of opportunity for any lineage capable of exploiting it. Disaster taxa like Lystrosaurus thrived not because they were advanced, but because they faced no competition. Remove competition, and even modest capabilities become sufficient for dominance. This dynamic explains the peculiar nature of post-extinction evolution. After major extinctions, life doesn't gradually rebuild pre-existing structures. It experiments, tries new body plans, tests different ecological strategies. Most experiments fail. Some succeed spectacularly, establishing lineages that will dominate for tens or hundreds of millions of years. The Permian-Triassic boundary marks one such turning point. The Paleozoic world, with its characteristic fauna and ecosystems, died. What emerged during the Triassic recovery was fundamentally different. New dominant groups, new ecosystem structures, new rules for who succeeds and who fails. Yet even as life slowly rebuilt, the planet itself continued changing. Continental drift proceeded. Pangaea persisted as a supercontinent but began showing signs of future fragmentation. Climate zones shifted. Ocean circulation patterns evolved. The stage was being set for the next great radiation of life, though nobody watching the barren early Triassic landscapes could have predicted what was coming. The emptiness didn't last. It couldn't. Earth operates on momentum, chemical cycles, physical processes that continue regardless of biological catastrophe. Rivers still flowed, carrying nutrients to the seas. Volcanic activity persisted, releasing gases that weathered rocks and fed primitive organisms. Solar radiation still reached the surface, driving whatever photosynthesis survived. The planet provided the raw materials. Life had to figure out what to do with them. By the Middle Triassic, about 10 to 15 million years after the extinction, something shifted. Not a sudden transformation, but a gradual acceleration in the pace of evolutionary innovation. Survivors had stabilized. Populations had grown. And now, with ecological space still largely vacant, natural selection began favoring not just survival, but expansion. Organisms that could exploit multiple food sources, that could tolerate varying conditions, that reproduced efficiently, these lineages spread rapidly across landscapes and seascapes that offered minimal resistance. On land, the synapsid lineage that would eventually produce mammals continued evolving. Cynodonts emerged, dog-sized predators and omnivores with increasingly mammal-like features. More efficient jaws. Better hearing. Possible fur. These weren't mammals yet, but they were heading that direction, experimenting with traits that would define an entire class of animals tens of millions of years later. Alongside them, early archosaurs appeared. The archosaur group would eventually split into two major branches. One leading to crocodilians. The other leading to dinosaurs and birds. In the late Triassic, dinosaurs made their entrance. Small, bipedal, unremarkable at first. Nothing about early dinosaurs suggested they would dominate terrestrial ecosystems for 160 million years. They were simply another group of reptiles trying to survive in a world still recovering from catastrophe. But they possessed certain advantages. Upright limb posture that improved locomotion efficiency. Relatively large brains for their body size. Fast growth rates. These traits didn't guarantee success, but they provided options. And in an unpredictable world, options matter. The oceans told a parallel story. Reef ecosystems, absent for 14 million years after the extinction, began rebuilding in the Middle Triassic. Not with the same organisms, those were gone, but with new reef builders. Corals returned, but different families than dominated Permian reefs. Marine reptiles diversified into the ecological roles left vacant by extinct groups. Ichthyosaurs evolved streamlined bodies for fast swimming. Plesiosaurs developed long necks for hunting in different ways. Placodonts specialized in crushing shellfish. Each group represented an evolutionary experiment, testing whether a particular body plan and lifestyle could work in post-extinction oceans. But here's what makes this recovery period so instructive. The late Triassic world looked nothing like the Permian world. Same planet, same basic physics and chemistry, completely different biological cast. Pangaea still dominated geography, but the ecosystems populating that supercontinent bore little resemblance to what came before. This reveals something fundamental about how life responds to mass extinction. Recovery isn't restoration. It's renovation. The old blueprints are discarded. New designs are tested. Most fail. A few succeed spectacularly. The late Triassic also demonstrated that recovery from mass extinction doesn't follow a smooth upward trajectory. Around 201 million years ago, Earth experienced another major extinction event at the Triassic-Jurassic boundary. Not as severe as the end Permian, but significant enough to eliminate roughly 70 to 75% of species. The cause appears linked to volcanic activity associated with the breakup of Pangaea and the opening of the Atlantic Ocean. Once again, massive carbon dioxide release. Once again, rapid climate warming. Once again, ocean acidification. Life had barely finished recovering from one catastrophe when another struck. This second extinction disproportionately affected certain groups while barely touching others. Many large amphibians vanished. Several archosaur lineages went extinct. But dinosaurs, still relatively minor players in late Triassic ecosystems, survived. And in the aftermath, with many competitors removed, dinosaurs underwent explosive diversification. By the early Jurassic, they had become the dominant large terrestrial animals. Their success wasn't inevitable. It was contingent on surviving the Triassic-Jurassic boundary while many other groups did not. What does this tell us about mass extinctions and recovery? First, that recovery timelines depend on the severity and nature of the extinction. The end Permian, with its combination of rapid warming, ocean anoxia, acid rain, and ecosystem collapse, required 10 million years for basic recovery and 30 million for full restoration of terrestrial ecosystem complexity. The Triassic-Jurassic event, less severe in its mechanisms, allowed faster rebound. Second, that which groups dominate after extinction depends partly on pre-existing traits, but largely on chance. Dinosaurs didn't dominate because they were superior to other Triassic reptiles. They dominated because they survived when others didn't, then exploited the resulting opportunities. The pattern repeats throughout Earth's history. 5 major mass extinctions punctuate the Phanerozoic Eon, each reshaping life's trajectory. The late Ordovician extinction, caused by glaciation and sea level changes. The late Devonian extinctions, possibly linked to plant evolution altering atmospheric composition. The end Permian, Earth's closest approach to complete sterilization. The Triassic-Jurassic boundary. And finally, the end Cretaceous asteroid impact that eliminated non-avian dinosaurs. Each event cleared ecological space. Each allowed previously minor groups to become dominant. Each reset the evolutionary clock, forcing life to rebuild from whatever survived. But mass extinctions aren't just destructive. They're also creative, in a brutal sense. By eliminating dominant groups, they allow innovation. Mammals remained small and ecologically marginal for 160 million years while dinosaurs dominated. Only after the end Cretaceous extinction did mammals diversify into the full range of body sizes and ecological roles. Birds, despite evolving flight during the Jurassic, remained relatively limited in diversity until after non-avian dinosaurs vanished. Remove the dominant players, and previously suppressed groups explode into vacant niches. The Paleozoic era, spanning 290 million years from the Cambrian explosion to the end Permian extinction, represents one complete cycle of this pattern. complex life emerged. Diversified. Filled ecosystems on land and in the sea. Developed intricate food webs, specialized forms, stable communities. Then catastrophe struck. And almost everything ended. The Mesozoic era that followed, the age of dinosaurs, represents the recovery and subsequent rediversification. A new cycle with new dominant groups, new ecosystems, new rules. That cycle also ended 66 million years ago, when an asteroid impact triggered the end Cretaceous extinction. And we're living in the recovery period, the Cenozoic era, when mammals and birds occupy the roles dinosaurs once filled. Earth's biological history is therefore not a steady progression toward increasing complexity or advancement. It's a series of boom and bust cycles. Long periods of relative stability punctuated by catastrophic extinctions, followed by recovery periods when life experiments with new forms and strategies. The Permian-Triassic boundary represents the most dramatic of these transitions, the moment when life came closest to complete failure. That it didn't fail completely, that survivors persisted and eventually diversified, testifies to life's resilience. But it also reveals life's fragility. Remove too many links from an ecosystem, disrupt too many chemical cycles, push environmental conditions beyond tolerable limits, and even 290 million years of evolutionary success offers no protection. Today, we're conducting our own extinction experiment. Atmospheric carbon dioxide levels are rising faster than at any time in the past 66 million years, possibly faster than any time since the end Permian. Global temperatures are increasing. Oceans are acidifying. Species are disappearing at rates hundreds of times above background levels. We're not at Permian-Triassic severity yet, but we're moving in that direction, and we're doing it with remarkable speed. The difference is that this time, the cause isn't volcanic eruptions or asteroid impacts. It's us. A single species, Homo sapiens, is altering planetary chemistry and climate at scales previously achievable only by massive geological events. The Paleozoic era ended in fire, suffocation, and acid. What emerged from that ending was a planet fundamentally transformed, populated by organisms that would have been unrecognizable to any Permian creature. Life continued, but not the same life. Recovery occurred, but not restoration. The world moved forward, carrying scars that remain visible in the fossil record 252 million years later. That's the pattern extinction establishes. Not death of all life, but death of familiar life. Not permanent ending, but permanent transformation. The question facing us now is whether we want to be part of the next transformation, or whether we'll join the trilobites, the tabulate corals, and the glossopterous forests in the fossil record of extinct failures. Earth will continue regardless. Life will persist in some form. But which forms survive, and which vanish forever, that's being decided right now, in real time, by choices we make daily. The Paleozoic era teaches us that recovery is possible. It also teaches us that recovery takes millions of years, and what emerges looks nothing like what was lost. The Permian-Triassic extinction stands as Earth's most severe test of biological resilience, a moment when the planet approached the threshold of complete sterilization. 96% of marine species vanished. 70% of land vertebrates disappeared. Even insects, those tireless survivors of every previous catastrophe, lost entire orders to the void. The numbers become abstract at this scale, almost meaningless, until you consider what they represent. Entire evolutionary lineages, hundreds of millions of years in the making, erased in perhaps 60,000 years. What makes this extinction uniquely terrifying isn't just magnitude. It's the mechanism. A cascade of environmental failures where each problem amplified the next until the planet's life support system simply broke. Volcanic eruptions triggered warming. Warming killed forests. Dead forests meant carbon accumulated unchecked. More carbon meant more warming. Ocean circulation collapsed. Oxygen disappeared from seawater. Hydrogen sulfide saturated the depths. Temperature gradients flattened. Mega El Niño events persisted for decades. Acid rain stripped nutrients from soil. Marine creatures suffocated in warming waters while their shells dissolved in acidifying seas. The Siberian traps weren't just large. They were sustained. 2 million years of eruption, cooking through coal deposits and organic sediments, releasing greenhouse gases at rates that overwhelmed every natural buffer. The planet tried to compensate. Photosynthesis removes CO2. Weathering locks carbon in rocks. Ocean chemistry absorbs atmospheric changes. But these processes operate on timescales of hundreds of thousands to millions of years. The Siberian traps dumped carbon into the atmosphere faster than any removal mechanism could respond. Positive feedbacks took over. The climate system entered runaway mode. Life, which had spent 290 million years building complex ecosystems, watched those ecosystems collapse from the bottom up. Primary producers died first, cutting energy flow at its source. Herbivores followed within months. Predators starved soon after. Food webs didn't simplify. They shattered. What remained were fragments, disconnected survivors in a world that no longer made biological sense. Recovery took 10 million years just to restore basic ecosystem function. 30 million years passed before terrestrial vertebrate communities matched pre-extinction complexity. Marine ecosystems required 50 million years to rebuild the intricate trophic structures and specialized gills that characterized Permian oceans. Half of 100 million years. And even then, what emerged wasn't restoration of what was lost. It was something fundamentally different. New dominant groups. New ecosystem architectures. New rules for survival. The Paleozoic era, that 290 million year span from the Cambrian explosion to the end Permian catastrophe, represents one complete cycle of life's experimentation on Earth. It began with the sudden appearance of complex body plans in hard-shelled organisms. It developed through the colonization of land, the rise of forests, the evolution of flight in insects, the diversification of fish, the emergence of tetrapods. It peaked with intricate ecosystems on every continent and in every ocean, billions of organisms filling every available niche. Then it ended. Almost completely. What survived wasn't the strongest or most advanced. Lystrosaurus wasn't superior to the synapsids that vanished. Small burrowing reptiles weren't more evolved than their extinct relatives. Marine bivalves that spread globally after the extinction weren't better designed than the diverse communities they replaced. Survivors possess traits that happen to match an unpredictable catastrophe. Small size. Low energy needs. Flexible diet. Ability to tolerate unstable conditions. These weren't advantages in the stable Permian world. They became advantages only when that world burned. This reveals something crucial about extinction and survival. Success during stable times doesn't predict success during crisis. Specialists dominate when conditions remain constant. Generalists persist when everything changes. The Permian-Triassic boundary rewrote Earth's ecological rules in real time, and only organisms capable of abandoning old strategies survived to write new ones. Today, we're running a similar experiment. Atmospheric CO2 is rising at rates comparable to the end Permian, perhaps faster. Global temperatures are increasing. Ocean pH is dropping. Species are disappearing at hundreds of times the background extinction rate. We haven't reached Permian-Triassic severity, not yet, but the mechanisms look disturbingly familiar. Rapid carbon release overwhelming natural buffers. Warming triggering positive feedbacks. Ecosystems stressed beyond their adaptive capacity. The difference is agency. The Siberian traps didn't choose to erupt. Continental drift didn't decide to form Pangaea. Geological processes operated according to physics and chemistry, indifferent to biological consequences. We have choice. We understand what's happening. We can see the trajectory. Whether we change course or accelerate toward our own boundary event remains an open question. The Paleozoic era ended in the greatest catastrophe complex life has faced. What followed was millions of years of struggle, experimentation, failure, and eventual success. Life continued because a few organisms, through luck and circumstance, carried the genetic potential for recovery. Their descendants rebuilt the world, but it took longer than our species has existed. Longer than our genus has existed. Longer than our entire evolutionary family has existed. That's the timescale of true recovery from severe mass extinction. Not years or centuries. Millions of years. And what emerges looks nothing like what was lost. The Mesozoic world of dinosaurs bore no resemblance to the Permian world of synapsids. The Cenozoic world of mammals shares little with either. Each era represents a fundamentally different experiment in how to organize life on a planet. We're living in the aftermath of the last major extinction, the one that ended the dinosaurs 66 million years ago. Our existence depends on that catastrophe. Mammals remained small and marginal for 160 million years while dinosaurs dominated. Only after the end Cretaceous extinction did our ancestors diversify, grow large, explore new ecological roles. Every human alive today descended from survivors of that event. Small, burrowing, generalist mammals that persisted when 75% of species could not. The Permian-Triassic extinction reminds us that survival isn't guaranteed. Dominance offers no protection. Complexity provides no immunity. When environmental conditions shift beyond tolerable limits, evolution's timescale becomes irrelevant. Adapt instantly or die. Most die. The few that survive do so not through superiority, but through possessing traits that happen to match unpredictable new conditions. Trilobites survived 4 previous mass extinctions across 250 million years. The end Permian erased them. Tabulate and rugose corals built reefs for hundreds of millions of years. They vanished completely. Glossopterous forests covered entire continents. They collapsed in hundreds of thousands of years. Success across deep time means nothing when the planet itself transforms. So what does this mean for us? We're conducting real-time experiments in atmospheric chemistry, ocean acidification, and rapid climate change. We're removing species faster than they can evolve. We're simplifying ecosystems that took millions of years to achieve current complexity. We're testing whether human civilization can survive conditions we're creating. The Permian-Triassic extinction suggests that complex, specialized systems are vulnerable. That rapid environmental change overwhelms adaptive capacity. That recovery takes timescales measured in geological eras, not human lifetimes. But it also demonstrates that life persists. Not all life. Not familiar life. But life in some form continues. The question isn't whether Earth will survive our impacts. It will. The question is whether we'll be part of what survives, or whether we'll join the catalog of extinct experiments. Whether humans in the far future will study our fossil record and wonder what went wrong. Whether some post-human intelligence will analyze the sharp boundary in rock layers marking the Anthropocene extinction and calculate that 90% of large mammals vanished in less than 100,000 years. The Paleozoic era began with explosion and ended with annihilation. It contained 290 million years of evolutionary innovation, ecological complexity, and biological success. None of it mattered when conditions exceeded tolerance limits. What mattered was which random organisms possessed traits allowing persistence through conditions nobody could have predicted. The survivors weren't chosen by merit. They were filtered by catastrophe. We had the knowledge to avoid becoming another extinction boundary. We understand the mechanisms. We can model the outcomes. We see the trajectory. The Permian-Triassic extinction wasn't preventable. It resulted from geological processes beyond any organism's control. Our current trajectory is different. It's driven by choices, by economic systems, by political decisions, by collective action or inaction. That means it's changeable. But only if change occurs fast enough to matter. The timescale is what makes this urgent. Geological processes operate slowly. Evolution operates slowly. Recovery operates slowly. But extinction, when it comes, happens fast. The end Permian proved that. 60,000 years from stable ecosystems to near-complete collapse. On human timescales, that's incomprehensibly long. On geological timescales, it's instantaneous. We're currently operating on timescales that match extinction, not recovery. Carbon release rates approaching volcanic eruption events. Temperature change rates exceeding most of the past 66 million years. Extinction rates hundreds of times above background. The Paleozoic era teaches us that planets can recover from almost anything. But recovery looks nothing like restoration. It's transformation. The world moves forward, permanently altered, carrying scars visible for hundreds of millions of years. Life continues, but not the same life. Ecosystems function, but not the same ecosystems. The survivors build new worlds from the ruins of old ones. And those new worlds, given enough time, develop their own complexity, their own diversity, their own intricate balance. Until the next catastrophe resets everything again. That's the pattern. Stability, catastrophe, recovery, stability, catastrophe, recovery. Earth's biological history is written in these cycles. The Permian-Triassic boundary marks the most extreme transition, the closest approach to total failure. It stands as evidence that even 290 million years of evolutionary success offers no guarantee of continued existence. That life is simultaneously resilient enough to survive near-complete destruction and fragile enough to be pushed to that brink. That the difference between survival and extinction often comes down to chance, to possessing the right traits at the right moment, to being small enough, flexible enough, efficient enough to persist when everything else fails. Every living organism today carries the legacy of Permian-Triassic survivors in its DNA. Every ecosystem functions according to rules established during the long recovery. Every species alive is here because some unknown ancestor, probably small and unremarkable, made it through Earth's worst day. We're all products of that catastrophe, descendants of disaster taxa that inherited an empty planet and slowly, over millions of years, filled it with new forms of life. The question now is whether we'll be ancestors or fossils. Whether future organisms will carry our legacy forward or study our remains. Whether we'll navigate the crisis we're creating or become another cautionary tale preserved in sedimentary layers. The Paleozoic era ended in fire and suffocation. The Mesozoic era ended with asteroid impact. The Cenozoic era's ending hasn't been written yet. We're writing it now, in real time, with every choice about energy systems, land use, ocean health, atmospheric composition. The Permian-Triassic extinction shows us what's possible when planetary systems break. Whether we heed that warning determines what comes next. Something's out there. Something massive, something cold, something so distant it takes 10,000 to 20,000 years to complete a single orbit around our sun. We haven't seen it. We can't photograph it. Yet the mathematical evidence suggests it's real, and it's 10 times more massive than Earth. This is Planet 9, a world that shouldn't exist according to everything we thought we knew about our solar system. But the math keeps pointing to the same conclusion. There's a giant lurking in the darkness beyond Neptune. In 2016, two astronomers at Caltech made an announcement that sent shockwaves through the scientific community. Konstantin Batygin and Mike Brown claimed they'd found evidence for a ninth planet, not through direct observation, but through pure mathematics and computer simulations. The prediction was bold. The implications were staggering. And the astronomical world split into believers and skeptics almost immediately. Here's what makes this different from every other "maybe there's something out there" claim. The evidence doesn't come from a single strange observation or a blurry photograph. It comes from watching how dozens of distant objects move, objects so far away they barely reflect any sunlight, objects that take thousands of years to complete their journeys around the sun. These frozen worlds are telling us something. Their orbits are aligned in ways that shouldn't be possible unless something massive is pulling on them. 6 of the most distant objects we've ever discovered all point in the same direction. They tilt downward from the plane of our solar system at nearly identical angles. The odds of this happening by random chance? About 1 in 500. Those aren't lottery odds. Those are "something's definitely going on here" odds. But let's back up. How do you lose a planet? The story begins much earlier than 2016. It starts over a century ago with an astronomer named Percival Lowell. In 1915, Lowell became convinced that something was disturbing the orbit of Uranus. He called this hypothetical object Planet X, where X stood for the unknown, not for the Roman numeral 10. Lowell died in 1916, never finding his mysterious planet. But his search continued at the observatory he founded in Flagstaff, Arizona. In 1930, a young astronomer named Clyde Tombaugh discovered Pluto. For decades, astronomers believed they'd found Lowell's Planet X. Pluto was celebrated as the 9th planet, the final member of our solar system family. Except there was a problem. Pluto was far too small to cause the gravitational effects Lowell had predicted. It turned out those perturbations to Uranus's orbit were measurement errors. They weren't real. The search for Planet X was based on a mistake. Pluto's story took another turn in 2006. The International Astronomical Union redefined what it means to be a planet. Pluto didn't make the cut. It became a dwarf planet instead, one of many similar objects in the Kuiper belt. the man who provided much of the evidence leading to Pluto's demotion, Mike Brown. He even wrote a book titled, How I Killed Pluto and Why It Had It Coming. The irony that Brown might now discover a true ninth planet hasn't been lost on anyone. So if the old Planet X was a myth based on bad data, why should we believe in this new Planet 9? Because the evidence is completely different. This isn't about measurement errors or wishful thinking. It's about patterns that emerge when you look at the most distant objects in our solar system, objects that exist in a realm we barely understand. The outer solar system is strange. Beyond Neptune, at distances greater than 250 astronomical units, that's 250 times farther from the Sun than Earth, you enter a region where sunlight is so faint it's almost meaningless. Out here, objects move in slow motion, taking millennia to complete their orbits. We've discovered dozens of these extreme trans-Neptunian objects, and they're revealing something unexpected. In 2014, astronomers Scott Shepard and Chadwick Trujillo noticed something odd. A handful of these distant objects had orbits that clustered together. They weren't randomly scattered across the sky like you'd expect. They were grouped, almost shepherded, as if something massive was organizing them. Shepard and Trujillo suggested an unseen planet might be responsible, something several times larger than Earth and more than 200 times our planet's distance from the Sun. Badigian and Brown heard about this clustering and decided to investigate. What they found went far beyond the initial observations. When they ran computer simulations of the outer solar system, trying to recreate the orbits of these distant objects, they kept hitting the same conclusion. Without a massive planet out there, the simulations couldn't reproduce what we actually observe. But add a planet with specific characteristics, about 10 Earth masses in a highly elongated orbit, and suddenly everything clicked into place. The simulations matched reality. The alignment of these objects isn't subtle. 6 of them point in the same direction when they make their closest approach to the Sun. They're tilted at about 30 degrees downward from the plane where the other planets orbit. This kind of alignment doesn't happen naturally in a chaotic system. Something organized it. Something massive enough to gravitationally dominate a region of space larger than any known planet. There's more. Badigian and Brown simulations predicted that a second group of objects should exist, forced into positions at right angles to Planet 9 and into orbits perpendicular to the solar system's plane. 5 known objects fit this prediction exactly. The math predicted they should be there before anyone found them. In April 2024, Badigian and Brown published new evidence. They focused on a different population of trans-Neptunian objects, ones with orbits that reach inside Neptune's orbit. These objects spread inward from Neptune in a pattern that matches Planet 9's predicted gravitational influence precisely. Without Planet 9 in the simulations, this pattern doesn't appear. With it, the match is almost perfect. The study was statistically significant at about a 5 sigma confidence level, which in scientific terms means the probability of this being a coincidence is vanishingly small. Mike Brown stated it plainly. I think it is very unlikely that Planet 9 does not exist. There are currently no other explanations for the effects that we see. If Planet 9 isn't real, astronomers would need 5 or 6 separate unknown explanations for all the oddities they're observing in the outer solar system. That's possible, but it violates a principle scientists hold dear. The simplest explanation that accounts for all the evidence is usually correct. But here's what makes this search so difficult. Planet 9, if it exists, is unimaginably far away. The most recent estimates place it around 290 astronomical units from the Sun on average. To put that in perspective, Neptune orbits at about 30 astronomical units. Pluto's average distance is around 40 astronomical units. Planet 9 is 7 to 10 times farther out than that. At such distances, sunlight is incredibly weak. The planet would reflect almost no visible light. It would be extraordinarily dim, with an apparent magnitude somewhere around 21 or 22. That's roughly 25,000 times fainter than the faintest stars you can see with your naked eye. Even with the world's most powerful telescopes, finding Planet 9 is like searching for a specific grain of sand on a beach at midnight. You know roughly which beach to search, but the grain could be anywhere along miles of coastline. Previous searches have eliminated about 78% of the possible sky positions. That sounds impressive until you realize 22% of an already enormous search area is still enormous. The planet moves slowly, incredibly slowly. At its estimated distance, it would take between 10,000 and 20,000 years to complete one orbit. In the time since humans built the pyramids, Planet 9 has moved only a fraction of its total path. This makes it harder to detect because astronomers typically find distant objects by watching them move against the background stars. If something barely moves over months or years of observation, it can easily be mistaken for a distant star or galaxy. There's another complication. The planet's orbit is highly elongated. Sometimes it might be relatively closer to the Sun, perhaps 200 to 300 astronomical units away. At other times, it could be 600 astronomical units out or even farther. According to Kepler's laws of planetary motion, objects move more slowly when they're farther from the Sun. Statistically, Planet 9 is more likely to be near the far end of its orbit right now, where it's moving slowest and is dimmest. That's the worst possible scenario for detection. Yet despite these challenges, astronomers are confident they'll find it soon. The reason for this confidence lies not in traditional telescopes, but in a revolutionary new observatory that's about to begin operations. The Vera C. Rubin Observatory in Chile represents a complete game-changer for astronomy. This facility, a joint project of the National Science Foundation and the Department of Energy, has been under construction for years. It's equipped with the largest digital camera ever built for astronomy, and it's designed to do something no other telescope can match. Starting in 2025, it will photograph nearly the entire southern sky every few nights, creating an ongoing movie of the cosmos. Mike Brown calls it the perfect tool for finding Planet 9. If you were to hand me a big wad of cash and say, go build a telescope to either find this Planet 9 or find the best evidence possible for Planet 9, I would probably go and build the Vera Rubin Observatory. The observatory will capture images of roughly 10 billion objects over the next decade. Computer systems will constantly compare new images to previous ones, flagging anything that changes or moves. If Planet 9 is in the southern sky, Rubin will almost certainly detect it. Scott Shepard estimates the observatory has a 70 to 80% chance of finding the planet if it's real. But what if it's not in the southern sky? What if it's in the northern hemisphere, outside Rubin's view? That's where other telescopes come in. The Subaru telescope in Hawaii, with its combination of large aperture and wide field of view, is conducting its own search. Two teams of astronomers, Badijan and Brown along with Trujillo and Shepard, are working together, expecting the combined search to take up to 5 years. 5 years might seem like a long time, but in astronomical terms, it's remarkably quick. We're talking about finding an object that's been hiding for 4.6 billion years, an object that's never been seen by human eyes, an object that challenges everything we thought we understood about how our solar system formed. Because that's the real question lurking behind all of this. If Planet 9 exists, how did it get there? A planet that massive, that far from the Sun, shouldn't exist according to standard models of planetary formation. The solar system's outer regions didn't have enough material to build something that large at such distances. So where did it come from? The answer might be even stranger than the planet itself. The leading theory suggests our solar system once had 5 giant planets, not 4. Jupiter, Saturn, Uranus, Neptune, and a 5th world roughly the size of Uranus or Neptune. During the chaotic early days of the solar system, these giants were packed much closer together than they are now. They orbited in a dense disk of planetesimals, the leftover building blocks from planetary formation. Then, over millions of years, gravitational interactions between the giant planets and these smaller objects caused the planets to migrate. Jupiter moved slightly inward. The others drifted outward. This migration wasn't smooth or gentle. It was violent, chaotic, a cosmic game of pinball played with planet-sized objects. At some point during this upheaval, the 5th giant planet got too close to Jupiter. Jupiter, the most massive planet in the solar system with more than twice the mass of all other planets combined, acted like a gravitational slingshot. It flung the 5th giant outward with tremendous force. Most computer simulations of this scenario show the ejected planet being thrown completely out of the solar system, lost forever to interstellar space. But in a small percentage of simulations, something different happens. The ejected planet doesn't quite reach escape velocity. Instead, it settles into a distant, highly elliptical orbit, exactly the kind of orbit predicted for Planet 9. This scenario is called the Nice model, named after the French city where it was developed. The Nice model has become widely accepted because it explains several puzzling features of the solar system. It accounts for the late heavy bombardment, a period roughly 4 billion years ago when the inner solar system was pummeled by asteroids and comets. It explains why Neptune's orbit is slightly eccentric. It clarifies the structure of the Kuiper belt. And now, it might explain Planet 9. The ejection hypothesis solves a fundamental problem. You can't build a planet as massive as Planet 9 at such extreme distances from the Sun. There simply wasn't enough material out there. The solar nebula, the disk of gas and dust from which planets formed, was too thin and spread out at those distances. Planet formation requires material to collide and stick together, and that process becomes exponentially slower when particles are spread across vast expanses of space. But if Planet 9 formed closer to the Sun, in the region where Uranus and Neptune formed, and was then ejected outward, suddenly its existence makes perfect sense. The physical characteristics match this origin story remarkably well. Planet 9 is predicted to have a mass of about 5 Earth masses, similar to Uranus and Neptune when you account for their thick atmospheres. It would be gaseous, an ice giant with a rocky core surrounded by water, methane, and ammonia ices, all wrapped in a hydrogen and helium envelope. These are exactly the characteristics you'd expect from a planet that formed in the outer solar system 6 billion years ago. But there's another possibility, one that's even more extraordinary. What if Planet 9 didn't form in our solar system at all? The Sun wasn't born in isolation. It formed in a stellar nursery, a dense cluster containing thousands of young stars packed into a relatively small volume of space. In such environments, close encounters between stars are common. During the first few 100 million years of the solar system's existence, another star could have passed within a few hundred astronomical units of the Sun. If that star had its own planetary system, and if one of its planets happened to be in just the right position during the encounter, the Sun's gravity could have captured it. This capture scenario would make Planet 9 an exoplanet, a planet that formed around another star. If confirmed, it would be the first exoplanet discovered within our own solar system, a world that spent billions of years orbiting a different sun before becoming part of ours. The odds of such a capture are low, but they're not zero. Computer simulations show that in dense stellar nurseries, planetary capture events do occur, though rarely. The capture hypothesis has an appealing elegance. It would explain why Planet 9's orbit is so different from the other planets, tilted relative to the solar system's plane and highly elongated. A captured planet wouldn't follow the orderly, nearly circular orbits of planets that formed in place. Its orbit would bear the fingerprints of a violent gravitational encounter, exactly what we see predicted for Planet 9. But here's what makes this puzzle even more complex. There's a third option that astronomers are seriously considering. What if there isn't just one Planet 9, but multiple smaller planets? What if instead of a single 5 Earth mass world, there are 2 or 3 planets, each a few Earth masses, scattered across the outer solar system? This multi-planet scenario could produce similar gravitational effects on trans-Neptunian objects while being harder to detect because each individual planet would be smaller and dimmer. Some researchers have proposed an even more exotic alternative involving a disk of small icy bodies with a combined mass of 10 Earth masses, spread across the outer solar system. This disk, if it exists, could gravitationally shepherd trans-Neptunian objects in ways that mimic the effects of a single large planet. The challenge with this disk hypothesis is that such a structure should be detectable through its infrared emission, and so far, surveys haven't found it. Then there are the truly speculative ideas. Could Planet 9 be a primordial black hole, a microscopic black hole formed in the early universe? Such an object would have the right mass to produce the observed gravitational effects but would be nearly impossible to detect directly. Or could it be a concentration of dark matter, the mysterious substance that makes up most of the universe's mass but doesn't interact with light? These exotic explanations remain on the fringes of scientific consideration because they require invoking phenomena we've never directly observed in our solar system. Occam's razor suggests the simpler explanation, a planet, is more likely. But the fact that astronomers are even considering such alternatives demonstrates how profoundly puzzling this situation is. What makes the formation question so crucial is this: the answer would tell us whether our solar system is typical or unusual. If Planet 9 is an ejected fifth giant, it suggests that planetary systems commonly lose planets during their chaotic youth. Astronomers have found evidence of ejected planets around other stars, so this wouldn't be unprecedented. It would mean that for every planetary system we observe, there might be one or more invisible planets lurking in the darkness beyond what we can easily detect. If Planet 9 is a captured exoplanet, it would suggest that stellar nurseries are dynamic enough to exchange planets between forming systems. This would have profound implications for understanding planetary demographics in the galaxy. How many stars captured planets? How many lost them? The architecture of planetary systems might be far more fluid and dynamic than we've imagined. Recent refinements to the Planet 9 hypothesis have actually made the formation question more pressing. A 2025 study using an expanded sample of 51 extreme trans-Neptunian objects proposed that Planet 9's orbit might be smaller than previously thought, with a semi-major axis of around 290 astronomical units rather than 400 to 600. If true, this would place Planet 9 closer to the Sun, making it brighter and easier to find, but it also changes the dynamical requirements for how the planet reached that orbit. The nice model simulations that showed a fifth giant being ejected to Planet 9's orbit were based on the earlier, larger orbital estimates. With a smaller orbit, the ejection dynamics would need to be recalculated. Could Jupiter fling a planet to 290 astronomical units without ejecting it entirely from the solar system? The answer depends on precise details of the ejection event, the angle, the velocity, the gravitational assists from other planets. What we're witnessing is science in action, the process of refining hypotheses as new data emerges. Each new trans-Neptunian object discovered adds another constraint to the models. Each failed search narrows the possible locations. The picture is becoming clearer, but the fundamental mystery remains. Somewhere out there, moving slowly through the darkness, there might be a world that holds the key to understanding our solar system's violent past. And the clock is ticking. Within the next few years, the Vera Rubin Observatory will either find Planet 9 or rule out large portions of the remaining search space. We're approaching a definitive answer to one of astronomy's biggest questions. But before we get there, we need to understand what finding Planet 9 would actually mean for the rest of the solar system, because its gravitational influence extends far beyond just those handful of trans-Neptunian objects. Planet 9's gravitational reach doesn't stop at those extreme trans-Neptunian objects. It extends throughout the outer solar system in ways that are subtle but detectable. Jupiter family comets, those periodic visitors that swing through the inner solar system every few years, would show different orbital tilts if Planet 9 exists. Computer simulations predict their inclination distribution should be broader, more spread out, than what we'd see in a Planet 9-free solar system. And when astronomers look at the actual data, that's exactly what they find. The distribution matches the prediction. Then there's the Oort cloud, that vast spherical shell of icy bodies surrounding the solar system at distances up to 100,000 astronomical units. Planet 9 would have affected how many objects got captured into the Oort cloud during the solar system's formation. Simulations show that with Planet 9 in its predicted orbit billions of years ago, fewer objects would have been flung into the Oort cloud. The gravitational stirring from a massive planet in the outer reaches would have ejected more objects entirely from the solar system rather than parking them in distant orbits. This prediction is harder to test directly since we can't count Oort cloud objects with any precision, but it represents another testable consequence of the hypothesis. What about the giant planets themselves? Would Planet 9 have left fingerprints on Jupiter, Saturn, Uranus, and Neptune? The answer depends on when Planet 9 reached its current orbit. If it was ejected early in the solar system's history, during the period of planetary migration described by the NICE model, then its ejection would have been part of the same chaotic reshuffling that moved all the giant planets. In that case, Planet 9's influence is already baked into the current orbital configuration we observe. The planets are where they are partly because of the gravitational chaos that sent Planet 9 outward. But there's a more direct effect. Planet 9's orbit is tilted about 6 degrees relative to the plane where the 8 known planets orbit. That tilt would exert a slow, subtle torque on the entire solar system over billions of years. Think of it like a spinning top that's slightly off balance. Over time, that imbalance affects the whole system's orientation in space. Calculations suggest that Planet 9 could explain a long-standing puzzle: why the Sun's equator is tilted about 6 degrees relative to the planetary orbital plane. The gravitational influence of a distant, tilted Planet 9, acting over 4.5 billion years, could have gradually torqued the entire inner solar system, including the Sun's rotation axis, into its current configuration. This prediction caught many astronomers by surprise when it was first published. It's one thing to affect distant icy bodies. It's another to tilt the Sun itself. But the math checks out. A 5 Earth-mass planet at 300 to 400 astronomical units, tilted at the right angle, applying gentle but persistent gravitational torque over geological timescales, could absolutely accomplish this feat. And suddenly Planet 9 isn't just explaining weird orbits at the edge of the solar system. It's explaining a fundamental property of our planetary system that's been visible since humans first pointed telescopes at the Sun. Yet another population shows Planet 9's fingerprints: high-inclination trans-Neptunian objects. These are objects whose orbits are tilted steeply relative to the plane where planets orbit, sometimes by 90 degrees or more. They're rare, which makes sense. Most of the solar system's material started in a flat disk and should stay relatively flat. But we found several objects with these extreme tilts, and they need an explanation. Planet 9's gravitational influence could pump up the inclinations of certain objects over millions of years through a process called causative oscillations. This mechanism causes orbits to trade eccentricity for inclination in a periodic cycle. An object might start with a moderately elliptical orbit in the plane, then gradually become more circular but more tilted, then swing back. With Planet 9's gravity driving these oscillations, some objects could get kicked into very high inclinations and stay there. 5 known objects fit this prediction with remarkable precision. They're positioned at roughly 90-degree angles to Planet 9's predicted location, and their orbits are perpendicular to the solar system's plane. When Badigian and Brown first ran their simulations, these objects appeared naturally in the results. The model predicted they should exist before anyone had specifically looked for them. Finding them in the real data wasn't just confirmation. It was the kind of successful prediction that makes scientists take notice. The gravitational mechanisms at work here operate on timescales that dwarf human civilization. An object in the outer solar system might take hundreds of thousands of years to complete one oscillation cycle. Planet 9 itself takes 10,000 to 20,000 years just to complete a single orbit. The gravitational dance between Planet 9 and these distant objects unfolds across geological epochs, invisible to any observer watching for less than millennia. We're catching a single frame of a movie that's been playing for billions of years. Does Planet 9 affect the known planets directly? Neptune's orbit should show tiny perturbations if Planet 9 exists, wobbles measurable in the planet's position over time. Astronomers have looked for these signatures. So far, the results are inconclusive, which actually makes sense given Planet 9's distance. At 300 to 600 astronomical units, its gravitational pull on Neptune is extraordinarily weak. Neptune is 30 astronomical units from the Sun. Planet 9 is 10 to 20 times farther out. Gravitational force drops off with the square of distance, so Planet 9's pull on Neptune is hundreds of times weaker than Neptune's pull on itself. Detecting such subtle effects requires decades of precise observations and sophisticated data analysis. We might not see clear perturbations in Neptune's orbit for years yet. Uranus presents a different puzzle. This ice giant is tilted on its side, rotating with its axis nearly in the plane of its orbit rather than roughly perpendicular like every other planet. Various theories have tried to explain this bizarre orientation: a giant collision during formation, gravitational interactions during planetary migration, or cumulative effects from multiple smaller impacts. Could Planet 9 be responsible? Probably not directly. The tilt likely originated during the solar system's chaotic youth, and Planet 9's current location doesn't provide the right geometry to have caused it. But if Planet 9 was ejected from the inner solar system, it might have been one of several factors contributing to the orbital chaos that tilted Uranus. Not the sole cause, but part of a complex sequence of gravitational interactions. The asteroid belt between Mars and Jupiter seems far removed from Planet 9's domain, but even here there might be subtle connections. Some asteroids have been found on highly elliptical orbits that take them far beyond Neptune. These aren't main belt asteroids in the traditional sense, they're trans-Neptunian objects that happen to pass through the inner solar system. Planet 9 could influence the population of these objects, determining which ones get perturbed onto planet-crossing orbits and which stay safely beyond Neptune. This matters for understanding impact rates on the inner planets over geological time. If Planet 9 exists, it's been shaping the flux of impactors throughout solar system history. Centaurs, those hybrid objects orbiting between Jupiter and Neptune, neither quite asteroids nor comets, show dynamical properties that could reflect Planet 9's influence. These objects are thought to originate from the trans-Neptunian region and get gravitationally scattered inward. The rate at which objects get scattered, and the specific orbits they end up on, depend on the outer solar system's gravitational environment. Add Planet 9 to the mix, and you change the probabilities. More objects might get kicked onto certain types of orbits, fewer onto others. Detailed population studies of centaurs could, in principle, reveal Planet 9's gravitational fingerprint even in the middle reaches of the solar system. What's remarkable about all these effects is their consistency. Planet 9 isn't just explaining one weird thing. It's explaining multiple independent phenomena across different populations of objects in different regions of the solar system. The clustering of extreme trans-Neptunian objects. The perpendicular objects. The sun's axial tilt. The distribution of orbital inclinations. Each one could potentially have a different explanation. But they all point to the same solution, a massive planet on a distant, tilted orbit. That's what makes the hypothesis so compelling. Still, correlation isn't causation. Maybe these effects have other explanations we haven't considered. Maybe observational biases we haven't properly accounted for are creating patterns where none exist. Maybe the solar system is just weird in multiple unrelated ways. These possibilities keep skeptics skeptical, and rightfully so. Extraordinary claims require extraordinary evidence. But with each new study, with each additional prediction that proves correct, the case grows stronger. The question isn't really whether Planet 9 affects the solar system. If it exists at all, it must. Gravity doesn't have an off switch. The question is whether we can measure those effects precisely enough to confirm the planet's existence indirectly. And that's where the cutting edge of observational astronomy comes in, techniques for detecting planets not by seeing them directly, but by mapping their gravitational influence on everything around them. The art of finding invisible planets has evolved dramatically since Neptune's mathematical discovery in 1846. Back then, astronomers noticed Uranus wasn't following its predicted path. Something was tugging on it. They worked backward from those perturbations, calculated where the disturbing mass had to be, pointed their telescopes at that spot, and there it was. A new world revealed through pure mathematics. Planet 9 requires a more sophisticated approach. We're not tracking one object getting pulled by an unknown planet. We're analyzing dozens of distant bodies, each with its own orbital peculiarities, trying to extract a common signal from the noise. It's less like following footprints to find what made them and more like being a detective at a crime scene with multiple witnesses who each saw something slightly different. The statistical methods matter enormously here. Brown and Badijan didn't just notice the clustering and declare victory. They quantified it. They developed techniques to measure observational bias in each individual object's discovery, because not all parts of the sky get observed equally. Some regions get scanned more frequently. Some get deeper exposures. Telescopes point where astronomers tell them to point, and astronomers have their own interests and observing programs. This creates selection effects. If you only search for distant objects in certain parts of the sky, you'll only find distant objects in those parts of the sky. Obvious, right? But correcting for this bias mathematically isn't straightforward. You need to know not just where you looked, but how thoroughly you looked there, and with what sensitivity. Brown and Badijan accounted for these factors, and even after corrections, the clustering remained statistically significant. About 1 chance in 500 that it's random. That number has gotten better with time. As more objects get discovered and the sample size grows, the statistical confidence strengthens. By 2021, Brown and Badijan reanalyzed with improved bias corrections and found a clustering significant at 99.6% confidence. That's getting into the realm where you start taking things very seriously. But statistics alone don't make a planet. The real power comes from prediction. If your hypothesis is correct, it should tell you what else you'll find. Planet 9 makes specific predictions about populations of objects that should exist but hadn't been looked for yet. And that's where things get interesting. One prediction involves objects perpendicular to the solar system's plane. Not just tilted. Perpendicular. Orbiting the sun almost vertically while everything else goes around horizontally. These objects shouldn't exist in a normal solar system. There's no mechanism to put them there. Yet Planet 9's gravitational influence, according to the models, should create exactly this population. 5 objects matching this prediction have been found. 5 might not sound like many, but remember, these are extremely rare configurations. Finding even one was surprising. Finding 5 that matched the theoretical prediction in detail. That's not coincidence. Another test came in 2024. Brown and Badijan published analysis of a different population, low-inclination trans-Neptunian objects with orbits that reach inside Neptune's orbit. These objects have nothing to do with the original clustering that suggested Planet 9. They're on completely different types of orbits. Yet when you run simulations with Planet 9 included, these objects' orbital distribution matches what we actually observe. Remove Planet 9 from the simulations, and the match disappears. The statistical rejection of the Planet 9 free scenario sits at 5 sigma. That's the same confidence level physicists demanded before announcing the Higgs boson discovery. Does this prove Planet 9 exists? No. Science doesn't deal in proof, only in evidence and confidence levels. But the confidence is getting high enough that dismissing the hypothesis requires explaining away multiple independent lines of evidence. The skeptics haven't been silent. Samantha Lawler and others argue that observational biases are stronger than Brown and Badijan acknowledge. In 2020, 2 large surveys, the Outer Solar System Origins Survey and the Dark Energy Survey, found hundreds of trans-Neptunian objects and saw no significant clustering. OSOS documented over 800 objects. DES found 316 new ones. Both surveys carefully accounted for observational bias and concluded the orbits could be explained by known physical processes without invoking a 9th planet. This disagreement cuts to the heart of how science works. Same solar system, same objects, different conclusions. Why? It comes down to which objects you include in your analysis and how you correct for bias. Brown and Badijan focus on the most extreme objects, the ones farthest out with the most elongated orbits. These are the objects most likely to be influenced by Planet 9 because they venture into its gravitational domain. OSOS and DES looked at a broader population including objects that wouldn't be strongly affected by Planet 9 even if it exists. The debate also involves sample size versus sample quality. OSOS has more objects, but are they the right objects for testing this particular hypothesis? 1,000 data points don't help if they're measuring the wrong thing. Meanwhile, Brown and Badijan's smaller sample focuses on exactly the objects that should show Planet 9's influence most clearly. It's a classic tension in observational astronomy. More data isn't always better data if it dilutes your signal. Alternative explanations have emerged. Maybe instead of one big planet, there's a disk of smaller icy bodies with combined mass equivalent to Planet 9. Spread that mass out, and you might get similar gravitational effects. Or perhaps these orbits were set during the solar system's birth, when the sun formed in a stellar cluster and neighboring stars perturbed the outer reaches. Some researchers even propose multiple Earth mass rogue planets rather than one super-Earth. Each alternative has problems. The disk hypothesis requires an improbably specific mass distribution that stays stable over billions of years. The stellar cluster scenario works for setting initial conditions but doesn't explain why the clustering persists today, billions of years after the Sun left its birth cluster. Multiple robes would need to have been captured with just the right orbits and masses to produce the observed effects without disrupting everything else. What makes Planet 9 compelling isn't that it's the only possible explanation. It's that it's the simplest explanation that accounts for multiple independent observations. One object, one orbit, multiple predictions that keep being confirmed. That's powerful. The thermal emission angle offers another avenue. Planet 9, if it exists, is still cooling from its formation. At 5 Earth masses, it retains significant internal heat. Even at 47 Kelvin, frigidly cold by any human standard, it radiates in the infrared. This radiation should be detectable with the right instruments, particularly submillimeter telescopes that specialize in this wavelength range. The challenge is sensitivity. Planet 9's thermal signature is faint, and the infrared sky is crowded. Lots of things emit at these wavelengths. You need long integration times, careful subtraction of foreground and background sources, and the patience to search a large area of sky. Several groups are working on this, using facilities like the Atacama Large Millimeter Array. No detections yet, but the search continues. Direct optical detection faces a different set of challenges. At aphelion, Planet 9 could be 800 astronomical units away or more, moving through the most distant parts of its orbit. At that distance, reflected sunlight is vanishingly faint. You need large telescopes with wide fields of view, and you need to image the same regions repeatedly to detect motion. The Subaru telescope in Hawaii has been conducting systematic searches. So have other facilities. They've covered about 78% of the possible sky positions where Planet 9 might be. That sounds good until you realize the remaining 22% is still a huge search space. Statistically, Planet 9 is more likely to be near aphelion than perihelion, simply because it moves slower out there and spends more time in that part of its orbit. That's where Kepler's second law bites you. Objects sweep out equal areas in equal times, which means they crawl along when far from the Sun. If Planet 9 is currently near aphelion, it's at its faintest and hardest to detect. Bad luck for us, but that's the reality. The search isn't hopeless. It's methodical, systematic, and ongoing. Every survey that comes up empty eliminates possibilities and narrows the remaining search space. Brown and Badijan have refined their predictions multiple times based on new data, updating the most likely orbital parameters. Their current best estimate puts the semi-major axis around 380 to 460 astronomical units, though a 2025 study by Siraj, Chaiba, and Tremaine suggests it might be closer, around 290 astronomical units. Closer means brighter. Brighter means easier to find. And then there's the Vera Rubin Observatory, scheduled to begin operations in 2025, which changes everything. The Vera Rubin Observatory is unlike anything that's come before. Sitting on Cerro Pachón in Chile at 2,700 meters elevation, it houses an 8.4-meter primary mirror and the largest digital camera ever built for astronomy. 3 gigapixels. That's not a typo. The camera weighs 3 tons and captures an area of sky equivalent to 40 full moons in a single image. Why does this matter for Planet 9? 2 reasons. Coverage and cadence. Rubin will survey the entire visible southern sky every few nights. Not targeted observations of specific regions. The whole sky, repeatedly, for 10 years. Every object in that survey gets tracked automatically. The system compares new images to previous ones, flagging anything that moves, changes brightness, or appears where nothing was before. Planet 9, if it exists, moves slowly. Over months and years, it would shift position relative to background stars. That's its signature. Rubin's repeated imaging strategy is custom-built to catch exactly that kind of motion. You don't need to know where to look ahead of time. You just watch everything and let the data tell you what moved. Mike Brown's enthusiasm about Rubin isn't exaggeration. He spent years searching with Subaru, which is an excellent telescope but requires choosing where to point. Rubin doesn't. It watches. Constantly. Systematically. If Planet 9 is anywhere in the southern sky at a detectable brightness, Rubin will find it. Not might find it. Will find it. Scott Shepard estimates the probability at 70 to 80%, assuming Planet 9 exists and has the predicted properties. Those are good odds in astronomy. Mario Jurek points out that Rubin will increase the known population of trans-Neptunian objects from around 4,000 to potentially 40,000. That's not just more objects. That's better statistics, tighter constraints, and a far clearer picture of what's actually happening in the outer solar system. The timeline is aggressive. Megan Schwam says the first year or two of Rubin operations should answer the question. Either Planet 9 shows up in the data, or the lack of detection tells us something definitive about where it isn't and what it can't be. This is the moment. After nearly a decade of predictions, simulations, and targeted searches, we're about to conduct the most comprehensive survey of the outer solar system ever attempted. If Planet 9 is real, 2025 through 2027 could be the years we finally see it. But what if Rubin doesn't find it? That's the scenario nobody talks about much, but it's worth considering. Absence of evidence isn't evidence of absence, except when you've looked everywhere the thing could possibly be. Rubin gets us close to that threshold. If Rubin completes its survey without detecting Planet 9, the hypothesis doesn't collapse entirely, but it gets significantly harder to maintain. Maybe Planet 9 is fainter than expected. Maybe it's in a part of the sky Rubin can't see as well. Maybe it's currently too close to the galactic plane where background star density makes detection difficult. There are outs. But they start sounding like special pleading after a while. The other possibility is that Planet 9 exists but isn't where the models predict. Orbital parameters have shifted multiple times as new data comes in. The 2025 Siraj study suggested a closer, less eccentric orbit than previous estimates. If that's correct, Planet 9 might be brighter and easier to find. But if the models are still off, we might be looking in the wrong regions entirely. Then there's the alternative explanations. What if the orbital clustering of trans-Neptunian objects isn't caused by a single planet but by a disk of smaller objects, each too faint to detect individually but collectively massive enough to produce gravitational effects? That hypothesis has been proposed. It works mathematically for some observations but struggles to explain others, particularly the perpendicular objects. Or what if the clustering is a transient phenomenon caused by something that's no longer there? A passing star could have perturbed orbits temporarily. The Sun's birth cluster could have left a dynamical imprint that's still visible today. Multiple captured rogue planets might produce similar effects without requiring a single massive world. These scenarios feel more contrived than a single planet, but they're not impossible. Brown has argued that rejecting Planet 9 means accepting 5 or 6 separate unexplained phenomena, each requiring its own cause. That's inelegant. Science favors parsimony. One explanation that accounts for multiple observations beats several disconnected explanations. But parsimony isn't proof. Sometimes reality is messy and complicated. The discovery of Ammonite, a new sednoid announced by the Subaru team, adds another wrinkle. Ammonite's orbit doesn't cluster with the other sednoids the way Planet 9's gravitational influence would predict. It's off on its own. That might mean Planet 9's orbit needs further adjustment to account for Ammonite. Or it might mean something else entirely is going on. Every new discovery refines the picture, but it also introduces new constraints that any hypothesis has to satisfy. That's how science works. You don't get to cherry-pick the observations that support your model and ignore the rest. You have to explain all of it. Brown and Badijan have been transparent about this process. They've updated their predictions multiple times based on new data. They've addressed criticisms. They've acknowledged uncertainties. That's not weakness. That's how you build a robust scientific case. You test it against everything, adjust when necessary, and see if it still holds up. The April 2024 paper on low-inclination trans-Neptunian objects is a good example. These are objects that cross Neptune's orbit, a completely different population from the extreme distant objects that sparked the original hypothesis. Yet when you model their distribution, Planet 9's gravitational influence predicts their observed spread with remarkable accuracy. No Planet 9. The distribution doesn't match. With Planet 9, it does. That's independent confirmation using a different dataset. Statistical confidence levels matter here. A 5 sigma result means the probability of the observation occurring by chance is less than 1 in 3. 5 million. That's the standard for discovery in particle physics. It's extremely stringent. The 2024 study rejected the no Planet 9 scenario at approximately 5 sigma confidence. That's not suggestive. That's strong. Brown's statement that he thinks it's very unlikely Planet 9 doesn't exist reflects this accumulating evidence. It's not arrogance. It's Bayesian updating. Start with a hypothesis. Test it against observations. Adjust your confidence based on how well it performs. After 8 years of testing, the hypothesis keeps working. That shifts the probability upward. Could everyone be wrong? Of course. Science has a long history of confident predictions that turned out to be false. Vulcan, the hypothetical planet inside Mercury's orbit, was predicted to explain perturbations that Einstein's general relativity later explained without needing an extra planet. The luminiferous ether seemed necessary until it wasn't. Confidence doesn't equal certainty. But here's the key difference. Vulcan was never observed. Planet 9 hasn't been observed either, but the effects attributed to it keep showing up in independent datasets. The orbital clustering persists after correcting for observational bias. The perpendicular objects exist. The low-inclination objects match predictions. The Jupiter family comet distribution changes in the expected way. The predicted reduction in Oort cloud capture efficiency aligns with simulations. That's a lot of things to get right by accident. It's not impossible, but it's increasingly unlikely. Each successful prediction raises the prior probability that the underlying model is correct. So where does that leave us as Rubin prepares to begin operations? We're at a decision point. Within 2 years, maybe 3, we'll have data comprehensive enough to either confirm Planet 9's existence or rule it out across most of its possible parameter space. This isn't the kind of question that lingers unresolved for decades. The technology exists. The survey is happening. The answer is coming. If Rubin finds Planet 9, it will be one of the most significant planetary discoveries in history. Not because it's a planet, though that's cool. Because of what it tells us about solar system formation, migration dynamics, and the possibility that our planetary census was incomplete for the entire history of astronomy. If Rubin doesn't find it, that's also significant. It means either the hypothesis was wrong, or Planet 9 is so far, so faint, or so weirdly positioned that even our best efforts can't detect it. That possibility can't be dismissed. The outer solar system is vast. Assumptions about brightness and location might be off. But at some point, a hypothesis that can't be tested stops being useful. The broader implications extend beyond our solar system. Discovering Planet 9 would make our system more similar to others. Most exoplanetary systems contain super-Earths, planets with masses between Earth and Neptune. We don't have any. Planet 9 would fill that gap, suggesting that our system formed similarly to others, but with a quirk: one of the planets got kicked outward instead of staying close. That has implications for planet formation theory. How common is ejection? What determines whether a forming planetary system ends up with all its planets in tight orbits versus having one or more in distant eccentric orbits? Are we typical or unusual? Planet 9 helps answer those questions. It also affects how we think about planetary migration. The NICE model explains a lot about how the giant planets reach their current orbits, but it's not complete. If Planet 9 is an ejected ice giant, that's a direct observation of a process we've only simulated. We could study its composition, atmosphere, and thermal properties. We could learn what happens to a planet that gets thrown into the outer darkness and survives there for billions of years. All of that depends on detection. And detection depends on Rubin. The next few years will be decisive. The search itself has an interesting dynamic. Brown and his team aren't the only ones looking. Multiple groups worldwide are scanning different portions of the sky, using different methods, hoping to be the first to spot it. There's a competitive element, though it's mostly friendly. Everyone wants the discovery, but everyone also wants the answer, regardless of who provides it. What happens if Rubin spots something in the right region with the right motion? Confirmation would take time. A single detection isn't enough. You need multiple observations over weeks or months to establish an orbit. Then you need spectroscopy to determine composition. Is it icy? Gaseous? Does it have an atmosphere? What's its temperature? All of that requires follow-up with other instruments. And here's something people don't always consider. If Planet 9 exists and Rubin finds it, that's not the end of the story. It's the beginning. We'd have a new planet to study. A super-Earth in our own solar system, close enough to observe in detail but far enough to be genuinely alien. What can we learn from it? For one thing, we could study its atmosphere if it has one. Most super-Earths we've detected around other stars are too far away for detailed atmospheric analysis. Planet 9 would be right here, relatively speaking. We could point infrared telescopes at it, measure its thermal emission, detect molecular signatures, and figure out what it's made of. We could also study its interaction with the surrounding environment. Does it have moons? Rings? A magnetic field? How does it affect the local dust distribution? Does it capture comets or asteroids into temporary orbits? All of those questions become answerable once we know where it is. The formation question would become more tractable too. If Planet 9 is an ejected ice giant, we could compare its properties to Uranus and Neptune. Are they similar? Different? That tells us about the conditions under which they formed and what happened during the ejection process. Did Planet 9 lose its outer layers during the encounter with Jupiter? Did it retain them? The answers matter for understanding how planetary systems evolve. There's also the possibility of detecting other distant planets. If one exists, maybe others do. The outer solar system is huge. We've barely scratched the surface. Rubin will find thousands of new trans-Neptunian objects, and some of them might have orbits that point to additional unseen bodies. Planet 9 might not be alone. But let's consider the other scenario. Rubin surveys the entire accessible region and finds nothing. What then? First, it doesn't immediately kill the hypothesis. The predicted orbit has uncertainty. If Planet 9 is near its aphelion and farther than expected, Rubin might miss it. Its brightness could be lower than predicted. There are scenarios where Planet 9 exists but remains undetected even after Rubin's survey. However, those scenarios become increasingly constrained. If Rubin doesn't find it, the parameter space shrinks dramatically. The planet would have to be fainter, farther, or in a very specific location that Rubin didn't cover. At that point, the hypothesis starts looking less likely. It wouldn't vanish entirely. Science doesn't work that way. But the community's confidence would shift. If the best survey we've ever conducted finds nothing, most astronomers would conclude that Planet 9 probably isn't there, at least not in the form currently predicted. That would leave the clustering problem unresolved. The orbital alignment of trans-Neptunian objects would still need an explanation. Maybe it's observational bias after all, despite the corrections. Maybe it's a temporary configuration that will disperse over time. Maybe there's a different mechanism at work that we haven't thought of yet. Or maybe there's something else out there, not a single planet but a different kind of object. A primordial black hole, for instance. That's been proposed as an alternative. A black hole with a few Earth masses could produce similar gravitational effects without being visible. It wouldn't emit light, wouldn't reflect sunlight, and would be nearly impossible to detect directly. The black hole hypothesis is speculative, but it's not absurd. Primordial black holes, if they exist, would have formed in the early universe from density fluctuations. Some could have been captured by the solar system. The gravitational effects would be identical to a planet of the same mass. The only difference is detectability. How would you find a black hole in the outer solar system? Gravitational lensing, maybe. If it passed in front of a distant star, the star's light would bend around it, creating a brief brightening. Rubin could detect that. It's a long shot, but it's possible. Another alternative is a disk of smaller objects, collectively massive enough to produce the observed effects. Instead of one planet, imagine thousands of smaller bodies distributed across a wide region. Their combined gravity could shepherd trans-Neptunian objects into aligned orbits. The disk hypothesis has problems though. To match the observed clustering, the disk would need a very specific mass distribution and configuration. It's not impossible, but it requires fine-tuning. A single planet is simpler. Occam's razor favors the simpler explanation, but simplicity doesn't guarantee correctness. So where does the evidence actually point right now, before Rubin starts observing? The statistical case for Planet 9 has strengthened. The 2024 study showing alignment of low-inclination objects was a big deal. That's a new population, independent of the original clustering, behaving exactly as predicted. The odds of that happening by chance are very low. But statistics aren't proof. Astronomy has seen confident predictions proven wrong before. The key is falsifiability. Planet 9 is falsifiable. If Rubin surveys the predicted region and finds nothing, the hypothesis is either wrong or needs major revision. That's good science. It's worth stepping back and asking why this matters beyond pure curiosity. Why should anyone care if there's a ninth planet? One reason is completeness. If we don't know our own solar system, how can we claim to understand others? Planet 9, if real, has been there the entire time humans have looked at the sky. Missing something that large in our own neighborhood would be humbling. Another reason is the connection to exoplanetary science. Super-Earths are everywhere. We've found thousands around other stars, but we don't have one here to study up close. Planet 9 would be the only super-Earth we can observe in detail, the only one we can send probes to eventually. That makes it scientifically valuable beyond its role in solar system dynamics. There's also the historical angle. Discovering a new planet is rare. It doesn't happen often. Neptune in 1846. Pluto in 1930, though that got reclassified. If Planet 9 is real, this generation gets to witness something that happens maybe once per century. That's not nothing. And then there's the sheer weirdness of it. A planet 10 times Earth's mass, orbiting hundreds of times farther from the Sun than we do, taking 20,000 years to complete one orbit. It's so far out that the Sun looks like just another bright star. Temperatures are near absolute zero. If it has an atmosphere, it's probably frozen solid. It's a world more alien than anything we've explored, and it's technically in our solar system. What would it be like to visit Planet 9? Impossible with current technology, obviously. The distance is staggering. Voyager 1, the fastest probe we've ever launched, has been traveling for over 40 years and still hasn't reached the predicted inner edge of Planet 9's orbit. Getting there would take centuries with existing propulsion systems. But imagine if we could. What would you see? The Sun would be a bright point of light, no disk visible, providing almost no heat. The sky would be black, filled with stars that don't twinkle because there's no atmosphere to distort them. The planet itself, if gaseous, might have faint cloud bands, though they'd be moving incredibly slowly in the cold. Moons, if they exist, would be frozen solid, covered in ancient impact craters. It would be the most isolated place a human could stand and still be within the Sun's gravitational influence. The nearest planet, Neptune, would be billions of kilometers away. Earth would be invisible without a telescope. You'd be at the edge of the solar system, looking outward toward interstellar space, inward toward the distant Sun, and feeling utterly, completely alone. That's Planet 9. If it exists. And within 2 years, we'll have a much better idea whether it does. Rubin starts observations soon. The data will accumulate. The algorithms will scan for slow-moving objects. And somewhere in those billions of data points, there might be a faint dot that moves just slightly between images. Or there might not. Either way, we're about to find out. The search for Planet 9 is entering its final phase. The question that's lingered for nearly a decade will finally get an answer. Whatever that answer is, it'll reshape how we understand the solar system. That's worth paying attention to. Looking at the timeline, we're living through something remarkable. The Vera Rubin Observatory represents the culmination of decades of technological advancement in astronomy. Its 2-gigapixel camera, its systematic survey strategy, its ability to track billions of objects simultaneously, all of it converges on this moment. We built the perfect machine for finding Planet 9 without necessarily intending to. The telescope doesn't care about the hypothesis. It's going to scan the southern sky regardless, cataloging everything that moves, everything that changes, everything that exists in its field of view. Planet 9 is just one target among billions. But if it's there, Rubin will see it. The odds are too good. The coverage too complete. The sensitivity high enough. What happens next depends entirely on what the data shows. If Planet 9 appears in the images, moving slowly across the background stars over weeks and months, the discovery will unfold gradually. Not a single eureka moment, but a pattern emerging from noise. Statistical confidence building with each new observation. Orbital parameters refined as more data points accumulate. Eventually, certainty. The confirmation process would take time. You don't announce a planet based on a handful of detections. You need enough measurements to calculate an orbit accurately. You need spectroscopy to determine composition. You need infrared observations to measure temperature and size. The full characterization might take years. But the initial detection, the moment when someone realizes there's a new object in the right region with the right motion, that could happen within months of Rubin's first light. And if it doesn't show up? If 2 years pass, then and the predicted regions yield nothing. The hypothesis doesn't instantly collapse, but it enters a different phase. The parameter space contracts. The alternative explanations become more attractive. At some point, you stop saying, "We haven't found it yet," and start saying, "It's probably not there." That outcome would be significant too. Not as exciting as discovering a new planet, but scientifically valuable nonetheless. We'd know the clustering of trans-Neptunian objects needs a different explanation. Maybe it's a selection effect we haven't fully accounted for. Maybe it's a transient configuration that won't persist. Maybe there's something else entirely driving those orbits, something we haven't considered. Science advances through ruling things out as much as confirming them. But the smart money is on detection. Brown and Badijan aren't betting their reputations on a whim. They've spent 8 years refining this hypothesis, testing it against new data, addressing criticisms, making predictions that keep being confirmed. The 2024 results on low-inclination objects were particularly compelling. Different population, same result. That pattern repeating across independent datasets is hard to dismiss. The formation question remains fascinating regardless of when detection happens. If Planet 9 is an ejected ice giant, it validates predictions from the NICE model that have been purely theoretical until now. We'd have direct evidence that planetary systems can eject giant planets without losing them entirely. That has implications for every exoplanetary system we observe. How many systems have hidden planets in distant orbits? How common is this configuration? If Planet 9 is a captured exoplanet, the implications are even stranger. It would mean our sun stole a world from another star during the solar system's youth. That's not impossible. Stellar nurseries are chaotic places where close encounters happen. But confirming it would be extraordinary. The first exoplanet discovered in our own solar system, a cosmic immigrant that's been here for 4.5 billion years. The public interest in Planet 9 reflects something deeper than just curiosity about astronomy. There's something compelling about the idea that we still don't fully know our own neighborhood. Despite centuries of observation, despite sending probes to the outer planets, despite thinking we'd mapped everything significant, there might still be a world we missed. A giant world, not some tiny asteroid, but a super-Earth mass planet that's been there all along. It's humbling in the right way. It reminds us that the universe doesn't reveal its secrets easily. Even in our own backyard, even with modern technology, there are still mysteries waiting to be solved. Planet 9, if real, represents the last great planetary discovery possible in the solar system. After this, we'll have found everything larger than Pluto. The inventory will be complete. The search itself demonstrates how modern astronomy works. No lone genius staring through a telescope. Teams of researchers analyzing data from multiple instruments, running simulations on supercomputers, developing statistical methods to extract signals from noise, publishing results for peer review, responding to criticism, refining predictions. It's collaborative, iterative, and methodical. Not as romantic as the old stories of discovery, but more reliable. Brown's comment about Rubin being the perfect telescope for finding Planet 9 wasn't hyperbole. The match between the hypothesis's requirements and the observatory's capabilities is almost perfect. Wide field for covering large search areas. Deep sensitivity for detecting faint objects. Repeated observations for tracking motion. Automated data processing for handling billions of measurements. Everything needed to find a slow-moving distant planet, built into one facility. The timing feels right too. The hypothesis emerged in 2016. The predictions have been tested and refined over nearly a decade. The search space has been narrowed through systematic surveys. And now, just as we're ready for a definitive answer, the ideal instrument comes online. It's not coincidence. It's how science progresses. You develop a hypothesis, you test it as best you can with existing tools, and eventually technology catches up to enable the critical test. Rubin's broader impact on astronomy will extend far beyond Planet 9. The 10-year survey will revolutionize our understanding of the outer solar system, discovering tens of thousands of new trans-Neptunian objects, tracking comets, detecting asteroids, mapping the structure of the Kuiper Belt. Planet 9 is just one target in a much larger program. But it's the target everyone will be watching for. Imagine the moment of discovery. Someone running detection algorithms on Rubin data, seeing an object appear in the right region with the right motion. The double-checking. The orbital calculation. The realization that this might actually be it. The careful announcement. The flood of follow-up observations from telescopes worldwide. The gradual confirmation as the data accumulates. The moment when doubt shifts to certainty. Or imagine the alternative. Years passing. The survey completing. The predicted regions thoroughly searched. Nothing found. The slow acceptance that Planet 9 probably isn't there, at least not where expected. The return to other explanations for the orbital clustering. The questions about what we missed or misunderstood. Both scenarios advance our knowledge. One gives us a new planet to study. The other forces us to rethink our interpretation of outer solar system dynamics. Either way, we learn something fundamental about our cosmic home. The next 2 years will answer a question that's persisted since 2016. Is there a ninth planet? The mathematics say yes. The statistics say yes. The predictions keep being confirmed. But science demands direct observation. We need to see it. And we're finally at the point where that's possible. Planet 9 represents the kind of mystery that makes astronomy compelling. Not abstract or impossibly distant. Right here in our solar system. Big enough to matter. Strange enough to surprise us. And just barely beyond our current ability to detect, though that's about to change. The giant lurking in the darkness beyond Neptune might soon step into the light. Or it might remain a mathematical phantom, a compelling hypothesis that couldn't be confirmed. Within 2 years, we'll know. Rubin will scan the sky, the algorithms will run, and the data will tell us what's really out there. The answer is coming. Beneath Jupiter's ancient surface, hidden under a frozen shell thicker than Earth's tallest mountains, lies an ocean containing more water than all of Earth's seas combined. This isn't speculation, it's measured fact. Ganymede, the largest moon in our solar system, harbors a secret that transforms everything we thought we knew about where life might exist beyond Earth. In 1610, Galileo pointed his primitive telescope toward Jupiter and discovered 4 bright points of light orbiting the giant planet. He had no idea one of them contained an ocean. For centuries later, we've sent spacecraft past Ganymede, mapped its magnetic field, and measured its gravitational pull with extreme precision. The data tells an astonishing story. Deep beneath ice that would crush any submarine we've ever built, liquid water moves in currents we're only beginning to understand. Ganymede is massive. At 5,268 kilometers across, it's larger than Mercury. If it orbited the Sun instead of Jupiter, we'd call it a planet without hesitation. But size alone doesn't make it remarkable. What matters is the structure we've detected beneath that scarred, ancient surface. Layer upon layer of ice and water, stacked like a cosmic sandwich, each zone distinct in temperature, pressure, and composition. The surface ice isn't uniform. Roughly 40% is ancient, dark terrain covered in impact craters that have accumulated over billions of years. The rest is younger, brighter, scored with grooves and ridges that reveal a violent tectonic past. Something powerful resurfaced large portions of Ganymede, and that something came from within. The ice didn't just crack. It moved. Plates shifted. Entire regions were rebuilt by forces we're still trying to explain. Scientists first suspected an ocean in the 1970s, but suspicion isn't proof. The breakthrough came from Galileo, the spacecraft, not the astronomer. Between 1995 and 2003, this robotic explorer made 6 close flybys of Ganymede, gathering magnetic field data that changed the game entirely. Ganymede generates its own magnetic field, the only moon in the solar system known to do so. That field interacts with Jupiter's powerful magnetosphere in ways that reveal what's hidden below. When Galileo's magnetometer detected variations in Ganymede's field, researchers noticed something unexpected. The patterns suggested electrical currents flowing beneath the surface. Salty water conducts electricity. Pure ice does not. The data pointed unmistakably toward a subsurface ocean rich in dissolved salts. Multiple independent studies confirmed it. Ganymede has liquid water, and lots of it. How much water are we talking about? Conservative estimates suggest the ocean contains more water than Earth's Atlantic, Pacific, Indian, Arctic, and Southern oceans combined. Some models push that figure even higher. The ocean might be 100 kilometers deep, possibly more, sandwiched between layers of ice in a configuration nothing like what we see on Earth. Temperature becomes strange at those depths. Near the surface, ice reaches temperatures around -160 degrees Celsius, cold enough to shatter steel. But pressure changes everything as you descend. The weight of overlying ice creates heat through compression. Radioactive decay in Ganymede's rocky core adds more warmth. Tidal flexing from Jupiter's gravity generates additional energy as the moon's shape distorts slightly during its orbit. All these factors combine to keep water liquid despite being farther from the sun than any ocean in our immediate cosmic neighborhood. The structure is layered in ways that challenge our understanding of planetary interiors. Scientists propose multiple distinct zones. The top layer is solid ice, perhaps 150 kilometers thick. Below that, the ocean. Beneath the ocean, another ice layer, but this ice forms under such extreme pressure that it becomes Ice 3, Ice V, or even Ice 6, exotic crystalline structures that only exist under conditions we can barely replicate in laboratories. These high-pressure ices are denser than water, so they sink, creating an inverted world where ice floats below liquid, not above it. At the very bottom sits Ganymede's rocky core, probably similar in composition to Earth's mantle but much smaller. This core might still be warm enough to drive hydrothermal activity. On Earth, hydrothermal vents pump minerals and heat into the deep ocean, creating oases of life in absolute darkness. Could similar processes operate on Ganymede? We don't know. But the possibility exists. Jupiter's influence on Ganymede cannot be overstated. The planet's gravity doesn't just hold the moon in orbit. It squeezes it. Ganymede's orbit isn't perfectly circular, so the strength of Jupiter's gravitational pull varies throughout each 7-day orbit. This variation flexes Ganymede's interior like a stress ball being repeatedly squeezed and released. That flexing generates heat, the same process that powers Io's spectacular volcanism and maintains Europa's subsurface ocean. But Ganymede's tidal heating is gentler than Europa's. Europa orbits closer to Jupiter and experiences far more intense flexing. Ganymede sits farther out, in a more stable orbit, receiving just enough tidal energy to maintain liquid water without the violent geological activity that resurfaces Europa constantly. This might actually be an advantage. A calmer environment could be more conducive to complex chemistry, perhaps even biology. The magnetic field discovery revealed something else. Ganymede's field is surprisingly strong, about 1% the strength of Earth's field. That might not sound impressive, but for a moon, it's extraordinary. The field originates from a metallic core, probably iron or iron-nickel, that remains partially molten. A dynamo operates inside Ganymede, circulating liquid metal and generating magnetism through the same basic process that protects Earth from solar radiation. This magnetic field creates a miniature magnetosphere around Ganymede, a protective bubble carved out of Jupiter's much larger magnetic environment. When Ganymede passes through different regions of Jupiter's magnetosphere, the interaction changes. Scientists use these variations to probe what lies beneath the ice. The ocean's salty water conducts electrical currents that alter the magnetic field in detectable ways. It's like using magnetism to see through hundreds of kilometers of ice without drilling a single hole. The composition of that ocean remains uncertain. We know it contains dissolved salts, but which salts? Sodium chloride, like Earth's oceans? Magnesium sulfate? Some exotic mixture we've never encountered? Spectroscopic observations of Ganymede's surface show evidence of various salts and minerals, including what might be hydrated salts pushed up from below through cracks in the ice. These surface deposits offer tantalizing clues about the ocean's chemistry, but they're just hints. We won't know the ocean's true composition until we sample it directly. Sampling that ocean presents extraordinary challenges. The ice shell is thicker than any ice we've drilled through on Earth. Antarctica's deepest ice cores reach about 3.5 kilometers. Ganymede's ice might be 50 times thicker. Drilling through that much ice would require technology we haven't developed yet, power sources that don't exist, and mission durations that stretch decades. Alternative approaches exist. Perhaps we could target thinner regions where the ice is fractured or where warm upwellings have thinned the shell. Maybe we could send a probe through one of the cracks visible on the surface, assuming those cracks penetrate deep enough. Or we might analyze material ejected from the subsurface by impacts or cryovolcanic activity. Each method has drawbacks, but they're being studied seriously. The surface itself tells stories about what lies beneath. Those parallel grooves and ridges covering the brighter terrain formed through tectonic processes. Ganymede's ice shell fractured and shifted, driven by forces from below. Some regions show evidence of past cryovolcanism, where liquid water or slushy ice erupted onto the surface, freezing instantly in the vacuum of space. These features are old, billions of years in some cases, but they prove the interior was once geologically active. The question is whether that activity continues today. Recent observations suggest it might. The Hubble Space Telescope detected evidence of water vapor plumes erupting from Ganymede's surface, similar to the plumes Enceladus shoots into space from its south polar region. If confirmed, these plumes would be game-changing. They'd provide direct samples of subsurface material without requiring us to land or drill. A spacecraft could fly through the plumes, collect particles, and analyze their composition. We'd learn about the ocean's chemistry, search for organic molecules, and potentially detect signs of biological activity, all without touching the surface. The plume evidence isn't conclusive yet. The signal is subtle, and alternative explanations exist. But if Ganymede does have active plumes, it joins Europa and Enceladus in a select group of ocean worlds that occasionally expose their hidden seas to space. This would make Ganymede a prime target for future missions designed to search for life beyond Earth. Understanding Ganymede's ocean requires understanding its history. The Moon formed roughly 4.5 billion years ago from the disk of gas and dust surrounding the infant Jupiter. Early in its history, Ganymede was probably much warmer. Radioactive decay and impact heating from late-stage bombardment would have created conditions warm enough to melt ice throughout the interior. The Moon might have been entirely liquid at some point, with a global magma ocean overlying a water ocean of staggering depth. As Ganymede cooled, it differentiated. Heavier rocky material sank to form the core. Lighter ices rose toward the surface. Water separated from rock, creating distinct layers. The cooling continued, but never reached the point where the entire Moon froze solid. Tidal heating from Jupiter and residual heat from radioactive decay kept the interior warm enough to maintain liquid water, even as the surface froze into the icy shell we observe today. That surface has been modified countless times. The dark, cratered terrain preserves Ganymede's ancient face, a record of impacts stretching back billions of years. The bright, grooved terrain is younger, perhaps 1 to 2 billion years old, created when the ice shell fractured and new ice flooded the surface. What triggered this resurfacing event? Possibly a period of enhanced tidal heating when Ganymede's orbit shifted slightly. Or perhaps a massive impact disturbed the ice shell enough to trigger widespread tectonic activity. The grooves themselves are puzzling. They run parallel for hundreds of kilometers, creating patterns that suggest systematic stress across the entire ice shell. Some researchers think the grooves formed as the ice expanded during freezing, creating tension that cracked the surface. Others propose they're the result of tectonic plates sliding past each other, similar to transform faults on Earth. The truth might involve both processes working together in ways we don't yet understand. What makes Ganymede's ocean potentially habitable? 3 factors stand out. First, liquid water, the universal solvent necessary for chemistry as we know it. Second, chemical energy sources, possibly including hydrothermal vents if the rocky core remains active. Third, time, billions of years for complex chemistry to evolve and potentially produce something we'd recognize as life. That third factor is crucial. Life on Earth didn't appear overnight. It took hundreds of millions of years for chemistry to cross the threshold into biology. Ganymede's ocean has existed for most of the solar system's history. If conditions have remained stable, that's an enormous window for biological processes to develop. Of course, stability doesn't guarantee life. It only provides the opportunity. Earth's ocean spent billions of years churning with chemistry before producing the first cells. And even after life emerged, it remained simple for most of Earth's history. Complex organisms, the kind that build structures or move purposefully, arrived remarkably late. On Ganymede, we're searching for something far more modest, evidence that chemistry has become organized, that molecules have learned to replicate, that energy flows through systems we could call metabolic. The search faces immediate obstacles. Ganymede's ocean lies beneath potentially 150 kilometers of ice. No drill exists that could penetrate that depth in reasonable time. No submarine could reach those depths and return data. We're forced into indirect methods, analyzing the surface for clues about what lies below, studying how the magnetic field behaves, watching for changes that might indicate activity deep within. But there's a faster approach. If Ganymede truly has water plumes erupting through the ice, we bypass the barrier entirely. A spacecraft flying through those plumes would sample ocean material directly. Instruments could detect organic molecules, measure salinity, identify chemical signatures that might indicate biological processes. We'd essentially be tasting Ganymede's ocean without ever landing. NASA and ESA are already planning missions that could make this happen. The Europa Clipper, launching in the 2020s, will study Jupiter's moons during multiple flybys. While its primary target is Europa, it will also observe Ganymede, searching for plume activity and mapping the magnetic field in greater detail. The JUICE mission, led by ESA, will spend years studying Ganymede specifically, eventually entering orbit around the Moon itself. JUICE carries instruments designed to peer through the ice using radar, to measure the thickness of different layers, to map variations in the gravitational field that might reveal ocean currents below. These missions won't answer every question. They're reconnaissance, not final proof. But they'll narrow the possibilities. They'll tell us where the ice is thinnest, where tidal heating concentrates, where future landers should aim. They're setting the stage for something more ambitious, a mission that actually touches Ganymede's surface and drills into the ice. Drilling on Ganymede presents challenges Earth's polar expeditions never faced. Temperatures hover around -160 degrees Celsius. There's no atmosphere to provide pressure or distribute heat. Solar power is weak this far from the sun. Any drill would need to carry its own energy source, probably a radioisotope generator, and melt through ice that's harder than concrete at those temperatures. The drill would need to operate autonomously, without real-time control from Earth. Radio signals take 40 minutes to travel between Jupiter and our planet, making remote piloting impossible. Every decision, every adjustment, would need to happen on site, guided by artificial intelligence capable of responding to unexpected conditions. Assuming we overcome those obstacles, what happens when the drill reaches liquid water? The bit punches through the final meters of ice and suddenly encounters resistance-free fluid. Sensors detect the transition. The drill stops advancing. A probe deploys, a small autonomous vehicle designed to separate from the drill string and swim freely in the ocean below. It carries cameras, though visible light penetrates only meters in these dark waters. It carries chemical sensors to taste the ocean's composition. It carries sonar to map the underside of the ice and detect objects in the surrounding water. And it carries life detection instruments, devices designed to recognize patterns that biology creates, organic molecules arranged in ways chemistry alone wouldn't produce. This isn't science fiction. Engineers have already built prototypes. They've tested autonomous underwater vehicles beneath Antarctic ice, demonstrating that machines can navigate and collect samples in environments humans will never visit. The technology exists. What's missing is the funding, the political will, and the decades of patience required to send these systems to another world. Why go to such extraordinary lengths? Because Ganymede represents a test case for a much larger question. If life can arise in an ocean sealed beneath ice, isolated from sunlight, dependent entirely on chemical energy from hydrothermal vents, then life might be common throughout the universe. Most planets don't orbit their stars in the narrow habitable zone where liquid water can exist on the surface. But countless moons orbit giant planets, experiencing tidal heating, maintaining subsurface oceans regardless of how far they are from their star. Ganymede-like worlds might outnumber Earth-like worlds by orders of magnitude. Finding life on Ganymede would fundamentally shift how we think about biology's place in the cosmos. It would suggest that wherever liquid water persists for geological time, life finds a way to emerge. The universe suddenly becomes much more alive, even if that life hides beneath frozen surfaces, invisible to telescopes, detectable only through patient exploration. The implications extend beyond biology. Ganymede's ocean, if inhabited, would represent a second origin of life in our solar system. Earth's life shares a common ancestor. Every organism, from bacteria to blue whales, descended from the same primordial cell. We're all one family, variations on a single theme. But life on Ganymede, if it exists, would have started independently. Its chemistry might use different molecules. Its cells might have different structures. Its metabolism might run on different reactions. Comparing Earth life to Ganymede life would reveal which aspects of biology are universal and which are accidents of our particular history. There's also the possibility that Ganymede's ocean contains nothing alive at all. That the chemistry never crossed the threshold, that the conditions weren't quite right, that life requires something we haven't identified yet. This outcome would be scientifically valuable too. It would tell us that liquid water and chemical energy aren't sufficient. That life requires additional factors we need to understand before we can predict where else it might arise. Every null result narrows the search, eliminating possibilities, focusing attention on what truly matters. Ganymede's future depends partly on Jupiter. The giant planet's gravity dominates the entire system. Tidal forces that keep Ganymede's ocean liquid also create stress throughout the Jovian system. Jupiter's moons interact gravitationally, their orbits shifting subtly over millions of years. These orbital changes affect how much tidal heating each moon experiences. Ganymede might be warming or cooling depending on these long-term cycles. The ocean could be expanding or contracting. The ice shell might be thickening or thinning. We're seeing a snapshot of a dynamic system, one that changes on timescales far longer than human civilization has existed. Looking billions of years forward, Ganymede's fate is tied to the Sun. Our star is slowly brightening. In roughly 1 billion years, it will be hot enough that Earth's oceans begin to evaporate. The habitable zone will shift outward. By the time the Sun becomes a red giant, perhaps 5 billion years from now, it will expand dramatically, possibly engulfing the inner planets entirely. Jupiter's orbit will be warm enough that Ganymede's ice shell melts completely. The Moon might briefly have a surface ocean, liquid water exposed to space, before the Sun's evolution continues and everything boils away. But that's distant speculation. Right now, Ganymede orbits Jupiter much as it has for billions of years. Its surface remains locked in frozen silence. Its ocean, if inhabited, contains life that has never seen a star, never experienced seasons, never known anything beyond the crushing pressure and absolute darkness of its submerged world. And yet, that life, if it exists, would be our cosmic cousin, proof that the universe creates biology wherever conditions allow, that we're not alone in finding ways to persist and adapt and thrive. The next steps are clear. We need to return to Ganymede with better instruments. We need to map the ice shell's thickness precisely. We need to confirm whether plumes are erupting. We need to identify the best drilling sites. And eventually, we need to send a drill, punch through that ice, and taste the ocean that has remained hidden since the solar system's birth. Each mission builds on the last. Each discovery raises new questions. The process is slow, frustratingly slow for those eager for answers. But it's also methodical, accumulating evidence piece by piece, building a case that will eventually become undeniable. Ganymede doesn't care whether we study it or not. The moon will continue its orbit, its ocean will continue to slosh beneath the ice, its magnetic field will continue to deflect Jupiter's radiation. But we care, because understanding Ganymede means understanding how common habitable environments might be throughout the galaxy. It means learning whether life is a cosmic accident that happened once, here on Earth, or an inevitable consequence of chemistry given the right conditions and enough time. That question drives everything we do in space, every mission we plan, every instrument we build. Ganymede's story matters because it rewrites what we thought we knew about where life can exist. For most of human history, habitability meant one thing: being near a star at just the right distance. Not too hot, not too cold. The Goldilocks zone. Earth sits comfortably within that zone, receiving sunlight that keeps water liquid on the surface. That seemed like the requirement. No sunlight, no warmth, no life. Ganymede ignores that rule completely. It orbits 5 times farther from the sun than Earth does. Sunlight there is faint, barely more than moonlight from our perspective. Surface temperatures hover around -160 degrees Celsius. By traditional standards, Ganymede should be a frozen wasteland, utterly sterile. Yet beneath that frozen exterior lies an ocean larger than all of Earth's oceans combined, kept liquid not by sunlight but by gravitational friction. The rules changed. Habitability no longer requires proximity to a star. It requires energy, and energy comes from multiple sources. This realization transforms how we think about the galaxy. Current estimates suggest hundreds of billions of planets exist in the Milky Way alone. Most orbit too far from their stars to have surface liquid water. We used to dismiss those worlds as uninteresting, too cold to matter. But if tidal heating can maintain subsurface oceans, then billions of frozen worlds suddenly become potential habitats. Ice becomes a protective shell rather than a barrier. Darkness becomes normal rather than fatal. The number of potentially habitable environments in the galaxy might be orders of magnitude higher than we previously calculated. Europa taught us this lesson first. It's smaller than Ganymede, closer to Jupiter, and experiences even more intense tidal heating. Its ice shell might be only 15 to 25 kilometers thick, potentially thin enough that drilling becomes realistic with current technology. Europa's ocean might be in direct contact with a rocky seafloor, creating conditions similar to Earth's hydrothermal vents. Those vents on Earth support ecosystems that thrive without sunlight, using chemical energy instead. If Europa has similar vents, it might have ecosystems too. But Ganymede offers something Europa doesn't: stability. Europa's surface is young, constantly resurfacing from geological activity. Its ice shell might be churning, cycling material between the surface and the ocean on relatively short timescales. That's good for delivering nutrients but chaotic for any life trying to establish itself. Ganymede's surface is ancient, some regions dating back billions of years. The ice shell appears thicker, more stable. The ocean beneath might be calmer, less disturbed. Life, if it exists, has had billions of years to develop in relatively stable conditions. That matters enormously. Consider Earth's history. Single-celled organisms appeared relatively quickly, within the first billion years after the planet cooled enough to have liquid water. But complex life took far longer. Multicellular organisms didn't emerge until roughly 600 million years ago. Large animals appeared only in the last half-billion years. Intelligence required even more time. Evolution isn't just about having the right ingredients. It needs time to experiment, to try countless variations, to stumble upon solutions that work. Ganymede has had that time. If its ocean formed during the solar system's early days, life there has had 4 billion years to evolve. What might 4 billion years of evolution produce in an alien ocean? We have no idea. That's the honest answer. Earth's oceans contain everything from microscopic bacteria to blue whales, from simple sponges to intelligent octopuses. Evolution explores countless paths, finding solutions we'd never imagine. An alien ocean isolated from Earth's evolutionary history would follow its own trajectory, shaped by different pressures, different chemistry, different constraints. The organisms there, if they exist, might share some basic principles with Earth life. They'd need to extract energy from their environment, maintain their internal chemistry, reproduce. But the details could be utterly foreign. Imagine organisms that navigate by sensing magnetic fields directly, creatures that communicate through electrical signals in ways far more sophisticated than Earth's electric fish. Perhaps something analogous to plants but using chemical energy from hydrothermal vents instead of sunlight, creating the base of a food chain. Predators that hunt in absolute darkness using senses we can't comprehend. Maybe colonial organisms, vast networks of interconnected cells functioning as single entities, stretching across kilometers of ocean floor. Or nothing like any of those things. Evolution is creative in ways that constantly surprise us even when studying Earth's own biosphere. The chemical composition of Ganymede's ocean adds another layer of complexity. Earth's oceans are salty, dominated by sodium chloride. Ganymede's ocean might have different salts, possibly magnesium sulfate or other compounds. The pH could be different. The pressure at the ocean floor would be immense, potentially several hundred times Earth's atmospheric pressure. Organisms there would need to function under those conditions. They'd have no concept of low pressure, no evolutionary history of dealing with anything but crushing weight. Bring them to the surface and they'd likely die instantly, cells rupturing from the pressure drop. We'd need to maintain samples at depth, study them in specialized chambers, never letting them experience what we consider normal conditions. Temperature poses another question. Earth's deep oceans are cold but not frozen, typically around 2 to 4 degrees Celsius. Ganymede's ocean temperature depends on how much heat reaches it. Near the ice shell, it's probably just above freezing, close to 0 Celsius. Near the ocean floor, if hydrothermal vents exist, temperatures could spike dramatically. Earth's vents can reach hundreds of degrees Celsius. Water remains liquid under pressure at those temperatures. Organisms on Earth thrive in these extreme environments. Ganymede might have similar temperature gradients, creating diverse habitats within the same ocean. Cold-adapted organisms near the ice, heat-loving extremophiles near vents, and everything in between. Salinity affects what can dissolve in water. Different salts change water's properties, how it conducts electricity, how it freezes, how molecules move through it. Life would adapt to whatever chemistry exists there, evolving proteins and cell membranes suited to that specific environment. We can't simply transplant Earth organisms to Ganymede's ocean and expect them to survive. The chemistry would be wrong. But native organisms, if they exist, would be perfectly suited to their home, finding our ocean as alien and hostile as we'd find theirs. Energy sources drive everything. On Earth, sunlight powers most ecosystems directly or indirectly. Remove the sun and Earth's biosphere would collapse within years. Ganymede never had that dependence. Its energy budget relies on tidal heating and possibly radioactive decay in the rocky core. That's a limited energy supply compared to sunlight. Earth receives roughly 174 petawatts of solar energy. Ganymede's tidal heating probably delivers far less, maybe a few terawatts at most. That limits how much biomass the ocean can support. An ecosystem there would be sparse compared to Earth's teeming oceans. Fewer organisms spread over vast distances, competing for limited chemical energy. Life, but not abundant life. Survival, but on the edge. This scarcity might actually favor certain evolutionary paths. On Earth, competition drives arms races, predators evolving better hunting skills, prey evolving better defenses. But in energy-poor environments, cooperation sometimes wins over competition. Organisms might evolve symbiotic relationships, sharing resources, supporting each other's metabolism. We see this on Earth in extreme environments. Lichens are partnerships between fungi and algae. Deep-sea tube worms host bacteria that process chemicals from hydrothermal vents. Ganymede's organisms might take cooperation even further, creating integrated systems that blur the line between individual and colony. Then there's the question of complexity. Does low energy availability limit how complex organisms can become? Large brains require enormous energy. Human brains consume roughly 20% of our total energy budget despite being only 2% of our body mass. Intelligence is expensive. In Ganymede's ocean, that cost might be prohibitive. Evolution might favor simple, efficient organisms over complex, energy-hungry ones. Intelligence might never emerge, not because it's impossible but because it's not worth the energy investment. Or perhaps intelligence evolves differently there, taking forms that don't require massive energy consumption. Distributed intelligence across colonies, simple individual units that collectively make complex decisions. We assume intelligence looks like human brains. It doesn't have to. Time becomes fascinating when discussing Ganymede's potential biosphere. Earth's organisms evolved alongside a changing planet. Continents shifted, climates changed, ice ages came and went. Mass extinctions wiped out dominant species, allowing new lineages to emerge. Evolution responds to change. But Ganymede's ocean might be remarkably stable. No seasons, no day-night cycle, no continental drift affecting ocean currents. Temperature and chemistry might vary little over millions of years. In such stable conditions, evolution might slow. Once organisms adapt well to their environment, there's less pressure to change. We might find living fossils, organisms essentially unchanged for billions of years because their environment never demanded innovation. Or perhaps stability allows evolution to explore other directions. Without constant environmental upheaval, evolution might focus on optimization rather than adaptation. Organisms becoming incrementally more efficient at extracting energy, processing nutrients, reproducing. Refinement over time rather than dramatic shifts. The result would be organisms exquisitely tuned to their environment but unable to cope with even slight changes. Specialists rather than generalists. That creates vulnerability. If something disrupts Ganymede's ocean, a massive impact delivering heat, a change in Jupiter's tidal forces, the ecosystem might collapse entirely, unable to adapt quickly enough. We're speculating, building hypotheses from limited data. That's necessary at this stage. We can't know what exists on Ganymede until we look. But we can make educated guesses based on physics, chemistry, and biology. We can identify what's possible, what's probable, what's unlikely. Those guesses guide mission planning, helping us decide where to look, what instruments to bring, what measurements matter most. Every mission updates our understanding, confirms some guesses, disproves others, raises new questions we hadn't considered. Europa offers a different scenario, another frozen moon hiding an ocean, but with critical differences. It orbits closer to Jupiter than Ganymede, experiencing stronger tidal heating. That means more energy available for life, potentially more active geology on the ocean floor. Europa's ice shell might be thinner, perhaps only 10 to 15 kilometers in some regions. Thinner ice changes the game entirely. With less ice overhead, more radiation from Jupiter penetrates deeper. Radiation breaks apart water molecules at the surface, creating oxidants, chemicals that can participate in biological reactions. Some of those oxidants might work their way down through cracks and fractures, mixing into the ocean below. That influx represents a different energy source, one independent of hydrothermal vents. Earth's early oceans lacked oxygen for billions of years. Life evolved without it, using other chemical pathways. When photosynthetic organisms finally started producing oxygen, it changed everything, enabling more efficient metabolism, more complex organisms, larger bodies. Europa's ocean might skip the waiting period. Oxidants from the surface could provide that efficiency boost from the start, allowing life to evolve along different trajectories than Earth's early biosphere. Europa's surface shows strange features, reddish-brown streaks and patches staining the ice. Spectroscopy suggests those colors come from salts, sulfates, and organic compounds brought up from below. The ice isn't static. It churns, cycles, brings deep material to the surface, sends surface material down. That exchange creates possibilities. If life exists in Europa's ocean, evidence might reach the surface eventually, locked in ice we could sample without drilling through kilometers of frozen water. NASA's Europa Clipper mission, launching soon, will study those surface features in detail, mapping the ice shell's thickness, measuring the ocean's salinity, searching for plumes of water vapor erupting through cracks. The Hubble Space Telescope spotted potential plumes back in 2012 and 2016, geysers shooting water hundreds of kilometers above the surface. If those plumes are real and consistent, a spacecraft could fly through them, collecting samples directly from the subsurface ocean without landing. Analyzing those samples for organic molecules, for chemical signatures inconsistent with purely geological processes, for anything suggesting biological activity. That's the dream scenario, finding life without the engineering nightmare of drilling through ice in low gravity on a radiation-blasted moon. But plumes are unpredictable. They might not erupt when we're watching. They might not contain material from deep enough to include biological signatures. We might fly through them and find only water vapor and ice crystals, learning nothing about what lies below. Still, it's worth trying. The payoff, if successful, justifies the risk. Then there's Enceladus, Saturn's tiny moon, barely 500 kilometers across. Too small for significant radioactive heating, too far from Saturn for strong tidal forces. By all rights, it should be frozen solid, geologically dead. Yet it's not. In 2005, Cassini detected massive plumes erupting from cracks near Enceladus's south pole, shooting water vapor, ice particles, and organic molecules into space. Those plumes feed Saturn's E-ring, a faint ring of ice particles orbiting the planet. Enceladus is active, spectacularly so, despite having no obvious reason to be. The best explanation involves tidal heating, but in a subtle form. Enceladus orbits Saturn in resonance with other moons, gravitational interactions that slightly alter its orbit over time. Those orbital changes generate just enough tidal flexing to keep a subsurface ocean liquid, at least beneath the south polar region. The ocean might be relatively small, perhaps confined to a layer beneath the southern hemisphere rather than encircling the entire moon. But it's there, and it's in direct contact with space through those cracks, essentially offering free samples to any spacecraft passing by. Cassini flew through Enceladus's plumes multiple times, analyzing their composition. Water vapor, as expected. Salts, indicating the ocean is salty. Molecular hydrogen, suggesting hydrothermal activity on the ocean floor where hot water reacts with rock. Organic molecules, simple ones like methane, but also more complex carbon compounds. Everything needed for life, delivered directly into space where we can study it. We've already sampled Enceladus's ocean, at least partially. We just haven't analyzed the samples thoroughly enough yet. Future missions could do better. A dedicated Enceladus orbiter could fly through the plumes repeatedly, collecting particles, measuring their composition with high-precision instruments. We could search for amino acids, the building blocks of proteins. For lipids, which form cell membranes. For nucleotides, the components of DNA and RNA. For anything that screams biology rather than chemistry. Detection wouldn't be simple. Organic molecules exist in space independent of life. Meteorites contain amino acids formed through non-biological processes. We'd need to identify patterns, ratios of molecules, specific configurations that make sense only in biological contexts. Left-handed versus right-handed amino acids, for example. Life on Earth uses exclusively left-handed amino acids in proteins, even though both forms exist naturally. Finding a strong preference for one-handedness in Enceladus samples would raise serious questions. Or we could look for complexity. Random chemistry produces simple molecules. Biology produces complex ones, polymers, chains of repeating units with specific sequences. DNA is a polymer. So are proteins. Finding long-chain molecules with non-random sequences would be compelling evidence, not definitive proof, but enough to shift the conversation from "might life exist" to "how do we confirm it exists?" Titan, Saturn's largest moon, presents an entirely different puzzle. It's the only moon in the solar system with a thick atmosphere, denser than Earth's, composed primarily of nitrogen with a few percent methane. That methane shouldn't be there. Sunlight breaks methane apart, converting it to more complex hydrocarbons. Without a continuous source, Titan's atmospheric methane should disappear within tens of millions of years. Yet it's still there, suggesting active replenishment from Titan's interior. Titan's surface temperature hovers around -179 degrees Celsius, cold enough that water ice behaves like rock, hard and unyielding. Liquid water can't exist on the surface. But liquid methane can. Titan has lakes, rivers, and seas of liquid methane and ethane, a hydrological cycle running on hydrocarbons instead of water. Methane rains from clouds, flows across the surface, collects in basins, evaporates back into the atmosphere. It's alien yet strangely familiar. Could life exist in liquid methane? Water-based life seems impossible at those temperatures. Biochemistry as we know it relies on water's unique properties, its ability to dissolve many substances, its role in biological reactions. Methane doesn't share those properties. It's a poor solvent, doesn't participate in the same kinds of reactions. Life in methane lakes would require completely different chemistry, different molecular structures, different energy sources. Some scientists propose hypothetical organisms with cell membranes made from compounds that remain flexible at cryogenic temperatures, metabolisms based on reactions between hydrogen and acetylene rather than oxygen and glucose. These organisms would be slow, unimaginably slow, their chemistry proceeding at rates millions of times slower than Earth life. They might take centuries to reproduce, millennia to evolve noticeable changes. But slow doesn't mean impossible. Titan also hides a subsurface ocean, this one made of water mixed with ammonia, buried beneath an ice shell perhaps 100 kilometers thick. That ocean wouldn't host methane-based life, but conventional water-based organisms might thrive there, similar to what we expect on Europa or Enceladus. Titan could potentially host two completely separate biospheres, one in surface methane lakes, another in the deep water ocean, neither aware of the other's existence. Testing these ideas requires landing on Titan, sampling the lakes, analyzing their chemistry. NASA's Dragonfly mission, scheduled to launch in the 2030s, will do exactly that. It's a rotorcraft, essentially a drone, designed to fly through Titan's thick atmosphere, landing at multiple sites, collecting samples, searching for organic molecules and chemical patterns that might indicate biology. Finding life in Titan's methane lakes would revolutionize biology more than finding it on Europa. Europa's ocean resembles Earth's early seas closely enough that life there might operate on similar principles. Titan's surface environment has no Earth analog. Life there would represent a genuinely independent origin, proving that biology isn't limited to water, that the universe might harbor forms of life we haven't imagined yet. Mars complicates the discussion. It's not an ocean world now, but it was once. Billions of years ago, Mars had rivers, lakes, possibly even a northern ocean. Liquid water flowed across the surface under a thicker atmosphere. That Mars could have hosted life, might still host remnants of that ancient biosphere in subsurface aquifers or deep rock formations. Recent discoveries strengthen that possibility. Orbital radar detected what appears to be a large reservoir of liquid water beneath Mars's south polar ice cap, kept liquid by salts that lower water's freezing point. If confirmed, that reservoir could harbor microbial life, organisms surviving in isolation since Mars's surface dried out billions of years ago. Mars also shows seasonal variations in atmospheric methane, concentrations rising and falling in patterns that don't match simple geological explanations. On Earth, methane comes primarily from biological sources, produced by microorganisms in oxygen-poor environments. Martian methane could have biological origins, microbes living underground, releasing methane that seeps to the surface. Or it could come from geological processes, reactions between water and rock that happen to produce methane. We don't know yet. Distinguishing between biological and geological methane requires isotope analysis, measuring the ratios of different carbon isotopes in the methane molecules. Biological processes prefer lighter isotopes, creating characteristic ratios. Future Mars missions will carry instruments capable of that analysis, potentially answering whether Mars's methane hints at hidden life. Venus seems like a terrible candidate for life. Surface temperatures exceed 460 degrees Celsius, hot enough to melt lead. Atmospheric pressure at the surface equals that found nearly a kilometer underwater on Earth. The atmosphere is mostly carbon dioxide with clouds of sulfuric acid. Nothing lives there, surely. But Venus's upper atmosphere, 50 to 60 kilometers above the scorched surface, offers more hospitable conditions. Temperature and pressure there resemble Earth's surface. The clouds contain water droplets, though mixed with sulfuric acid. Some microorganisms on Earth, extremophiles living in acid mine drainage, tolerate similarly harsh chemistry. Could microbes drift through Venus's clouds, floating in the relatively mild upper atmosphere, avoiding the hellish surface below? In 2020, scientists detected phosphine in Venus's atmosphere, a gas that on Earth is produced almost exclusively by anaerobic microbes. Phosphine shouldn't persist in Venus's atmosphere, it should react with other compounds and break down quickly. Its presence suggests active production from some source. Biological production would explain it, though geological processes might also be responsible. The detection sparked intense debate, subsequent observations yielded conflicting results, and the question remains unresolved. Looking for life beyond Earth isn't just about finding microbes in ice or gas signatures in distant atmospheres. It's about understanding whether biology is common or rare, whether Earth represents a cosmic fluke or an inevitable outcome when conditions align. Every ocean world, every atmospheric anomaly, every unexplained chemical signature adds evidence to one side of that fundamental question. The search methods keep evolving. Early approaches assumed alien life would resemble Earth life closely, using the same chemistry, the same energy sources, the same molecular building blocks. Modern astrobiology thinks broader. Life might use different solvents, different genetic molecules, different metabolic pathways. Methane could replace water. Silicon might substitute for carbon. Genetic information could be stored in molecules we haven't considered. This flexibility makes detection harder but more interesting. Biosignatures, the signs life leaves behind, fall into several categories. Atmospheric biosignatures include gases that don't belong, oxygen accumulating where it shouldn't, methane appearing in suspicious patterns, phosphine showing up without clear geological sources. These gases react with other atmospheric compounds, breaking down over time. Their persistence means something keeps producing them. Surface biosignatures include patterns visible from space, regular seasonal changes in color suggesting vegetation cycles, reflectance spectra matching chlorophyll or similar pigments. Temporal biosignatures involve changes over time, day-night variations in atmospheric chemistry, seasonal fluctuations in gas concentrations, patterns that match biological rhythms rather than simple physics. Each type has limitations. Geology mimics biology frustratingly well. Volcanoes produce gases that resemble metabolic waste. Mineral reactions create compounds that look biological. Seasonal temperature changes drive chemical cycles that mirror life's rhythms. Distinguishing biology from geology requires multiple lines of evidence, combinations of signals that together become harder to explain without invoking life. The James Webb Space Telescope changed the game for atmospheric analysis. Its infrared instruments can detect molecular signatures in exoplanet atmospheres during transits, when planets pass in front of their stars. Starlight filters through the planet's atmosphere, certain wavelengths get absorbed by atmospheric molecules, and the resulting spectrum reveals what's present. Webb can detect water vapor, carbon dioxide, methane, ammonia, and more complex organic molecules. Finding oxygen and methane together would be significant. Those gases react with each other, so their coexistence suggests continuous production from some active source. But even that wouldn't prove life conclusively. Jupiter's moon Io has a thin oxygen atmosphere produced entirely by geological processes, radiation breaking apart water molecules frozen on the surface. Methane appears in many planetary atmospheres from purely chemical reactions. The combination becomes interesting, especially if accompanied by other biosignatures, seasonal variations, or unexplained ratios between different isotopes. Searching ocean worlds requires different strategies. We can't directly observe their buried seas. Instead, we look for indirect evidence, plumes of water vapor erupting through surface ice, chemical analysis of that vapor, detection of organic molecules or unusual isotope ratios. Enceladus makes this relatively easy. Its geysers spray ocean water into space where spacecraft can fly through and analyze it. Europa will be harder. Its ice shell is thicker, plumes less frequent, but still potentially detectable. Future missions will carry more sophisticated instruments. Europa Clipper, launching in the 2020s, will conduct dozens of close flybys, using ice-penetrating radar to map the ocean's depth and salinity, analyzing any plume material it encounters, measuring the thickness and structure of the ice shell. If conditions look promising, a follow-up mission might attempt to land, drill through the ice, and deploy a submarine into the ocean below. That mission is decades away, requiring technology that doesn't exist yet, but the concept drives current planning. The technical challenges are immense. Europa's radiation environment is lethal. Jupiter's powerful magnetic field traps charged particles that bombard the moon's surface. Any lander would need heavy shielding, limiting how long it could operate. Drilling through potentially tens of kilometers of ice requires enormous energy, autonomous operation without real-time control from Earth, and mechanisms that can handle unknown ice compositions and temperatures. The submarine would need to function in complete darkness, navigate without GPS, avoid obstacles it can't see, and operate for months or years on limited power. These aren't problems that stop the mission, they're engineering challenges that get solved incrementally. Each generation of spacecraft pushes capabilities further. Perseverance on Mars carries instruments that earlier rovers couldn't have supported. Webb's mirror deployment, unfolding in space with terrifying precision, seemed impossible when first proposed. Technology advances, budgets fluctuate, but the trajectory points toward increasingly ambitious missions. Finding microbial life would answer one question while raising dozens more. How did it originate? Does it use DNA or something else? How does it metabolize? What's its evolutionary history? Is it related to Earth life, perhaps spread between planets by meteorite impacts, or did it arise independently? That last question matters enormously. One genesis of life in our solar system could be coincidence. Two independent origins would suggest life emerges readily when conditions permit, implying a universe full of biology. Panspermia, the idea that life spreads between worlds, has scientific support. Meteorite impacts can blast rocks into space, some eventually landing on other planets. We've found Martian meteorites on Earth, rocks blasted off Mars by ancient impacts that drifted through space before falling here. The journey takes thousands to millions of years, but microbes can survive that, forming dormant spores that withstand radiation, vacuum, and extreme cold. If Earth life originated on Mars and spread here, or vice versa, finding Martian microbes might reveal them as distant cousins of terrestrial organisms, sharing the same genetic code and basic chemistry. Testing that hypothesis requires sequencing alien DNA, if it has DNA, comparing its structure to Earth life, looking for similarities too specific to arise independently. If Martian life uses the same 20 amino acids, the same 4 DNA bases, the same genetic code translating base sequences into proteins, common origin becomes likely. If it uses completely different molecules, independent origin is certain. The philosophical implications cut deep. Religions and philosophies have grappled with humanity's place in the universe for millennia. Finding alien life, even microbes, shifts those discussions fundamentally. We're not unique. Biology isn't a miracle exclusive to Earth. The universe makes life when given the chance. That realization could unify humanity around our shared biological heritage or fracture existing worldviews that place humans at creation's center. History suggests both reactions will occur simultaneously, different cultures and belief systems integrating the discovery in different ways. Science fiction imagined first contact as dramatic, flying saucers landing on the White House lawn, radio signals from distant civilizations. Reality will probably be subtler, a chemical signature in an exoplanet's atmosphere that takes years to confirm, a microscopic fossil in a Martian rock sample, organic molecules in Europa's ocean that match no known geological process. The discovery won't happen in a single moment but through accumulating evidence that gradually shifts scientific consensus. And what if we find nothing? What if Mars is sterile, Europa's ocean empty, Titan's lakes purely chemical? That answer matters too. It would suggest life requires specific conditions we haven't identified, that Earth's combination of factors is rarer than we thought. The galaxy might be full of potentially habitable worlds that remain lifeless because some crucial ingredient is missing. That wouldn't prove we're alone, the universe is too vast, but it would suggest loneliness is possible, that consciousness might be precious because it's rare. Current evidence leans toward optimism. Organic molecules appear everywhere we look, in comets, asteroids, interstellar clouds, even in the atmospheres of planets where they can't support life. The chemistry of life isn't exotic, it's common. Water, once thought rare, turns out to be abundant, locked in ice throughout the outer solar system, probably common around other stars. Energy sources exist anywhere geology stays active, tidal heating, radioactive decay, chemical gradients between different rock types. The ingredients are widespread. Whether those ingredients combine into biology remains unknown. Chemistry becoming life requires specific conditions we're still mapping. Temperature must fall within certain ranges, too cold and reactions stop, too hot and complex molecules break apart. Liquid solvent is necessary, probably water though alternatives might work. Energy flow must be steady and sustained, not sporadic bursts. Concentration mechanisms need to exist, ways to accumulate organic molecules rather than letting them drift apart. Protection from radiation helps, thick atmospheres or deep waters shielding fragile chemistry from destructive high-energy particles. Earth had all those conditions simultaneously. Did Mars? Does Europa? The uncertainty drives exploration. Every mission returns data that refines our understanding, narrows the possibilities, points toward where life is most likely. Negative results inform as much as positive ones, each sterile world teaching us what conditions aren't sufficient, helping identify what additional factors matter. The timeline stretches across decades. Sample return from Mars won't happen until the 2030s. Europa exploration extends into the 2040s. Titan submarine missions, if they happen, lie further still. Breakthrough discoveries could accelerate everything. Finding clear biosignatures might trigger urgent follow-up missions with compressed timelines. Or progress might slow, budgets tighten, priorities shift, and the search extends across centuries rather than decades. What remains certain is that the search continues. Humans seem compelled to explore, to push boundaries, to answer fundamental questions about existence. Whether life exists elsewhere ranks among the most fundamental questions we can ask. The answer, whatever it is, reshapes how we understand our place in the cosmos. Finding life beyond Earth wouldn't just answer whether we're alone. It would fundamentally alter how we understand biology itself. Every organism we've studied shares the same basic architecture, DNA, RNA, proteins built from the same 20 amino acids, cells with similar structures. That's not because it's the only way to build life, but because everything alive today descends from a single origin point roughly 4 billion years ago. We've been studying variations on one theme, different arrangements of the same fundamental components. A second genesis would show us an entirely different approach to the problem of being alive. Consider what Martian life might teach us. If we find microbes in subsurface water, the first question becomes how similar are they to Earth organisms? If they use DNA, That's either remarkable convergence, the same solution emerging independently, or evidence of panspermia, life transferring between planets on meteorites. Mars and Earth have exchanged material constantly throughout their histories. Impacts blast rocks into space, some eventually falling onto the other planet. Microbes could survive that journey, protected inside rock from radiation and vacuum. If Martian life shares our genetic code, we'd need to determine whether Mars seeded Earth, Earth seeded Mars, or both were seeded from elsewhere. The implications multiply rapidly. But what if Martian biochemistry is completely different? Different genetic molecules, different amino acids, different cellular organization. That would prove life isn't locked into one configuration, that chemistry can solve the problem of self-replication and metabolism through multiple pathways. Every difference would reveal something fundamental about what life requires versus what's merely one option among many. Does all life need cells, or could some function as connected networks without clear boundaries? Must genetic information be stored in double helix structures, or would other shapes work? The variations would illuminate the core principles underlying all biology. Europa's ocean presents different possibilities. If life exists there, it evolved in darkness, powered entirely by chemical energy rather than sunlight. Earth has similar ecosystems around hydrothermal vents, entire communities thriving on chemicals rather than photosynthesis. But those ecosystems exist alongside photosynthetic life, sharing the same ocean, likely descending from the same origin. Europa's life, if it exists, would have developed independently in perpetual darkness, never knowing sunlight, possibly never developing anything resembling plants. The food chain would start with chemosynthetic microbes and build upward from there. How complex could such an ecosystem become without the energy abundance photosynthesis provides? The energy question matters enormously. Photosynthesis captures sunlight directly, providing massive energy input that drives Earth's biosphere. Chemical energy from geology offers far less, limiting how much biomass an ecosystem can support. Europa's ocean might contain only sparse microbial populations scattered around hydrothermal vents, nothing more complex because insufficient energy exists for larger organisms. Or perhaps life there discovered methods we haven't imagined, ways to extract energy more efficiently, to build complexity with less power. The possibility remains open until we explore. Titan raises even stranger questions. If life exists in liquid methane at -179 Celsius, it's operating on completely alien chemistry. Water-based biochemistry relies on specific properties, how water molecules interact, how they dissolve certain substances while excluding others, how they remain liquid across a temperature range suitable for complex reactions. Methane has different properties entirely. Life using methane as solvent would need different molecules, different reaction pathways, different cellular structures if it has cells at all. We can barely imagine what such organisms would resemble. They might not even be recognizable as life initially, their metabolism so slow and alien that distinguishing biology from geology requires years of study. Each potential discovery carries philosophical weight beyond the scientific. Confirming we're not alone eliminates one form of cosmic loneliness while potentially introducing another. If we find only microbes, simple organisms that never evolved complexity, we'd know life exists elsewhere but might still be alone as conscious beings. The galaxy could be full of bacterial worlds but empty of minds, civilizations, anyone to communicate with. That's simultaneously comforting and isolating, proof that Earth isn't unique in hosting life but confirmation that intelligence might be. The cultural impact would unfold gradually rather than explosively. Religious traditions would adapt as they always have when new information emerges. Some would see divine creation extending beyond Earth, life as something the universe was designed to produce abundantly. Others might view Earth as specially blessed, the only place where God's creation achieved consciousness. Most would probably integrate the discovery into existing frameworks without fundamental disruption. History shows humans are remarkably good at absorbing paradigm shifts while maintaining continuity with the past. Science would transform more dramatically. Astrobiology would shift from theoretical to empirical, from asking whether life exists elsewhere to studying how it works. New fields would emerge: comparative biochemistry examining different solutions to biological problems, xenobiology studying organisms with fundamentally different architecture, ecosystems analysis comparing Earth communities to alien ones. The questions multiply endlessly. How does evolution operate under different conditions? Do alien ecosystems show similar patterns of predator-prey relationships, symbiosis, competition? Does complexity emerge through similar stages everywhere or follow completely different paths? The practical applications might surprise us. Studying organisms adapted to extreme conditions often reveals useful chemistry. Enzymes that function in boiling water now enable DNA analysis techniques. Microbes that survive intense radiation inform medical treatments. Life from Europa might show us how to operate in low-energy environments, relevant for long-duration space missions. Titan organisms could teach us chemistry that works at extremely low temperatures, potentially revolutionizing refrigeration technology or cryogenics. You can't predict what studying alien biology might enable because the discoveries themselves are unpredictable. Search strategies continue evolving. Next-generation telescopes will examine exoplanet atmospheres in unprecedented detail, looking for chemical signatures that suggest biological activity. Oxygen combined with methane creates disequilibrium. The gases react and destroy each other unless something continuously produces them. On Earth, that something is life, photosynthesis making oxygen while bacteria produce methane. Finding that combination around distant stars would warrant intense scrutiny, though geological processes might create similar patterns without biology. The challenge lies in distinguishing life from clever geology across light years of distance. Sample return missions represent the gold standard, bringing material back to Earth where sophisticated laboratories can analyze it thoroughly. Mars sample return launches in the early 2030s, collecting rocks that might contain fossilized microbes or chemical traces of ancient life. Those samples will be studied for decades, examined by technologies that don't exist yet, potentially preserving evidence until analysis methods become sophisticated enough to detect what we currently miss. The same rocks might be reanalyzed by scientists in 2100 using techniques we can't imagine today. The timeline for discovery remains uncertain. It could happen next year, a Martian rover stumbling across clear biosignatures in an unexpected location. Or it might take centuries, requiring technology we haven't developed, missions to destinations we haven't reached. The search itself drives progress regardless of outcome. Building instruments sensitive enough to detect alien life requires advancing multiple technologies simultaneously: better sensors, more sophisticated analysis techniques, improved propulsion for reaching distant targets. Those advances benefit other fields: medical diagnostics, materials science, environmental monitoring. The search pays dividends even before finding anything. What happens if we discover not just life but intelligence? That possibility seems remote within our solar system. No evidence suggests conscious beings exist on Mars or Europa. But the galaxy contains billions of stars, billions of planets, vast distances we've barely begun exploring. If intelligent life exists, we might detect their signals, radio transmissions, or other electromagnetic evidence of technological civilization. Or perhaps they're quiet, either deliberately avoiding detection or simply not broadcasting. The space between stars is enormous. Signals weaken with distance, making detection challenging even if civilizations exist. Contact with intelligent aliens would raise questions we can barely formulate. How do you communicate across species that evolved independently, with no shared reference points, possibly no common sensory experience? Mathematics might provide a starting point, universal patterns that any technological civilization would recognize. But moving beyond basic concepts to actual communication requires bridging gaps we've never encountered. Human languages evolve together, sharing deep structure even when superficially different. Alien languages might operate on completely different principles, perhaps not using sound at all, maybe based on chemical signals or visual patterns or something we haven't conceived. The discovery of any life beyond Earth, whether microbial or intelligent, simple or complex, would mark a threshold in human history. We'd become the species that learned we're not alone, that answered one of the oldest questions our ancestors asked while staring at stars. That knowledge changes how we see ourselves, our planet, our future. Earth stops being the only known living world and becomes one example among many. Our biology stops being the only known solution and becomes one variation on a theme that plays out across the cosmos. Right now, we exist in uncertainty, not knowing whether life is common or rare, whether the universe teems with biology or whether Earth represents a singular miracle. That uncertainty drives the search forward, propels missions to Mars and Europa and Titan, motivates scientists to keep looking, to keep testing, to keep pushing the boundaries of what we know. The answer waits somewhere in the data we haven't collected yet, in the samples we haven't analyzed, on the worlds we haven't explored. And every mission, every discovery, every new piece of evidence brings us closer to knowing whether we're alone or whether life, in all its strange and wonderful forms, fills the universe around us. The search for life beyond Earth isn't just about finding microbes in ice or detecting unusual gases in distant atmospheres. It's about understanding whether we're witnessing something unique or something inevitable. Every ocean world we study, every atmospheric anomaly we investigate, every chemical signature we can't explain adds weight to one side of that question. Ganymede orbits Jupiter exactly as it has for billions of years. Its ocean sloshes beneath ice that has remained frozen since the solar system's youth. The magnetic field deflects radiation. Tidal forces squeeze the interior rhythmically. None of this changes whether we look or not. The moon exists independent of our curiosity. But we look anyway because understanding Ganymede means understanding how common habitable environments might be throughout the galaxy. The missions we're planning won't answer everything. Europa Clipper will map the ice shell and search for plumes but won't drill through to the ocean. JUICE will orbit Ganymede and measure its gravitational field but won't deploy submarines into the depths. These are reconnaissance missions, gathering data that will inform what comes next. They'll tell us where the ice is thinnest, where tidal heating concentrates, where future landers should aim. They're setting the stage for something more ambitious, missions that actually touch these frozen surfaces and taste the oceans below. That next generation of exploration lies decades away. The technology needs refinement. The funding requires political will that fluctuates with election cycles. The engineering challenges demand solutions we're still developing. But the trajectory is clear. We're moving from observation to investigation, from studying these worlds from afar to directly sampling what they hide. Whether Ganymede harbors life remains unknown. The ocean exists, that's confirmed. Whether chemistry there crossed the threshold into biology depends on factors we're still mapping. But the possibility alone justifies the search. If life arose independently in Jupiter's system, if organisms exist that never saw sunlight, that evolved in darkness powered by tidal forces and chemical gradients, then the universe becomes fundamentally different than we imagined. Life stops being Earth's miracle and becomes the cosmos doing what it does when conditions align. And if we find nothing, If Ganymede's ocean proves sterile despite billions of years and all the necessary ingredients, that answer teaches us something too. It reveals that liquid water and chemical energy aren't sufficient, that life requires additional factors we haven't identified. The search continues either way, moving to Europa, to Enceladus, to Titan, to exoplanets around distant stars. Each investigation refines our understanding, narrows the possibilities, points toward where life is most likely. The answer exists whether we find it or not. Somewhere in the frozen depths of Ganymede, or the radiation-blasted ice of Europa, or the methane lakes of Titan, or the atmospheres of worlds we haven't reached yet, the universe has already decided whether biology emerges readily or remains vanishingly rare. Our searching doesn't change that reality. It only determines when we learn what's already true.